We have basically interviewed 27 of the past, of all 49 investigator track faculty. I've listened to 40 hours of interviews, and I'm going to tell you some of the take-homes that came out of that. We picked our talks today, 17 of them, so we're going to be busy. 14 of them are past trainees in the department, so they're pre-docs, postdoc fellows, and they're going to tell you about the science that they're now doing after they've left the Department of Genetics. And then we'll have 3 additional talks from faculty. The talks cover evolution and diversity, cancer and its treatments, neurobiology, male fertility, agriculture, and education of future scientists and physicians here in St. Louis. So we're really happy to have you here to listen to these kind of things. As I listened to all of these tapes, there were several themes that came out. First, the department, almost everyone mentioned that we're collegial, that we're collaborative, and that we're actually amazingly democratic. There was also that there was an amazing sharing of new ideas between and among faculty and students. There was a preponderance of people who consider themselves to be tool building, and we'll give you some examples of that. They were supportive of different experiences before people came to WashU. There was the importance of model organisms, and we have a lot of organisms that have been represented in the department. And then finally, there were a lot of people who were thinking about moving from single genes, which was what genetics did for a long time, to thinking about how you attack it from an organismal viewpoint. Because we're scientists, I'm going to give you a few pieces of data that support these themes. So Maynard Olson, his main theme was that he was a tool builder, and that he realized that there were questions he wanted to ask that were at the organismal level, that he needed tools to be able to do that. Phil Green is sort of a representative of someone who came from a very different background. He told us he always was a mathematician. He wanted to be a mathematician until he read molecular biology or the gene. And he was really grateful that WashU, a place that he thought the science was exciting, and they were willing to take a risk on a mathematician as a faculty member who had never taken a biology course. Ted Hansen is a little bit of an exception in this, because Ted was really working on very different questions. He was looking at how peptides bind to the MHC receptor and molecules, and as you'll hear, that work is continued on, as you'll hear in Will Gillanders' talk. But he also showed that there was amazing sharing, and he remembers a time in a faculty meeting when Maynard got up and gave his presentation of yaks, and Ted realized that he had saw and heard the beginning of genomics. Elaine Martis considered herself a tool builder. She thought it as a way to build robots for doing the Human Genome Project, and then to take those tools on to looking at cancer genomes. Joe Dockery, one of the newer members of the department, was interested in questions about psychology and autism, but he realized that the tools that psychologists had weren't good enough, so he decided he wanted genetics, but he realized that genomics could be the force multiplier. And then finally, Gary Patty was a faculty member who did metabolism. And at the time that Gary joined our department, metabolomics was not really a thing. And Gary saw that he really wanted to think about metabolism in the same way that people thought about genetics and genomics. He wanted to be able to do metabolomics on a whole genome set. And he realized when he interviewed at WashU, the one out of 7 places that he interviewed, that it was the only one that people got his idea. He said that WashU saw the forest through the trees. And so as you listen today, I hope you really love the past of the genetics department, the present of the genetics department, and I hope you can envision the science that might happen in the next 10 years, maybe the next 25 years of the Department of Genetics at WashU. I turn it over to my co-chair.
Thank you, everybody. And so to begin with, I'd like to give a big thank you to the Planning Committee, without which today could not have happened. And a few quick logistical notes. So the Planning Committee is going to be doing the introductions of the speakers. Now, because today is weaving together the research, history, and lived experiences of this department, we have a lot to fit in. So the intros are going to be very short. The full-length introductions you can find in the program, so if you want to know more, look there. This does also mean that we only have 3 minutes total for questions for each speaker. So please keep your questions as succinct as you possibly can, and we'll be very mindful of the time throughout. And lastly, there are signs showing you where to go for the restroom and for the couch building where we'll be having lunch so that you don't have to wander.
So our first speaker of the morning is Swathi Arur from MD Anderson in Houston, and she was a postdoctoral fellow with Tim Shadle.
Thank you so much, Susan, and to the whole planning committee for inviting me here. It's such an honor and such pride to be standing here on the 50th celebration. I have to say, I did my PhD in India at the All India Institute of Medical Sciences, and I was not trained as a geneticist. I wasn't even trained as a basic scientist. I was in the Department of Pediatrics. So when I came to the US, I worked with the mass spec for like about a year to finish up some work that I was doing in India, and that was up at UConn. And then I wanted to really get into model systems because I came across C. elegans. And so I applied at WashU, and Tim was very kind to take on somebody who had never, ever looked at C. elegans, did not know the G of genetics, because my background was a little bit of cell biology and not much genetics at all. So the first thing I did, besides actually learning all of C. elegans from Tim, and you can tell how much we loved it, that people in the lab even put it on cookies. But what was really interesting was like, Tim said to me, okay, you really should take the advanced genetics classes. I was like, oh, okay, I'll go take the advanced genetics classes. So I was the only postdoc in a room full of graduate students who took advanced genetics. And I have to say, it was overwhelming. But the good thing was I didn't have to take the exams. I was just auditing the class. But what it really, I think, impressed upon me was the rigor with which every single person came to the class, every single faculty member took the time to explain what they were doing. I met Mark there, Jim, Tim, and everyone really spent time to explain the concepts. I would go to them afterwards, and pretty much everyone took time out. And that, I think, set the foundation for what I was going to do for pretty much the rest of my career. So in many ways, I consider WashU as my academic home. I think this is where I really became who I'm continuing to be. And what was really cool in Tim's lab wasn't just all the biology we were doing, we do germ cells, and that was really cool. Of course, Tim is super rigorous, and that continued through my entire postdoctoral training. So I was in the lab from 2003 until the end of 2009, and so it was a really, I think, solid time to learn everything. But what I loved about the 8th floor of the McDonald building, which is where I was right across from us was Susan's lab. And I remember the number of times when she would walk through, start talking to us, and she would look at me and go, what? That didn't make any sense. And we would go, okay, I'm going to go think about it. And that really set up this culture for everyone collectively coming together to help you think. It wasn't just a place where I learned to do experiments or learned genetics or took classes. It was a place where I really started to grow up as a scientist, and I learned to think about science. I learned to think about how to ask the right kind of questions. I really started to think about, why should I do this? Like, why does it matter? I can do it, but why should I do it? And that was really fundamental, I think, to my growth as a scientist. The other thing that I started to realize was how much each one of them was intentional about taking the time out to talk to postdocs, to talk to graduate students. And so by the time I went through my 6 years, I feel like, I had started to really get a sense of how to do this. Maybe I could go run my own lab. So I wanted to go hit the job market in 2008 when everything collapsed. It seems to happen recurrently. And I had to write this research proposal. So I started to write a research proposal. And until then, I had only written with Tim, which is very different. When you write, students and postdocs will know this. When you write with an advisor, you kind of have a parent who's kind of helping you write. So I was going to try, and Tim said, okay, you should learn to write on your own because this is what you have to do. It's a career in writing. You really have to learn how to write. So I said, okay. So I started to write, and Jim, who looks very proud here with his trout, he actually spent a lot of time reading some of the stuff, and he was like, "You don't know how to write." And I was like, He in fact told me, and Jim is known by everyone to be very kind, but he actually told me, "You're walking backwards in circles." And I was like, okay, this is going to be fun. So, so now I had to learn how to write. And this was what was really, I think, remarkable is this was coming from somebody who was a friend and a mentor. So you could take this kind of feedback that would seem pretty harsh, but was always well-intentioned. People took the time out to read your stuff and give you that feedback. And that feedback actually helped, he even helped me out with my first grant. In fact, I owe my first grant to Jim because I think he really helped me get it. But together, I think what all of this started to, I think what was really amazing for me was how, what I thought as a postdoc at the time was this came naturally to people at WashU. Everyone stopped, they talked to you, they gave you, they just engaged with your research. And then I started my own lab. I looked for a culture like this one where everybody was collegial. And when I got to my own lab, and I basically began to realize, well, it took a lot of intention from a lot of people to want to come together, to build this culture and to keep it going. And it was it felt like it was baked in the ethos of this place and how it, and that's something I've tried to move forward where I am. We try very hard to have the afternoon teas, which was started on the 8th floor of the, I don't know if Mark remembers, but we used to have these teas on the 8th floor of the McDonald buildings where students and postdocs used to come together and talk. So we try to do that a lot where I am, trying to build that culture, which clearly takes a lot of good people with a lot of good intention to keep going. So that's something I'm trying to move forward that I've taken beyond the science, is the culture from here and the collaborativeness. And of course, I also have a candy drawer. So I don't know how many of you know, but Tim's lab always had a candy drawer because at 3 o'clock, it was the place to go when you needed that extra fuel to think about a good scientific question. It was also a good place for people to come meet with us. I remember Mike Nonne used to come hang out because there was a candy drawer. So now I have that. And it actually seeded a lot of good collaborations because we ended up working with Mike. So anyway, so with all of that, I have a huge amount of pride in the fact that I come from here, from Tim's lab and from this department. And thank you for giving me, for inviting me today, and happy birthday. And with that, I'm going to share with you a story today that really starts from my work in Tim's lab, and we're continuing to use C. elegans as a model system where we study oocyte development. And I'll share how some of this work on oocyte development led us to a potential biomarker for cancer detection and metastasis in humans. So much of what my lab's been interested in and studying for the last 15 years is really how oocytes, which are specified in utero, in humans, how are these oocytes specified? Because in humans, they're of course specified in utero. By the time a female child is born, these oocytes are arrested in meiosis I, and they stay arrested through pretty much puberty, where they undergo meiotic maturation, and then after that, every month when they undergo ovulation until fertilization, where you get a progeny. So what work in my lab has really been interested in studying how do these oocytes get specified, what is the impact of nutrition, of maternal nutrition and diet on the specification and formation of oocytes, on meiosis I progression, and we've been trying to link some of these events to meiotic maturation and onset of embryogenesis. And of course, the model system we use, which I think is the coolest model system that I learned here, is C. elegans. So right here, for people who might be unfamiliar, that's the head and that's the tail. And these are the two U-shaped gonad arms that are right in the center. And if I zoom into one of these gonad arms, what you'll notice are these progenitor zone cells, which host a population of germline stem cells that differentiate and go through meiosis and produce oocytes pretty much through entire adulthood, which makes it, I think, a remarkable model system to study events of oocyte formation, production, meiosis I arrest, and that's coupled with meiosis II and fertilization in C. elegans. So it's really, I think, a one-stop shop for everything you want to know about oogenesis, which makes it a super powerful system. And the real reason I think I joined Tim's lab was because years ago, David Greenstein, in collaboration with Tim, had identified that active ERK has this very striking spatial pattern. So total ERK is everywhere in this gonad arm, but active ERK has this very striking pattern. And it was striking because, I mean, it's striking because it is cool looking. But the real reason I was interested is because this is a syncytial germline. And so when I interviewed with Tim, one of the major things I wanted to really try and figure out is what causes those patterns to form. I can tell you we don't know the answer, right? But what I did end up joining the lab to figure out was all of the processes that ERK regulates. And one of the first papers that we published from my own lab was we figured out that presence of food engages the insulin receptor and insulin signaling to mediate ERK activation only in meiosis 1. So this is mediated through insulin signaling, and David Greenstein's lab had shown that the ERK activation in the arrested oocytes is mediated through sperm signal, which has a short range hormone signaling. And of course, what's really cool, I think, about the germline is we can dissect out these gonad arms, fix and stain them. And I apologize to everyone who has green color blindness. I only noticed last night that my slides weren't fixed for color. So what you're looking at here in blue is the nuclei, but what I want you to appreciate is these oocytes that can be visualized by this green marker. And here you're looking at a staining for active ERK, which you just basically dissect out these animals, throw the antibody on, and you can visualize active ERK in these two striking patterns. And these are the kinds of micrographs I'll show you throughout, where I'll pretty much be pointing the progenitor zone of the stem cells to my left and the oocytes to the right of the screen. And the screen that I had done in Tim's lab was really to identify all of the the processes that ERK regulates, but also the downstream targets of ERK. And one of these targets that I pulled out from the screen was dicer. So this is the small non-coding RNA biogenesis enzyme, very well known for its role in generating siRNAs and microRNAs. And when I started my own lab, one of the first questions we asked is, what does phosphorylated dicer do? And we had an antibody to phosphodicer that I had generated as one of the last things I had done here at WashU. And back then, this was really cool because what you're looking at in green here is the pattern for phosphorylated dicer antibody. And what you'll notice is phosphorylated dicer is both in the nucleus and in the nucleoplasm. And this was very striking when we first saw this almost 12 years ago, because at that time, no one had described a pattern of localization for dicer in the nucleus, because it's known to function in the cytoplasm as a biogenesis enzyme for non-coding RNAs. And so we asked, okay, so what does phosphorylated dicer do? So we generated animals that did not, where we removed the phosphorylation sites. And what we found was in the absence of phosphorylation, the germlines don't develop. So you don't get oocyte formation. What you end up getting are these giant holes in the middle of meiosis 1. These are pachytene arrested. I mean, they basically are pachytene cells, but you get these giant holes. And the oocytes don't form. They're disorganized. And these animals are completely sterile, right? So it turns out that phosphorylation of dicer is essential for formation of, for progression of meiosis 1 and formation of oocytes. So what does phosphorylated epitope actually do? And what it does is we found it doesn't affect microRNAs. So we did some small RNA sequencing, and we did this in collaboration with Andy and Fire in Stanford. And what we found was that there was no impact on microRNAs, but instead the phosphorylated mutants don't process this class of endo-siRNAs called 26G endo-siRNAs. These are germline for the most part, and they're 26 base pair long versus the standard siRNAs, which are 21, and start with a guanine. And what you're looking at in pink there are the maternal RNAs, and I'm just mapping the null on the y-axis here with wild type, where you can see the maternal RNAs are formed, whereas in the that are targeted by the siRNAs, whereas in the phosphomimetic they're not. And what is striking about dicer is that it's phosphorylated in that meiosis 1 arrested oocyte, but right before transition to embryogenesis, this oocyte undergoes meiotic maturation, which is characterized by a nuclear translocation of the oocyte to what will be the future anterior of the embryo. And you also have a striking dephosphorylation of active ERK, and you lose phosphorylated dicer. And turns out that this switching off or dephosphorylation of dicer is essential for formation of embryogenesis. So if you don't have this, you actually don't form embryos. And so basically what we found was that phosphorylated dicer gets phosphorylated, gets translocated to the nucleus, and also stays in the nucleoplasm and a little bit in the cytoplasm, but it largely does not generate 26G endo-siRNAs. And we think that's because it has to protect maternal RNAs. So these 26G endo-siRNAs target maternal RNAs and degrade them. And in the C. elegans germline, transcription stops right about there. So pretty much all of oocyte development is transcriptionally silent, very much, which is also true in mammals. And turns out that what you really need to do is protect these maternal RNAs, and if you don't, you have lots of bad things happening. But just as you're about to undergo fertilization and form embryogenesis, what you really need to do is dephosphorylate dicer. So now it's back in the cytoplasm where it can target and degrade those maternal RNAs. And that's important for activation of embryonic genome, at least that's what we think, and causes a lot of, and remodels the maternal RNAs. So this was basically what we, this reprogramming function is what's essential for during oocyte to embryo transition in C. elegans. But one of the features of the screen that I had done in Tim's lab was we had looked for conservation. So I had actually painfully aligned each of the potential targets, and that alignment had shown me that dicer was conserved, or at least its C-terminal tail was conserved at the sequence and in the presence of the two phosphorylated sites. So right on top here, you're looking at C. elegans, and right at the bottom, there is human. So we made we basically mutated the two potential sites on dicer in human dicer, and we did kinase assays, and we found that this was not phosphorylated when we mutated them. So then we made an antibody to human form of phosphorylated dicer. We made monoclonals to each of these sites. And Raisa was an MD PhD student who joined my lab and wanted to know, is dicer phosphorylated in humans? And she was interested in cancers. So here she's looking at a KRAS-dependent non-small cell lung carcinoma. And what I really need you to focus on, if I go in here, are these brown dots. So these are the nuclear staining of phosphorylated dicer in this non-small cell lung carcinoma. And she did this whole tissue microarray and found that phosphorylated dicer was associated with presence of KRAS and EGFR mutations in human cancers. So then we asked the question, does phosphorylated dicer drive tumor progression? And we decided to use mouse. Now, I was not a mouse geneticist, but remember, I was very intentional about finding a department very much like the one I've had here at WashU, because on my own, I'm not intelligent enough to do much. So I needed a department that was just as collegial. And Gigi Lozano had recruited me as a faculty. She's a chair of the department and a mouse geneticist. She does P53 genetics. And she said, we'll help you make the mouse. So they actually made the mouse, and we had a joint student who was very brave to join a C. elegans and a mouse lab and did a joint project with us, and we ended up generating the phosphodicer mouse. And it was very nice because this was around the time that CRISPRs came along, and we basically ended up making this with CRISPR. And turns out that phosphomimetic dicer is small, and the animal has other hypermetabolic phenotypes, and I'm going to speed up. So, so on its own, we could not analyze these animals. So we took the heterozygous animals, which are largely wild type, and we crossed them into a heterozygous KRAS G12D allele, which on its own gives you these lung tumors that you can see here, but you get no metastatic tumor nodules. But presence of one copy of heterozygous dicer, phosphomimetic dicer, causes these tumors to grow larger and metastasize. So here you're looking at these tumors in the heart. So we zoomed into these tumors at the primary site, and we asked what's causing these tumors to metastasize. And here you're looking at a single cell sequencing data set from tumor epithelial alveolar type 2 cells. So these are all tumor cells. And what we noticed was something very unexpected. What we found was these genes that were turning on, which were gastrointestinal in lineage. So here you're looking at the gastric gene, GKN2, and it was there both in the phosphomimetic dicer, but also in the KRAS tumors. So I came from the show me state, so I didn't believe it. I thought we did something wrong. So we zoomed right in, and we did some fish experiments on these tumors. And in purple, you're looking at GKN2, which is the gastric gene, and in green, you're looking at prosurfactant, which is the marker for the alveolar cells. And what you notice here are cells that are double positive for both the green and the purple, and those are the cells that are taking on a new identity. But then there are cells that have completely switched over to just the GKN2. So these cells are undergoing lineage plasticity and transdifferentiation. So if I cartoon that out here, what we're seeing is tumor cells that are taking on a dual identity and then transitioning completely to gastric. So how does phosphorylated dicer do this? Well, it doesn't process microRNAs in tumors either, but it does seem to associate with opening the chromatin. So here you're looking at an ATAC-seq of the GKN2 locus, where you see that in the KRAS dicer double mutants, you see this nice peak, whereas you don't really see much of that in KRAS, although there's a little bit. We went back up to human tumors and we asked, well, do you see the same change in lineage plasticity and transdifferentiation? And this was one of the human tumors that was very aggressive. It's an invasive adenocarcinoma, and turns out that it has both phosphorylated dicer and a lot of it, as well as the purple cells that are GKN2. So with that, what I would like to propose is, and what we've been now planning to do next, is use phosphorylated dicer as a potential biomarker. Okay, so it's a biomarker. There we go. So, all right, that's my lab. I'm going to shut up there. Sorry about that. I went over. I'm so sorry.
I think we thanked Swathi for an amazingly great talk, and you should grab her at the coffee break to learn more and ask your questions.
Thank you.
Our next speaker is John McCutcheon. He's at the Arizona State University and is a Howard Hughes Memorial Institute Fellow, and he was a pre-doctoral fellow with Sean Eddy.
All right, is that good? Yep. Yeah, it's on, okay. Yes.
Go.
Oh, thank you. 20 minutes. All right. Thanks, everybody. It's really an honor to be here. Thank you for inviting me. So I was a graduate student 2000, 2006. So my perspective, what I'll say on the next slide, is from that of a graduate student. But I want to tell you a little bit about what we do. So we study bacteria that form long-term beneficial infections inside of host cells. We mostly work in insect systems, as you'll see. But I think about this as sort of a three-stage progression. I'll tell you about one of those steps today, some of our work on that. The green cell is a free-living bacterium, so that's something in the environment. This gets captured or infects a eukaryotic cell. If it stays there, if the host requires it, it sort of moves into this blue state, which is a long-term infection. The end process of this are the mitochondria and the plastids, right, which are just long-term beneficial bacterial infections. So my lab studies this process. So when I was thinking about what I wanted to say, and I'm going to try not to go on too long, but again, I was a graduate student, and graduate students need different things from graduate school. I needed a lot of things. So I was raw, right? And I'm really appreciative. So I learned a lot of technology here, which helped my career and still does. So that part's all good. But really when I think about what this place means to me, it's the excellence, the expectation of excellence from the faculty. They, I would say, beat that into me. And when if I think about one thing, so there's lots of examples, and I won't tell you what they are, but I failed my qualifying exam. I had some just knockdown, drag-out committee meetings. Sorry about that. But the student talks were really good. That was one of my favorite things about the department. And I got a ton of feedback, which I still use, and I still tell my students now. So I really appreciate many of you in this room pulling me aside and saying that wasn't good, it needs to be better, that wasn't, this wasn't good, that wasn't good. I really appreciate that. But the one place you never wanted to be was in your second slide, and a hand would come up from the front, from a person in this room right now, and they would say, "Why should we care?" Which means you're not explaining your stuff. You need to motivate this. You need to really think about what you're going to say in your talk. And I really appreciate that. I needed that, and this is not a question you usually got asked twice. So the progression of a free-living bacterium to something like a mitochondrion is pretty well established in terms of the steps that it takes. It's pretty well stereotyped, it turns out. So this is just a drawing of a free-living bacterium in a eukaryotic cell. Upon establishment of an endosymbiosis or an intracellular infection, if that organism becomes an intracellular pathogen, say, like Mycoplasma genitalium, it will tend to lose genes. The bacterial genome will get smaller. So you'll see pathogens in this state, and you'll see host beneficial infections in this state. If the host cell or organism requires that infection to survive, like the things that I work on, this will progress from left to right. So that bacterial genome will get much smaller. Occasionally, you'll see horizontal gene transfers, other bacterial genes transferred to the nuclear genome. Those will be imported into the bacterium, to the endosymbiont. So this is many of the things that we study. We study this state. We study the biochemical and cell biological integration of these bacteria into their host cells. And of course, the end point is something like a mitochondrion, where you have lots of horizontal gene transfer. You have lots of protein import into the compartment that allows that compartment to function. So again, this is the progression that we study. I'm going to tell you about some of the work we've done here at the very beginning of the infection. Okay, so these are two model systems. We're trying, this cicada will never be a model. It's terrible. It lives underground for most of its life. But it has two bacteria that live inside of special cells, a red bacteria and a green bacteria. The red one makes 2 of the 10 essential amino acids. The green one makes 8 of the 10 essential amino acids. It is a sap-feeding insect. You can see the piercing, sucking mouth part there. So it only sucks sap. You can't grow an animal eating sap. It's too nutrient poor. So you need these essential amino acids to supplement the diet of the insect. I'm not going to talk any more about that, but this one here, this is what we have in the lab. We can grow those little tiny mealybugs in the lab on little potatoes or on little pumpkins. And here is one insect cell. That's the nucleus. This darker blob here is a beta proteobacterium. And then living inside of that beta proteobacterium are gamma proteobacteria. So it's a bacterium inside of a bacteria inside of an insect cell symbiosis. And again, these two bacteria work together to make essential amino acids that the animal can't make on its own. And so these inner bacteria are going to be the feature, the bacteria that I'm working on or talking about today, these orange ones. So if you look, this is just a tree of gamma proteobacteria. So Salmonella and E. coli, Yersinia are there boxed in red. Those are things you probably know. What you don't know or what's in blue here, this is a group of bacterium called Sodalus. It is the most interesting group of bacteria. And if you look at not just one, but in this case, this is a paper where we looked at 9 or so mealybug species, different mealybugs, you will see that the inner bacterium is different in every case. The inner bacterium has been kicked out and replaced by a different bacteria, which forms a really complicated symbiosis with this whole system. But every time a bacterium is kicked out and replaced, it's always replaced by a Sodalus. And this is over 100 million years of evolution. So Sodalus is repeatedly infecting these insects. If you look at insects more broadly, so this is 400 million years of evolution, you can see Sodalus has repeatedly, over time, over 400 million years, infected insects and become these intracellular infections, long-term intracellular infections. And so this, to me, is really interesting, right? E. coli is not forming these interactions. Sodalus is over and over and over again over 400 million years. And these Sodalus genomes are really similar to each other. So from comparative work, you know Sodalus is important. But you don't know how it works because you don't have the free-living antecedent. And sometimes science works. Well, sometimes you get lucky. I didn't do anything. Colin Dale at the University of Utah found, accidentally, because a guy impaled himself with a branch, and he had this infection that wouldn't clear, and that infection was Sodalus. But it was a free-living version, very closely related to these insect endosymbionts. And it's called Sodalus precaptivus. So just super lucky, weird breakthrough. So we can grow Sodalus, this free-living version. And since it's so similar, we can use it. The way we're thinking about it is, okay, if we infect naive hosts, what does it do? How does it behave as an intracellular resident? Right? Something about it is good at becoming these types of beneficial infections. If you look at the genome, it looks like a path. If you're a bacteriologist and I showed you this, you'd say, oh, it's some sort of plant or animal associate or pathogen or something like that. It has 3 type 3 secretion systems, so little machines that pump effector proteins into host cells. It has it has quorum sensing, so it can sense its environment and change its pathogenicity gene expression. It has 2 component systems, so it can sense its environment and change its transcription. It looks like a bacterium, right? And we can do genetics. We're doing whole genome screens on it. We can make any color we want. We have complete control over it, which is not typical for us, because I've been working in fields for 15 years where we had no genetics. Couldn't do anything. And some of these bacteria that we work on have 150 genes. Can't grow them in a plate, can't do anything. So we want to know what Sodalis does inside of cells. And normally, if you're, say, a macrophage, and you phagocytose a bacterium, normally what happens is this. It enters the endocytic pathway, and that plasma membrane gets remodeled into an early endosome. The details don't matter. It's decorated with Rab5. That early endosome is remodeled into a late endosome. It's decorated with Rab7. That late endosome fuses with a lysosome, becomes a phagolysosome, which has different decorations, and that destroys the bacterium. This is what's supposed to happen when a bacterium is phagocytosed inside of a cell. Any eukaryotic. This is a eukaryotic cell trait. Pathogens use things like I'm telling you that Sodalis has, type 3 secretion systems, all this sorts of stuff, to deflect from the phagolysosome formation, right? So it can either remodel that vacuole to become some sort of hidden part of the cell where it can grow and replicate, or it can escape the vacuole itself and replicate in the cytoplasm. So pathogens want to avoid progressing down the endocytic pathway because that's where they're destroyed. So the weird thing about what we're studying here is that we don't know what the phenotype's going to be, right? We know from comparative work that this thing's important. We know because we accidentally found it out in nature that it's close and it behaves in interesting ways, or it might at least compared to other pathogenic bacteria. But the problem is that most intracellular bacteria, including the first one that was actually studied, this is the paper describing basically the beginning of the germ theory from Pasteur, which was in insects. Most bacteria that cause infections give you a phenotype, right? That's how they're found. You don't study pathogenic bacteria for another reason. You study them because they give you a phenotype. In this case, sick caterpillars. And I just want to point out that we don't know what to expect in terms of phenotype because I'm telling you maybe it's a pathogen, but maybe not because it's often ending up as a beneficial infection. But I'm wasting time, but I do want to point out that Pasteur was pretty clever. He's pretty good. This is a drawing of the thing he thought was making the caterpillar sick. We now know it's Serratia marcescens. That's an EM of Serratia marcescens. It looks exactly like his drawing. I just find that unbelievable. So I keep it in there even though it's not really worth making the point. So right now we're using Drosophila cells and Drosophila animals as our model naive host. And we are asking what happens if we infect Sodalis into Drosophila. So I'm going to show you just a little bit of data from Drosophila cells. So what does it do, if anything? Is it destroyed? Does it become a vacuole resident? Does it become a cytoplasmic resident? So we know that it gets phagocytosed by S2. This is very boring. We know that it enters the endocytic pathway. That's what it's supposed to do. So there it is at the top, surrounded by Rab7. So Rab7 vacuole, that's what it's supposed to do. And there, we know it's in a vacuole because we can do EM. But after 24 hours or so, we rarely see, here's a Sodalis, a little cluster of bacterial cells. Here's a phagolysosome. We rarely see it in phagolysosomes. So we know right away, just from these simple experiments, that it's deflecting itself away from phagolysosome formation. It's protecting itself in some way. And then all sorts of things happen later. So the phenotype, right? We don't know what to expect, but what I can tell you in flies and in fly cells is that the infection forms and it stays for very long periods of time. And it doesn't really make the animal that sick, but it forms this massive infection. And I don't know, oh yeah, there we go. So here's 5 days later. This is an S2, you can't see the cell, but that's an S2 cell. Those are bacteria in a vacuole just swimming like crazy after 5 days. Nutrients should be gone. So it's deflected in the phagolysosome. It has remodeled its vacuole well enough that it can get nutrients and keep swimming. Here it is cruising across the nucleus. I don't know if you can see that. That cell is probably not doing well. But in general, Drosophila cells that are infected don't care. They just keep dividing. If they have bacterium, the bacterium gets provisioned to the mother or the daughter cells, and it just keeps going. So it's kind of the phenotype we expect. No one cares, but the infection stays. So we think that's really cool, actually. Okay, so I'm going to end, but I'm going to try to explain one more time why I think this is interesting. So what are the possible outcomes of having a bacterial infection, right? So there's sort of two easy ones. The host wins. Let's say this is a generic macrophage and it phagocytoses a bacterium. The bacterium is destroyed and the host wins, right? That's one outcome. The other outcome is that it's a pathogenic bacteria. It's some sort of intracellular pathogen, and it can do funny business in the cell, and the pathogen wins, right? That's the other outcome you expect from an infection. The cool thing about these kind of beneficial symbioses is that that's not what we see. We see, I think, the beginning of the infection looks a lot like a pathogen, and that makes sense because pathogens don't get destroyed by their host cells. And so these things don't want to be destroyed by their host cells, but they also don't want to kill their host cell. So the beginning looks like a pathogen, but then something happens, which we're trying to understand, and you end up with these long-term infections that are host beneficial and that don't kill the endosymbiont. And I think that's a really weird and cool outcome. Paul does all our bacterial genetics. Philip did all the genomics early on. Carla does all our cell infections. Adam, who's joint with Norbert Perriman at Harvard, does all the fly infections. And Dalton does the EM. Thanks for listening. Yeah, Brock. I don't know how much time I have.
John, thanks for the epic committee meetings. They were really some of my favorite memories of this place.
Yeah.
The
I wish you
question
could have
is-
all been there.
Yeah, no, it was terrific. How do you know it was repeated infection? Is the phylogeny of the bacteria very discordant with the phylogeny of the
I
of the
skipped
bug?
over all that stuff. We thought about that really carefully. So we know it's repeated infection because if you look at the phylogeny of the insect and you look at the phylogeny of the bacteria, they're all mixed up. The bacterial genomes, we can tell they're all Pseudolus, but the different Pseudolus genomes are clearly of different ages. Some of them are 4 megabases, so a recent infection. Some of them are 0.3 megabases, so a very old infection. And so we can sort of time it. And we have this idea that some of them are old, some of them have been replaced more recently. So we can kind of see it from the phylogeny and the size of the genome. Yeah, Lucia?
Thank you. My question is, have you ever seen this something happen to vertebrates?
No, so things with adaptive immunity don't do this too often. I think so, but I don't know. But in general, no. Yeah, thank you. Yeah.
Thanks for the beautiful talk. Is there a threshold required before a parasite can become an endosymbiont? Like, does it have to be not too lethal?
Yeah, well, that's what we don't know. We have one example.
Oh, I see.
Right? So you can, the thing I like about Pseudolus is that we're not forcing anything. So you can force anything to happen with strong enough selection in the lab, right? You can take E. coli, you can jam it into yeast, and you can make something happen but with a very strong selection. Here, we have no selection. We're just introducing the bacterium and the host to each other, and we see these long-term infections happen, right? Except in mutants. I didn't tell you about any of the mutants, but if we make certain mutants of the bacterium, we'll have mutants that will be killed by the host in a fly, but will completely destroy cells in a dish. So it's really interesting, but we can talk more about that. But yeah, so we have one example of something that is naturally this way. Most things end up as one of the two outcomes I told you before. The host wins or the pathogen wins. But this is the only example that I know of that doesn't do that.
Since you keep calling it an infection,
sorry.
It's detrimental to the host for sure, always.
Infection, I use infection and symbiosis agnostically.
Okay.
And this is a definitional thing, but I don't know what else to call something inside of something else. So I call it an infection, yeah. Yeah, I like using words that, yeah. And then it makes people kind of angry, so infection biology get a little bit angry when I say it, which is also a benefit.
Yeah. So you say that those insects are getting repeatedly infected, right? So it has to be in their diet?
Yeah,
I'm wondering, so is it in the phloem?
We think it's in trees.
So it's interesting because aphids, the different colors in aphids, they've gotten gene transfer cross domains, right?
Yes,
From
yes.
the fungi into the aphids, and so
Here too.
So does it live in the cells of the plant as well, or is it in the phloem?
We don't know, and this is something that I've been trying to. Actually, I have a student coming who is willing, it's called the dead stick microbiome. So she's gonna drive around the Southeast and collect sticks and try to sequence, because that's where it's come up twice. It's come up in two different people, 2 different times someone impaled himself with a dead stick. And the result of that infection, because the stick was still there, luckily, it wasn't, the bacterium's not super nasty, but it's been recovered twice from people sticking themselves with dead sticks. So we need to look at dead sticks. But I think that's where it is. Yeah. I don't know how.
You have a minute now. With the,
Do you see a limit?
Oh, sorry, I meant you're next, but no, yeah.
okay. With these repeated infections, do you know how they are competing with each other?
No, and we're trying to set it up in the lab right now, so we can sort of destabilize one of the players through careful antibiotic use, and then just expose it to the saliva. It hasn't worked yet. It tends to infect them, but not on the right cells, so we don't know why. But yeah, I think there must be in evolutionary time, a very fast competition. I don't know in sort of ecological or human time. Yeah, can we do one more?
Do you see a limit on the number of infections of a single cell in this Drosophila system, and how do you see the passage to the daughter cells?
Yeah, the daughter cells. So it's the infection dynamics are complicated, but in general, there is a limit. These clusters of cells that are swimming get to be about the size I showed you, and it's about 50 bacteria. There's also many cells that are infected in the cytoplasm where they're just swimming around. And when those mother cells divide, the bacteria just go and they replicate. If there's a big ball, if there's just one, only one of the daughters get it. But if there's 2, they tend to be divided between mother and daughter. So it does persist for. I mean, we've grown these things for months and they're still infected, totally happy. We need to watch individual cells more carefully over time. We haven't done that yet. Thank you.
Our next speaker is Dr. Chris Hettinger. He's currently a professor of genetics from University of Wisconsin, and he did his postdoc training with Dr. Mark Johnston in the department.
All right, thank you very much for that introduction, and I want to thank really the entire organizing committee for putting on such a great event. It is a true honor and privilege to be back here to share science and memories with all of these people who made such a tremendous impact on my career. And so I'm going to dive in with some science to start off with. I see that my title was turned in a few months ago and has a verboten word on it about the genomic making of yeast metabolic diversity. But I really want to start by framing just how much diversity we're talking about here, because it's easy to underestimate it. So across the budding yeast subphylum Saccharomycotina, we're talking about really as much diversity as in the entire animal kingdom, and really quite a bit more than the entire plant kingdom. And so yeasts, of course, are heterotrophs, right? And so they have to take advantage of the energy and the carbon that has been fixed by plants. And so this is really important to how they partition their ecology. There wouldn't be so many different species of yeasts if they were all doing exactly the same thing. And so they see the world as kind of a menu of possible things that they could eat. And they make different strategic decisions about the order in which to eat things. They have different capabilities in terms of which carbon sources they can exploit and process to get energy and build building blocks that they need. And then they also make different decisions about what they do with that. Of course, this department in particular has done a lot of amazing work on Saccharomyces cerevisiae and has also appreciated the products of aggressive fermentation from time to time. That's actually an evolutionarily derived trait. Most yeasts actually don't do that. If given oxygen availability, they will actually make other decisions in how to manage their energy budget and make other products. And I'm not going to talk a lot about it today, but that is the basis of some of our more applied projects in the lab, where we try to understand how to push and manipulate the carbon budget of yeasts to make either delicious beverages, coated in brown there, or sustainable biofuels and bioproducts. So a few years ago, we set out to build a grand genotype-phenotype map across the budding yeast subphylum. So this is covering more than 400 million years of evolution. We called it the Y1000 Plus project because there are more than 1,000 species of yeasts that are known, and we also knew that we were discovering more with our undergraduates out in the field. And that started in Clete Kurtzman's lab at the USDA in Peoria, Illinois, and he runs one of the curated collections where people deposit strains when they describe a new species. When you describe a new species, you have to deposit it in 2 different countries, and this is one of the main ones for the US. Everything had to be revived, came up I-39, and then now resides a copy of it in our freezer at UW-Madison. We then applied Illumina sequencing to generate genomes for every known taxonomic type strain and also a few others, and did high throughput phenotyping, quantitative growth curves, and when it's running, we can do nearly 5,000 experiments at a time. And then the idea was we were going to do some computational magic in collaboration with Antonis Rokas at Vanderbilt and analyze this massive data set. And of course, reviewers often pointed out that we didn't really have a clear plan on how to do that, which is why it probably took several times to get the grant funded. But I knew it would work, and I knew that because of my time at WashU. And so here is a slide resurrected from my faculty job talk. The question here doesn't matter that much, although it's an interesting question. But you can see sort of the naive idea was that we were going to generate a bunch of sequencing data with this new Selexa platform then, and then we were going to use it to answer our question. And of course, I double-clicked on my file. My idea was that I would need computers and coffee, but when I double-clicked on my file, I got a spinning wheel of death, and that's when I realized I needed to learn programming from all of the people. So I audited Barack in Rob's course. Some of you were TAs in that course and helped me figure out how to get out of this, and kind of the rest is history. And of course, I then moved with Mark to Colorado. You can see here we are getting ready for lab meeting. And of course, I was not the first person to think about building a yeast genotype-phenotype map. And indeed, you know, a lot of the yeast genome was sequenced here in Mark Johnston's lab. And when I tell my students that as they were finishing the yeast genome, they were already knocking out every gene to do high-throughput phenotyping, to build that genotype-phenotype map, I mean, they honestly don't even believe it. Mark got scooped on this. This MIT group published a few genomes, but Mark then, with Paul Clifton, actually, I think, showed in some ways, a little bit of a better roadmap because they included some more diverse yeast. And so there's actually not a lot of diversity in the Kellis paper because they just focused on the genus Saccharomyces. And so you can already see a footprint here of a broader framework about what kinds of things you can learn by going deeper into the phylogeny. And then we also worked early on with JGI on some biotechnologically important yeasts, kind of filling things out. And you can start to see what a yeast genotype-phenotype map looks like. You have genes that are associated with traits. So we published the first tranche of data in 2018. Here there's 332 species. There are 12 major clades across 400 million years, and I'll just orient you to kind of some of the common models. It's actually a little easier to see on this slide than some of the later ones. So there's a genus Saccharomyces, our model system, and delicious generator of hybrids and other products. Whole genome duplication was about 100 million years ago, and so that's also been a really important model. If there's one other yeast you've heard of, it's probably Canada albicans, which I know Victoria worked on in Mark's lab. There's an independent evolution of aggressive aerobic fermentation in the Brettanomyces, also gives us some sour beers. And then there are also some biotechnologically important traits for our bioenergy research as a clade of xylose fermenting yeast, and then a bunch of oleaginous yeasts that make lots of fatty acids. We also have had the privilege and excitement of getting to describe new species discovered by our undergraduates, and sometimes we have fun with it. We've named a species for Bucky Badger, and of course for the Packers. Not capital P, lowercase p, we don't want to get sued. And we've learned a few things. I'm just going to skip the data. You'll have to trust me. I've shown you that yeasts are extremely diverse. But from that study, we learned that there were trait syndromes, and that a lot of carbon metabolism traits actually go together and govern metabolism. We also learned over the years that gene and trait losses are actually major factors in diversification. And that really leads to kind of an interesting contradiction. How can loss lead to all of this diversity? And another way of thinking about that is really what are the consequences of having these interconnected carbon metabolism pathways? And so almost a year ago now, we published the big data set with essentially every known yeast genome sequence, 1154, 24 quantitative growth conditions in a new environmental ontology. And I'm just going to acknowledge there is a lot of information on this slide. And actually part of the point of it is that there's actually not much pattern. So if we come out and look at the ecology, the isolation environments, there's no pattern. That's because yeasts are evolving in parallel, convergently, over and over again into different things. Yeast ecology is very dynamic. Even if we go to the next wheel out and bin things based on the number of carbon sources they use, in blue we have generalists that eat a lot of things, in pink we have specialists that eat few things, you can see there are some clusters of blue and pink in that wheel, but not actually a lot. And that turns out to be the saving grace here, is that because yeasts are evolving into specialists and generalists over and over again in parallel, and if we looked for any individual trait, they're going back and forth, that gives us a lot of power to not have to correct for phylogenetic signal, although we do when we can, and actually test the hypotheses of what genes are underneath which traits. So the first thing we wanted to do, having binned things into generalists and specialists, was my postdoc, Dana Apolente, was trained as an ecologist. She really wanted to ask this classic question about whether there were trade-offs. Are specialists growing faster, better in the carbon sources they pick? We tested that hypothesis in a couple different ways, but I'll just tell you the answer is no. Generalists are actually growing better across the board. So they grow, generalists actually grow faster. They're in the faster category generally. This is true even if you only analyze the carbon sources where a specialist is actually growing at its fastest point. Indeed, there is also no case in the data set where a specialist is the fastest grower. So generalists are benefiting from having highly connected metabolism. We can also ask whether there's a trade-off with nitrogen, and again, we see the exact opposite. Nitrogen generalists tend to be carbon generalists. So because there's not a lot of phylogenetic signal in the data, we can actually just apply machine learning in order to ask whether there are features in the genome that allow us to classify yeasts as generalists or specialists. And so here's what the kind of data set looks like in rough form. We've got 2050 K orthologs or KOs, and this has mainly been done with presence-absence data, although we're starting to play with copy number variation and other approaches. And you can see here, we use random forest to build a bunch of decision trees and then see how they perform. You make 100 things that kind of look like this, and you kind of go down, do you have KO1, yes or no? Do you have KO2, yes or no? And then when you get to the end, you are able to make a decision, and you can see how well it does. So it does great. I also acknowledge the first time I saw a ROC curve, or receiver operating characteristic curve, was here at WashU. And you can see this is exactly what we want. We want this upside down hockey stick. It means that our model is getting things right, in this case more than 90% of the time, and we're well above that red diagonal. On the other side is a confusion matrix, and again, this is looking at our predictions versus the ground truth, and again, what we want is the bulk of data along that diagonal in this case. So there's a lot of information in the genome about whether a yeast is a generalist or specialist. One of the other nice things about random forest is it's really easy to ask what the most informative features are. And so at the very top of the list is complex 1. This is the part of the electron transport chain. It's been lost in Saccharomyces and its ancestors. It turns out that actually in many other yeasts it's been lost in parallel, which is why it shows up. So having complex 1 makes you more likely to be a generalist. Beta-mannose also makes a bit of sense. Yeasts put a lot of mannose into their cell walls. It's also a common carbon source, so being able to break down chains of mannose seems also to make you tend to be a generalist. The fourth highest one is actually carnitine biosynthesis, and this is really interesting because carnitine is used to kickstart respiratory metabolism, and this is something that is really highly variable across yeasts, how aggressively they're able to respire and how they make that transition. And then finally, having a really connected pentose phosphate pathway also is highly predictive of whether you're a generalist or a specialist, and that makes my DOE funders happy because it tells us how to work on getting flux through this pathway, which is really famously difficult. So to share a couple take-homes here, generalists are growing more robustly than specialists, and we can distinguish specific pathways that actually allow us to make predictions about whether they're going to be a generalist or a specialist, and we think that that is telling us that they have better connected parts of metabolism and are able to more robustly manage their carbon flux. I'm not going to show you the data for time constraints, although I have backup slides if anybody wants to take the bait and ask about it. But they got that way, generalists got that way by losing fewer traits, fewer genes, and actually gaining a few traits in some cases, which also are interesting. And then finally, generalists have more genes. And we can actually tell you a generalist has on average, for every carbon source that they use, they have on average 36 more genes. That is not just carbon metabolism genes, it's genes in general. So they're also bringing along managers, transcription factors, transporters, and other things. Okay, I want to tell one final vignette, and again, this is of course in honor of Mark and his work on the GAL pathway. It wasn't just a technology in genome sequencing. I remember learning about the GAL pathway, the galactose utilization pathway, when I was an undergraduate. You know, it's still in the textbook that I teach from at Wisconsin, and it's hard to really understate how important this was in crystallizing our understanding of how eukaryotic molecular biology worked. So I had, of course, been following that work very closely, and here's Mark's famous review. Here's the actual biochemical pathway. And so, of course, we looked. Galactose is one of the carbon sources we tested, and we get it right about 94% of the time if we just look for the GAL genes, the enzymes that are required to process galactose. And you might say 94% is pretty good for high throughput data, right? High throughput, low quality. Victoria remembers the song there, right? But we knew that in 1 or 2% of the cases where there was a mismatch between whether they had genes encoding the GAL enzymes and were or weren't able to use galactose, we had verified in some of those cases there were false positives and false negatives. So in some of the cases, we knew that there was a mismatch. Well, here's machine learning to the rescue. Again, we make a model, we get upside down hockey stick, that's great. We look at our predictive features, okay, GAL1, GAL7, those are the enzymes. That's kind of a sanity check. But the thing that popped up here is the ability to use galactitol. So here we're actually not just using genes, we're also incorporating traits. And it turns out whether you can use galactitol or not was highly predictive. But galactitol is a sugar alcohol modified version of galactose, and it turns out there's actually a pathway that had been worked out a few years ago in filamentous fungi that's an oxidoreductive pathway instead of the kinetic pathway that you see in bacteria and humans and yeast and textbooks. And so we wondered if maybe some yeasts that could use galactose without a GAL pathway, maybe they have an oxidoreductive pathway. So in the initial paper, Emily Ubalode, my graduate student, verified that we could detect that biochemical activity. She's now going on to do RNA-seq. This is on a relative of Candida auris that can use galactose without a GAL pathway. Apparently, when I made the slide, I was thinking more about bioenergy, and it says xylose versus glucose, but it's galactose versus glucose. But I think you don't need WashU level of sophistication to analyze this data set. Here's the log-fold change and the number of counts. And boy, look, there are some serious outliers there. And not only that, they're in a gene cluster. Emily is working on knocking this out right now, but I think I would not bet against this cluster for anything. So these are the most highly expressed genes. They have the right annotations, and not only that, in that cluster is a transcription factor homologous to GAL4. So with that, that's just one example coming out of the project in the lab. We're really excited about using this genotype-phenotype map to really figure out novel biology that has been missed by just studying model organisms. With that, I want to acknowledge all the people that did the work. They've got asterisks or yellow if they contributed slightly more than others, especially Dana and Dana Apulente and John Walters from my group, and then Marie-Claire Harrison and Abby Labella from Antonis Rokas' group. And I will have a few minutes for questions. Yeah? Yeah, I mean, growing on more carbon sources, growing faster. So the quantitative data that we looked at was maximum growth rate. Yeah, that's a great question. We haven't done direct competitions between species. It's something that we proposed in one iteration of the grant that didn't get funded. I think it's actually a really good idea. Reviewers did not. But I think, and I think you're also raising kind of an interesting caveat here, right? That we've tested this at 1 temperature, 1 pH, laboratory medium, right? And out in nature, they're experiencing all kinds of different things. They're in exotic environments. And so there are, of course, you know, the limitations that a specialist could be a specialist for a reason that isn't related just to carbon metabolism. It could be an interaction between carbon metabolism and something else in the environment. Could be another organism that they're interacting with. And so, you know, there's kind of a limitation to how we frame the question and define things in order to make it answerable in the lab. Yeah.
So my question is, like, in the nature, are journalists more likely to be found in more complicated environments? Are they, or like finding environments that have more different carbon sources?
Yeah, that is a great question, and it's a real limitation, I think, of how yeast ecology has traditionally been approached. So one thing to note about the study, because we focused on taxonomic type strains, right, is that we only have one representative from each species, really, in the data set. That may or may not be representative of the species as a whole and all the possible places that it could be. One caveat is that, you know, most yeast species have only been seen once or twice by the people who isolated and discovered it. So in a lot of cases, we're not going to be able to go in more depth, but in some cases, we might be able to. The other limitation is, one of the things I glossed over is before those 12 major clades that were color-coded are now officially taxonomic orders. But that only happened very recently, working with our taxonomist collaborators to apply these phylogenomic methods to reclassify things. Having that reclassified is actually going to enable microbial ecologists to now actually parse out their analysis and data sets in a way that is more informative than Canada SP and Saccharomyces SP.
Okay, thank you.
Okay, very exciting. Next up, we have James McCarter, who is an MD, PhD graduate of WashU. He did his thesis work in Tim Shadle's lab, and he also founded the Young Scientist program while he was here. He is currently, if I got this right, managing director of Biogenerator Ventures in St. Louis.
Great, thank you so much. Thanks, Susan, for the invitation to speak. And so, it sounds like the microphone's picking up okay? Okay, excellent. Yeah, great. So I have the opportunity to share with you what has really been a collaborative project around many of us over the last 34 years, and that is helping to provide an opportunity for students from disadvantaged backgrounds to get some exposure to science and science education, and then for those who are interested to pursue a career in science. And so I'd like to, in a second I'll pose this, is to get a show of hands if during your time at Washington University, have you either volunteered with a teaching team or as a tutor or as a mentor or hosted a student in your lab and any kind of connection with science outreach? Please raise your hand. Yeah, so I think it's about 2/3 of the audience has had some connection with this. And so this is really a collective project of all of us, and I just get to be the messenger to kind of share the story and to give you kind of an update on what's been going on with the Young Scientist program over the last 34 years. So there are 3 components to the Young Scientist program. There are other things that the program does as well, but I'll just highlight these 3. Summer Focus is a paid 8-week summer internship for high school juniors, rising seniors. Teaching teams are outreach within the schools to provide hands-on demonstration of science and doing scientific experiments where kids actually get to conduct those experiments in the classroom. And then third, continuing mentoring is a more recent program that allows a connection over all 4 years of high school, 9th through 12th grade, of having a mentor from Washington University spending time with those students on a frequent basis. And so this is a science, technology, education, and math STEM outreach program, and it's really run by graduate student volunteers, also postdocs and staff and faculty involved as well, but really the graduate students are the real driver of the activities. And its mission is to engage the next generation of diverse scientists from St. Louis through hands-on research and a one-on-one mentoring between K through 12 students and active scientists. And so back in 1991, I arrived in 1990, and with some of my MD, PhD classmates, we started kind of talking about what our experience was like landing in St. Louis. And one of the things that we kind of saw in our daily lives was that there was really a disconnect between the broader St. Louis community, particularly folks from disadvantaged backgrounds that we were seeing in our daily lives, and the amazing work that was going on here within the School of Medicine and within the university more broadly. It was almost like two separate worlds. And there were outreach programs run by medical students, but there was not much in the way of active outreach by graduate students. And so we began to address that with 2 students in 1991 and then 5 students in 1992 with a summer program to allow those students to come in and spend 8 weeks embedded in a lab with a graduate student or a postdoc, actually conducting research, taking a course on how to actually conduct research, how to prepare for college, how to write up their results, how to present their results, sort of giving them sort of a full, fast experience of what it's like to have a career in science. And so the statistics for that over the last 34 years, we're now close to nearly 500 students have come through that summer program. And the majority come from the St. Louis City Public Schools, another sliver from other under-resourced schools, and then about a third come from the better-resourced schools in St. Louis County. And you can see we kind of have grown that by the decade. We're about halfway through the current decade and on track to kind of be similar to our statistics from the last decade. And all of the students that have responded to our follow-up and we do outreach to figure out where students have gone, all of those students have gone on to college. Now, I wanted to give you kind of one vignette of a particularly remarkable student. After running the Young Scientist program in the early 90s, I then was a tutor and then had the chance to be a mentor. And within Tim Shadle's lab, we were able to recruit Bart Bartlett. He grew up on the north side of St. Louis, went to Metro High School, and Bart spent the summer with us, actually stayed with us then in the lab for a few years and published a couple of papers in C. elegans. Bart then went on to a full scholarship at Washington University in chemistry, specializing in inorganic chemistry, PhD at MIT, postdoc at Caltech, and now he's the chairman of chemistry at the University of Michigan. So just super proud of Bart. Had a chance to see him last fall and meet his 2 young sons when my son and I were doing a college tour. So, kind of a remarkable example of launching a scientific career. Teaching teams then is really driven by graduate student interest in taking things that they've observed in their own scientific education and creating modules that you can actually then go and teach those units within the schools. So the students bring to the school and work with the teachers to teach, anatomy, physiology, forensics, genetics, genomics, all kinds of different topics. And the primary ones are more than anything else in the area of genetics and genomics and neuroscience. These, in the last decade, have been nearly 200 events that have reached nearly 15,000 students. One of the things I'll mention in here was just the impact of COVID. And so the COVID pandemic really ended for a few years the summer internships, in-person summer internships. There were some remote internships, but that's not the same. And then also the teaching team stopped. And so we're really in the recovery mode of getting this going again. You can see in purple, that's what's kind of happened post-COVID, and it's still relatively small to the scale that we were at prior to the pandemic. And then continuing mentoring, this got started I think around 2016. One of the things that's been really amazing about the Young Scientist program is that because it is student-led, and it's supported by the Division of Biology and Biomedical Sciences, and it's supported by the faculty, including Jim Skeeth, who's been the faculty sponsor for the program for many years. But it's really the students who take the initiative. And so it allows them to kind of recreate and reinvent the program every few years in terms of how it's delivered and what the components of the Young Scientist program are. And so continuing mentoring, I think, was about the third or fourth kind of attempt to find a way to have this impact with continuity over a student's high school career, and giving them exposure to what science is and what a career in science is like early in their high school career, then helping them through the steps that it would take to actually get into a college program in the sciences, and then helping them through the college application process. And so this began at Solden International School in 2016, and then expanded to Collegiate School of Medicine and Bioscience, and then more recently, in just the last few years, at Roosevelt High School. And so that's reached 228 students as of to date are part of that. So what I wanted to turn to now was not just what is the impact of the Young Scientist program on the high school students who participate, but what is actually the impact on the graduate students who are leading that program? What impact did that have on their career? What's it like while you're a graduate student trying to write do your thesis experiments and write your thesis, that you're also running a 100+ person volunteer program? And so I, over the last few weeks, have done interviews with 6 of the student leaders from the last 2 decades. And so I wanted to share a little bit about their experiences and how it impacted their career trajectory. So Mariah Beck was the head of the Young Scientist program in 2005 and 6. She's now a professor at Wichita State University. And she mentioned that this was her first graduate student publication, was actually outcomes from the Young Scientist program that she published in Science. She went on then to co-found the Association for Women in Science WashU chapter that was very active during the time of the AAAS meeting in St. Louis. And then as a professor, she's then created a first-year STEM research program for undergraduates at Wichita State that has 25 students annually are having a research experience. And she's modeled the research skills course that she teaches to those students based on the research skills course that she created for Young Scientist program. Stephanie Rodriguez was the director of the Young Scientist program from 2012 to 14. And her whole career now, for the last decade since she left WashU, has been around science education and science policy related to workforce, including a time at AAAS, and then at the US Department of Labor, and now back at AAAS again, where she's the head of the STEM Opportunity Alliance. And she mentioned the conversations that she was having with the WashU administration was really critical in her career. It showed her the importance of having a seat at the table, and then led to what she's doing now to try to have a broader impact on science education and science careers nationally. Molly Gibson was head of YSP in 2014-15, and she is now with Flagship Venture Capital, part of their Flagship Pioneering Group, where she's the origination partner. And so she does kind of what I'm doing now, which is to create new biotechnology companies. And she's co-founded 2 AI-based biotech startups in the last couple of years. And, but she mentioned that sort of problem solving in resource-constrained environments is kind of what she did managing this large volunteer organization on a shoestring budget. And then that's kind of what she's trying to do now within the context of helping to start new fledgling companies. So Raika Jayasinghe was also in 2014 timeframe, and she's now a research assistant professor at WashU. And she mentioned that when she applied to WashU, the connection of the graduate students to the city and the broader St. Louis community was really eye-opening to her, and she didn't see that at the other universities to which she was applying, and that's part of what attracted her to Washington University. And so YSP was kind of on her radar from the beginning. And she described the volunteers as being kind of a small inspiration to those high school students to see that, that this is maybe something that they could do. And she learned the supervisory skills, mentorship skills, collaboration skills that she now uses to help run a large genomics lab here at WashU. So Boima Adu Apong was YSP director 2015 to 17, and she's now a director of computational biology with Terran Biosciences. And she said that the Young Scientist Program and being the leader of YSP was just really pivotal in her career, how to manage her time between the demands of her PhD work along with running the Young Scientist Program. She then went on to co-found a nonprofit called Show Me the World with several YSP teachers in the St. Louis public schools that provides international study abroad opportunities to St. Louis City public school students. And she said the connection of the Young Scientist Program Advisory Board really was a launch pad for her career. Barry Goldman on that board connected her to opportunities at Monsanto and at Pluton. And she said she used those skills that she used in running the Young Scientist Program led her to take on leadership roles and directorship roles at an early stage in her career in industry. And then Celine St. Pierre, who's defending her PhD thesis next week. She is the first Young Scientist Program director to have actually been a YSP student when she was in high school. And so she was a YSP scholar in the summer of 2009. And I'd like to really thank Celine for leading the Young Scientist Program through the tumultuous period of COVID and keeping the program alive. You can see that she was director for 4 years, which is not the usual tenure, and helped to really kind of rebuild the program post-COVID, and also published a paper on student outcomes and advances in physiological education. So that gives you a sense kind of how running a large volunteer organization while being a graduate student is something that's been meaningful to these 6 directors. And then to myself as well. So I had this experience early in graduate school of running the Young Scientist Program, and that gave me the confidence to go on to be involved in biotech startups. I started Divergence in my last year of graduate school, and thanks to Bob Waterston, who let me spend 20% of my time on the company while I was a postdoctoral fellow at the Genome Sequencing Center. We got Divergence off the ground, ended up selling the company about a decade later. Got involved in another startup called Verta Health in the area of metabolic disease, where I led clinical research and clinical operations, and really used those skills that had come from the Young Scientist Program in terms of leading those startup organizations as well as other corporate roles. And then 6 months ago, I took on leadership of Biogenerator Ventures. We're the startup arm of the nonprofit BioSTL, which fosters the innovation ecosystem in St. Louis. And so Biogenerator has, over the last 15 years, helped start 134 biotech companies in St. Louis, about 3/4 of those in human health, about 1/4 of those in agricultural technology, and about half of those companies are WashU spinouts. Either a WashU founder or a WashU license is at the heart of those companies. And we've then, that $50 million that we've put in has been resulted in thousands of jobs and $2.8 billion in follow-on investment from other sources. And so, what I'm trying to do now, in the same way of mentoring young scientists now, is to mentor the next generation of scientific entrepreneurs in St. Louis. And so I can tie a lot of that back to kind of those learnings from 34 years ago, starting the Young Scientist Program. And then a huge thank you to, I think it was, 2/3 or 3/4 of you raised your hands. So thank you all so much. For a special shout out, just mentioning 2 people, Jim Skeeth, who's really kept the program, alive and kicking for the last decade. And then to Alyssa Labonte, who's the current director of the Young Scientist Program, and who's responsible for many of these slides. And thank you very much.
That was a great talk. I can't help but ask the elephant in the room question. Is your program at all reliant on federal funding, and is it now under threat?
Yeah, so the Young Scientist Program is, I fund it myself for the summer interns, for my family does, and then Washington University, it's coming out of various budgets with sources within the university, and then we have a small endowment, and we have annual charitable contributions. So we don't have any federal support at the current time.
So it shouldn't be. It shouldn't be under threat.
It's vulnerable in the same way that all of our hosting labs for the summer, all of our graduate students who are running the so it's vulnerable in the way that everything within the university is vulnerable. But we don't have a large federal grant that's supporting the program. We were lucky early on to get support from HHMI. We had Howard Hughes funding for, I think, 8 years when they used to support K through 12 science education. And then when they stepped away from that, there was no really other source to step in. Yeah. And I don't know if Alyssa's here, or if you want to answer kind of if you want to answer with the current funding, is there a mic that we can give you?
We currently get a lot of funding from local grants. So we have a finance director who applies for all kinds of local St. Louis or Missouri-based grants to help the kiddos in our community. So no federal funding right now.
Thanks. I think there's some questions in the back there.
You showed really impressive data showing the number of students who participate in YSP going to college. I wondered if you had any data on how many persist in the sciences.
That's an excellent question. I'm going to have to rely on Alyssa again if we can get her a mic back, because I feel a little guilty being the sort of spokesperson up here for this, but she really knows the day-to-day.
We are working on that. Anecdotally, we have a lot of our students who continue in the sciences, lots of students who have gone on to get PhDs and go to graduate school or medical school, but I don't have any numbers on that right now. It's really hard to keep track of them once they lose their high school email accounts that they sign up with.
I just had a question about what does it take for a high school student to be successful in this program in terms of support from the community that they are from, besides the graduate student mentor? How is the program perceived in that community, and what sort of support systems have to be there for that particular student to be successful?
Yeah, maybe I'll start, and then Alyssa, if you want to chime in as well. But going back to the early 90s, having at least support within their family of the importance of education, and the belief that there was the opportunity to take on a scientific career. But in terms of recent years, if you want to just, and this will be the last comment.
Yeah, I think they have a lot of support from their teachers at school. They're the ones who encourage. We usually ask the teachers at the local high schools to select a few students who clearly show an interest but need that extra push. And so they have a lot of support from their schools as well as their families.
Thanks. Yeah, thank you. Thanks.
So we're going to have a short break. We'll see you back in here at about, I think, 10 after 11. So welcome back, everyone. The next session is actually going to have a little more emphasis on cancer. And our first talk will be Dr. Will Gillanders. He's an MD, PhD from Washington University. He worked as a fellow in Ted Hansen's lab, and he's now a professor in the Department of Surgery here at WashU. Thank you, Will.
All right, thank you very much, Susan, for that kind introduction, and I can't tell you how thrilled I am to be here today. It was a real formative experience for me to work in Dr. Hansen's laboratory. I look back very fondly on those years, and I'm very proud to represent the Hansen Laboratory. So I'll just say a few things about the Hansen Laboratory. You know, the main focus in the Hansen Laboratory was on MHC class 1 antigen presentation, and specifically how the MHC class 1 molecule interacts with antigenic peptides, beta-2M, CD8, the T cell receptor, and chaperone molecules. And this is just a schematic of the antigen presentation pathway, and you can see here that the intracellular proteins are broken down by the proteasome and then transported to the endoplasmic reticulum where they're loaded onto class 1 and then transported out to the cell surface where they can be recognized by T cells. And one of the unique things that the Hansen Laboratory discovered was a unique monoclonal antibody that recognizes a peptide receptive transition state of the MHC class 1. And this really allowed detailed studies of how MHC class 1 molecules are loaded with peptide and the role of chaperone molecules in this process. And these are really kind of pioneering studies. And they really spanned over decades. You know, the first study they published using this antibody was in the early 1990s. There was a series of studies. I was in the lab between 1993 and 1996, but there was a series of studies in the 1990s about MHC class 1 interaction with chaperone molecules. And ultimately, in 2012, they did a crystal structural analysis of MHC class 1 in this antibody, and they also mapped the epitope onto multiple other class 1 molecules, but they discovered that there's a conformation change after peptide interaction with class 1, and it results in this hinged door that closes to hide the 6437 epitope after peptide binding. And I personally was involved in studies looking at interaction of MHC class 1 peptide complex with T cell receptors. And literally we created, you know, a whole series of mutants in the MHC class 1 as seen here. And then we tested it against a whole series of T cell receptor or T cell clones and identified the most critical residues for interaction between the T cell receptor and the class 1 molecule, as kind of seen here on the crystal structure diagram. And so I took that passion for antigen presentation, and it had direct applications for the development of strategies to target cancer neoantigens. And so cancer neoantigens are expressed in cancers and are typically the result of genetic alterations. The majority of these cancer neoantigens are unique to individual tumors, and they were until recently relatively understudied because of the difficulty identifying these antigens. You know, what we've used is tumor normal exome sequencing to identify the genetic alterations, and then we have a whole series of bioinformatic pipelines to determine and predict which ones will be recognized by the immune system. And of course, you know, a huge shout out to the McDonald Genome Institute and more recently the Griffith brothers for helping with all of these studies, which without their support, they would never have been possible. And these vaccine strategies targeting cancer neoantigens have advantages over strategies targeting tumor-associated antigens like HER2 or mammoglobin. Targeting cancer neoantigens, since they're typically only expressed in the tumor, decreases the risk of autoimmunity. They're not limited by central mechanisms of self-tolerance. And because a lot of these genetic alterations that are targeted are cancer driver mutations, it limits antigen loss, which is a common tumor escape mechanism with other forms of less specific cancer immunotherapy. And so just kind of going back to this schematic, let me see if we have like a mutant protein, in this case, mutant PI3 kinase. It's, and this is like, how does the immune system recognize these cancer neoantigens? A lot of times people think that they're all cell surface antigens, but that's not correct. Actually, the majority of our intracellular proteins. And the question is, how does the immune system recognize these? Well, the mutant proteins, and this is like mutant PI3 kinase is a common driver mutation in breast cancer. They're broken down by the proteasome, and they go through this pathway that was so elegantly mapped out by Dr. Hansen and others in the 1990s, and then they ultimately make it to the cell surface, and they're presented by the class 1 molecules. And in this case, the mutation is illustrated here. The yellow is the antigenic peptide, which is typically 8 to 10 amino acids in length. And then that kind of is facing up towards the T cell receptor. And actually, it's an open question as to, which peptides are the most antigenic. Is it the peptides that are mutated at an anchor residue that allows them to bind to MHC class 1 molecule, or is it the peptides that have a mutation in the T cell facing parts of the peptide? So that's something that we're trying to use, machine learning algorithms and the database of different epitopes that we've identified so far to determine which would be best. Well, as a surgical oncologist and someone who specializes in breast cancer surgery, I was most interested in kind of trying to target these cancer neoantigens in triple-negative breast cancer. Triple-negative breast cancer is actually one of the subtypes of breast cancer. There's luminal breast cancers, HER2-driven breast cancers, and triple-negative breast cancer. But triple-negative breast cancer is the subtype that's most responsive to immune checkpoint inhibition. It's a mutationally complex breast cancer subtype. The tumor mutation burden is higher in these. And also, there's a lot of evidence to suggest that the presence of tumor-infiltrating lymphocytes, which are more common in TNBC, actually are associated with improved outcome in triple-negative breast cancer with like neoadjuvant chemotherapy and chemotherapy. So there's abundant evidence to suggest that the immune system was playing an important role in triple-negative breast cancer, and that's kind of why we prioritized it. In terms of our vision for clinical translation, this is an old slide, but we imagined that patients would be enrolled and they would have a research biopsy, and then we would perform tumor normal exome sequencing and tumor RNA sequencing. Then we would identify the genetic alterations and then use these computational pipelines in the PVAC tools suite for neoantigen identification and design a vector. And then actually for these DNA vaccines, which was our platform of choice for the first clinical trial that I'm discussing here, they were all produced in-house at the Biologic Therapy Core facility in 7 Southwest Tower. And we performed the manufacture and all the product release tests. And then they were administered to patients using an electroporation device, which dramatically improves antigen expression. And then kind of like in collaboration actually with Ted Hansen, our first studies were to develop and validate a polyepitope vector design. And I won't go into great detail about this, but we tested different ways to include these individual epitopes. Remember, we're trying to target up to 20 different epitopes in our personalized DNA vaccines. And so we have a polyepitope design, and we tested whether or not they should be concatenated with spacers or without spacers, and then leveraging kind of our insights into MHC class 1 antigen processing pathways. We also included a mutant ubiquitin at the beginning of the construct that basically immediately targeted this concatenamer to be degraded by the proteasome and improved the presentation of these different cancer neoantigens after DNA vaccination. And so in terms of our initial phase 1 clinical trial, the primary objective was safety and feasibility. A secondary objective was to assess the immune response using ELISpot and flow cytometry. And then we also performed TCR sequencing as another measure for the immune response. And just shown here is the clinical trial schema. These patients actually, a lot of phase 1 trials are done in patients with metastatic disease, but the FDA allowed us to perform this trial in patients who had neoadjuvant chemotherapy, but then persistent disease at the time of surgery. Those patients tend to have a very poor outcome. So they got neoadjuvant therapy, they had surgery, and then adjuvant radiotherapy. And during that time, we were able to perform the sequencing and vaccine design and manufacture. The vaccine was administered after all the standard of care treatments, and then we followed these patients. And like I said, we used, all these were sequenced in the clinical laboratory environment at the McDonald Genome Center, and the vaccine prediction was performed using the PEDVAC tools suite of software. And these were all reviewed at an immunogenomics tumor board that we've established, and then manufactured at WashU. And so just in terms of our immune monitoring protocol, ELISpot is a assay that detects interferon gamma production by T cells, so that we tested peripheral blood specimens before and after vaccination, and also for those neoantigens where we detected a response, we also tested flow cytometry for intracellular production of interferon gamma. And here's just kind of some representative ELISpot analysis results, which demonstrating, dramatic increases in some instances following vaccination. And then, not all of these neoantigens produce an immune response. So there's certainly room for improvement in our algorithms. About 20% of the neoantigens produced an immune response, but the ones that did produce an immune response, they were relative. They were specific. In this case, there was no activity against the wild type, but there was activity against the mutant version. And then just to kind of summarize, we saw results in immune responses in 14 out of the 18 patients that were treated. And like I said, not all of the neoantigens produced a response, probably between 20 and 25% of those. And actually, one of the surprising things that we did find is that although our algorithms really focused on CD8 targets, so class 1 binders, we saw a lot of responses to class 2. And so we were able to determine that on the flow cytometry using multicolor flow cytometry. And I think the class 2 responses are more promiscuous, class 1 responses are more specific. So again, a little bit of a surprise there, and we and others have observed this. We did do bulk TCR sequencing and saw a pretty dramatic expansion of specific TCR clonotypes, and ultimately we we expanded some of those cells, performed single cell sequencing, identified some of the putative clones from our T cell cultures, and then we're able to confirm that those putative T cell clones from our cultures were actually expanding dramatically in vivo, and then also we were able to clone those T cell sequences into a lentivirus and show that they were specific to the neoantigen of interest. And then, these vaccines were extremely well tolerated, mostly just grade 1 and grade 2 adverse events, like specifically, injection site reactions kind of were the most common, and then kind of like a flu-like symptoms were also common. And I guess one of the most exciting things is that we did ultimately compare the outcomes of these patients to our own historical controls. So this is this comes from a published paper by our medical oncology colleagues, where they looked at all of our patients with triple negative disease and persistent disease following chemotherapy. Like I said, that's known to be a very poor outcome, and our patients appeared to have a much better outcome. So certainly a very encouraging signal that these could be inducing a beneficial immune response. So in summary, we treated 18 patients. They completed 3 vaccinations. There really were no significant adverse events observed. The vaccination induced neoantigen-specific immune responses in 14 of 18 patients. And we saw improved progression-free survival compared to our own institutional and published historical controls. And we have a number of different things that we're kind of doing now to try and improve our vaccine. We've tested actually multiple different vaccine platforms, the DNA vaccine platform, synthetic long peptide vaccine platforms, and now we're working very hard to bring mRNA vaccines to WashU so we can test those. In addition to that, we're also looking at alternative sources of cancer neoantigens. To date, we've really focused on single nucleotide variants and indels, and we know that there's a whole other potential universe of alternative sources of neoantigens. So we're working again with the Genome Institute, doing long read sequencing, working with the Bursky Center, and doing immunoproteomic analyses and hoping to identify additional sources of neoantigens. And so, again, I'd like to recognize all the people who have helped, in my lab, also at the Genome Institute, and in medical oncology, and Bob Schreiber and the immunomonitoring laboratory. Thank you very much.
That was a great talk. In the middle of it, I sent a photograph of you to your sister, Liz, who, as you know, is a senior leader at the National Cancer Institute, and she insisted I ask you a hard question.
Okay.
So I've been trying to think of a hard question to ask, but Liz says hi, and she's very proud that you just gave this talk. But my serious question is, this is impressive work. If you fast forward 10 years from now, what's the vision for where this kind of therapy will be delivered? Is it always going to be in a major cancer center at a place like WashU, or do you envision this will find its way to routine community hospital or
I think that this field is moving very rapidly and has been very successful. And there's already been a randomized phase 2 trial in melanoma that showed that the clinical outcomes were improved. And so I think that this is gonna be like immune checkpoint inhibition, that there will be a lot of potential applications for this, and it will become, a standard of care treatment.
Oh, that's great.
All right. Thank you very much, everybody. Here it's making a noise.
back when I was thinking about this, you know.
Go ahead, I'm looking.
Okay, so James Allen is our next speaker. He carried out his PhD research in the late Steve Johnson's lab, where he was an HHMI Gilliam Fellow. He's now at Mass General, where he is an HHMI Hannah Gray Fellow. Since I have one more second, he was also the first ever student in the first ever cohort of the IMSD program. James.
Ever expect to be asked back here once I left, but shows you don't really know anything as a grad student. So anyway, I wanted to talk a little bit about the work I do. So when I was here, I worked in the lab of Steve Johnson on zebrafish, and after I left this place, I decided to go to Boston and work with David Langenau, where we are really interested in using the zebrafish model to understand and investigate pediatric cancers. And so I'm particularly focused on acute lymphoblastic leukemia, which, if you're not familiar, is a very commonly diagnosed pediatric cancer characterized by a developmental arrest of immune cell development. So these diseases present this way. And I say diseases because this is a recent paper from St. Jude, which basically did sequencing of 1,300 patients of T-ALL. And basically what you're seeing here is that even though we call these patients as having acute lymphoblastic leukemia, there's actually a surprising amount of diversity present in the patient population. So there's at least 15 different molecular subtypes, each with different genomic drivers, responses to therapies, and prognosis. And so quite paradoxically, at the same time, we've known for a while that Notch is a dominant oncogene in most subtypes. So about 60% of these patients, even in this UMAP space, will have some activating mutation in this pathway, which makes Notch a dominant oncogene in this disease. And so this kind of creates an interesting paradox because you have this remarkable amount of diversity seemingly driven by commonly converging oncogenic pathways. So what I think is one of the missing pieces is that we don't have a very complete understanding of the collaborating events that might occur along the development of the disease in order to explain the diversity that we see in the subtypes. So this is where I come in. So our lab uses zebrafish models of acute lymphoblastic leukemia. So we achieve this with thymic restricted expression of human oncogenes. So for example, c-Myc or Notch1, and then you can give the fish leukemia, which you can see here, the spreading of the blast from the thymus, which is shown here in a normal fish. So when you express the oncogene, the blasts that are present in the thymus will spread and invade the surrounding tissue. And this is a lethal condition, and they respond to therapies similarly to humans, such as radiation and targeted therapy. So we use this to model the disease and discover factors that might drive its aggressive behavior. And so my project, what I was really interested in, taking inspiration from conversations I had at WashU actually, is I wanted to develop a way to discover genetic conditions that might lead to the initiation of this disease. So this is very hard to study in a patient because it's called acute because there's no staging of the cancer. Once patient shows up, it's already progressed quite advanced. And so what I wanted to do is I wanted to use the zebrafish to discover collaborating genetic events that can initiate leukemia. And so just to be clear, what I did here is I took a large amount of candidate ALL oncogenes, plasmid DNA, and I linearized them and put them in a single mix and injected them into the zebrafish. And so this simple but a little bit chaotic, if you work in the zebrafish world approach, basically has a readout where because the integration of these linearized plasmid is relatively inefficient, what you're going to get is concatenameric integration of the genome, wherein each animal injected contains a different collection of the transgenes I have injected into the the fish. And so what I'm actually looking for here is a unique situation where the combinations present in that animal are sufficient enough to lead to the onset of leukemia. And so these candidates that I selected for this pilot study were chosen based on recurrent genomic alterations in relapsed patients. I figured this was the best way to hedge this bet. And so this worked actually quite well. And so what I'm showing you here is basically an example of a normal fish with H&E staining. So you can see the thymus, and then you can see normal gills and normal muscles. And so when I injected this fish and it rapidly developed a leukemia, which I could see with fluorescent microscopy, you can see that the blasts have spread from the thymus and they're infiltrating the entire animal, present throughout the entire body, including severe infiltration of the gills and muscle. And so I collected a large amount of these samples of fish. So this basically worked as a proof of principle. So I was able to make a mix of candidate oncogenes that can integrate into the fish, and something present in that pool is able to initiate the leukemia, but I don't really know what. So I sequenced all of these animals that I collected, and I tried to discover commonly co-segregating combinations of transgenes that are present across the fish. So again, because I said this is concatenameric integration, the idea here is that a lot of these, the makeup of the pool is going to integrate into the fish. I only really care about the ones that are commonly present in most of the fish that get the leukemia, so I can nominate candidates that are likely driving the cancer. And so I basically arrayed this out and looked for the transgene expression, and selected candidates that I could validate and work. And one thing I should note here, I told you that I selected candidates from T-cell patients. I actually ended up getting mostly B-cell leukemias, which was a bit of an uh-oh moment. So it's sort of an interesting and unexpected, but very exciting possibility, because it means that we can further model this disease with different presentations of the different subtypes. So anyway, so I validated a couple of these combinations. So the one that really stood out mostly was this proto-oncogene SET. So SET collaborates with both Notch and IL-7 to initiate the leukemia. So that made us think that most likely what's happening when I inject the pool is SET is collaborating with those genes to cause the leukemia formation. And I should say, I'm not showing it here, but all of the individual constructs, that is SET, Notch, and IL-7, are not sufficient to make leukemia. It's only when they're combined and you get a genetic synergy that you actually get the leukemia in the zebrafish. So I did this, and this was very exciting. So we discovered a novel role for SET in ALL initiation in an in vivo model. But I wanted to reach further than this, so because I'd only started with 10 genes, I decided to expand this to 66 and repeat the model, so the approach. And so when I did this worked out quite well. So in addition to leukemias, I got weird presentations of cancers that seemed more like sarcoma-like, things that were more thymoma, lymphomas, things like that, and weird skin cutaneous presentations of the cancer as well. So basically, this seemed to be a robust system that we could use to rapidly interrogate complex genetic systems using the zebrafish model with in vivo resolution. And so, again, when I sequenced all of these samples, in total in our study over 2 years, we collected 155 tumor samples. And so I'm showing you here that when we analyzed them with a principal component analysis, we were able to discover that we had actually identified 8 unique subtypes of samples, only one of which was MYC-driven, the model I showed you before. So not only were we making new models, they're distinctive from the one we had always used in the lab and the field had used entirely. And so that's just showing you the subtype of the cancers that we called. So this was very exciting for us. So we had made these new models that presumably we'd never seen before in the fish, and they were different from the one we'd always used for our experiments. So opens up a lot of possibilities for how we can actually use these models to design interesting experiments to understand this cancer. And so in addition to transcriptional differences that we observed through the UMAP spacing, I can appreciate that this might not be clear to people who don't spend a lot of time looking at fish histology, but this is a big wow moment for me, anyway. So basically, what I'm showing you here, G8 is the MIC model that I told you that our lab has used for decades. So basically, you see blasts that have severely infiltrated the body, including the gills and muscle. We found that some of the clusters mimic the MIC model, like G6, but some of the other ones had unique presentations. For example, some of them tended to infiltrate the gills but not the muscle, or the muscle and not the gills, or some of them actually were restricted, like G2 and G5, seemingly restricted to the skin and didn't infiltrate the body at all. So not only were we getting transcriptionally different, we could see histopathology that showed that actually these were different models that we were making. And so, again, we looked at all of them transcriptionally, and this is just showing you the results of the entire study. And so you can see, based on just the T and B markers that we use to call them as whether or not they originate from a T or B cell, you can see very distinctive clusters of gene expression patterns across the clusters. So we're very happy with that, and it's very interesting to look at like how we might use these. But we also use NMF to discover programs that might be active in these clusters to try to learn what is actually going on to drive the malignancy of these cells. And so I'm going to highlight one example of this. So I'm calling it Program 4. So it's mostly ribosomal biogenesis programs, it seems, and it's mostly active in the MIC cluster I told you about, and then also G6, which would seem to be MIC-like. So what I didn't tell you is, in addition to the complexity of T-ALL, B-ALL is even more complex with over 20 molecular subtypes from a similar study from St. Jude. You can read the PMO-ID there. So basically, when you look at the B cell patients, we wanted to take the Program 4 that we had discovered in the fish and see if it was actually present in the human patients, and it actually is. So we see that Program 4 highlight a subset of the samples, mostly some unique subtypes of this, so they seem to be really active for this program. And then we went beyond that and we wanted to look at, okay, because we know our clusters, our models, G6 and G8, are active for Program 4, and these clusters are active, the human patients are active for Program 4, do G6 and G8 point to anything? And the answer is yes. So we see that G8, probably not surprising because it's a MIC-driven model, points to a MIC BCL2 subtype of BALL, and then G6 markers that we identified in our study point to a different subtype. So right now what we're doing is actually just got this email, so this is unfortunate because I had already sent the slides here, but we're looking at the patients, the outcome data to see if we predicted anything about the human patients using the zebrafish. And so that's a very exciting possibility because the potential is that we've used the unbiased zebrafish screening approach to learn something that could diagnostically be significant for human patients. So I think that's very exciting, and it's a paradigm that I'm hoping to follow up more in my career. So anyway, I hope I've convinced you, or at least made a compelling argument that fish are cool, and that you can use them to do some really quick, rapid screening of some complex gene networks that might be causing these complex diseases. My PhD mentor, Steve Johnson, used to say he thinks I'm allergic to hypotheses because I don't like them. So I usually like to start with a phenotype and then work backwards, but that's just me. He tried his best to teach me how to do the other way, but I just, Anyway, so yeah, this is what we did and this is what we're working on. So there's a lot of interesting possibilities for how we can expand the leukemia field and learn how to use these models to understand the patients. But yeah, so these are the people that helped me out with it in my lab, and I'll take any questions.
Oh, hello. This is really powerful, but I have a technical question. Where did you get all these plasmids overexpressing oncogenes?
Yeah, that's a good question. I'm going to answer the way that I've been told not to answer this question because I'm here I was one of you back in the day, so I'm just going to be real with you. So the proper answer is they were selected based on recurrent genomic alterations in T cell relapse patients. So we have the clone DNA driven by the RAG2 promoter to drive lymphoid restricted expression of the gene. So this is basically just how we do gene expression in zebrafish. The actual answer is, during COVID, I dug through the freezer and I found a box called plasmids. And so I pulled it out and I mixed them together, and then this happened. And now, so.
Beautiful.
Susan, I'll be brief. So I saw KMT2A on there. Infantile leukemia, I think, is attributed to KMT2A variants, and I think your system is looking at fish that have already developed and then you induce cancer. But are you able to learn things about developmental origins of cancer, like infantile leukemia, from your system, do you think?
Yeah, that's something I'd really be interested in going after, because I think you're right. One of the limitations of this is that by the screening protocol, they've already gotten the cancer, so I'm not really observing, it's happening or whatever. So we've thought about ways to maybe do some more clonal approaches to sort of capture the sweet spot between pre-leukemic and transformation. So I think that's actually one of the things I'd be very interested in designing yet another absurd screen to capture that phenomena. But you're right, this is not going to help with that particular, but a very interesting and encouraging prospect for the future.
Hi, I was curious, you showed the kind of differential infiltration of the different structures in the fish. At least with humans, right, infiltration of leukemia into the brain is a big concern. Is that something you saw with your model here?
Yeah, actually. It wasn't a particular focus just because I chose to highlight the more peripheral, fish-specific stuff. But yes, we do see pretty heavy infiltration into the brain. I mean, when you look at these animals, the entire body is basically covered with the blast anyway. And to your question before, seeing, what maybe hotspots are hit first in the infiltration would be really powerful. To understand where the blasts are being homing to before I just see everything just covered in red, if you will. So yes, it's definitely something worth looking into. But we have seen it, yes.
All right, up next, I am thrilled to introduce Katie Varley, who did her thesis work in Rob Meacher's lab. She is now an investigator and professor at the Huntsman Cancer Institute in the University of Utah. So welcome, Katie.
No, you should definitely say. All right, well, while we're getting set up, I guess I'll do a mic check. And I want to thank you so much to Susan and the organizers for inviting me to celebrate with you today. So I was thinking about it, and I think the last time I was in a room with this many of you was actually this 2008 genetics holiday party. And these were such fun events. I mean, look at this all-star cast. But we had a holiday baking contest. And so amazing creations, right? There's this mitotic cake. I was so proud of my methylated cytosine cookies. But the Fay Lab definitely stole the show with their Bacardi Bunsen burner. And I think Debbie's in the audience. I'm going to give her credit for that one. So tremendous amount of fun. All right. So I did my PhD with Rob Meacher, computational biology PhD between 2004 and 2009. And this was such an exciting time to be in the department and just in this field. So the human genome had recently been sequenced, right? We had computational protein folding was starting to be able to actually predict protein structures. We could barely see next generation sequencing on the horizon. It was such an exciting time. I did my first rotation with Gary Stormo in 2003. In the summer, I came early because I was so excited. And it was an amazing experience to really get into sequence analysis and really start to see that. I learned about personalized medicine from Jeff Milbrandt and Rakesh, and then Brock taught me way more than I needed to know about gene regulation, and I still think about him every day. In my thesis committee, I had Paul, Elaine, and Mike on there to just advise me, and it was such a great experience. But really where I got hooked on science was in 4444 Forest Park, in Rob Meach's lab. We were down a spiral staircase, down the hall from the sequencing center, and I just thought Rob was a molecular wizard. This was so much fun. So we were doing crazy glass chemistry. I was doing Fourier transforms to analyze image analysis. We were modeling polymerase kinetics. I mean, it was amazing. And I was working on Polyny sequencing, and I was so enamored with the idea that we could invent second generation sequencing. So it was an amazing experience. And this was actually, so everything was going well until 2005, 2006 was a tough year. All right, and this is where I learned my first big lesson in science here. And so it was a tough year, like I said. So Rob didn't have tenure yet, so there's some pressure. My mom died of breast cancer. I developed rheumatoid arthritis, and there was also the commercial launch of second generation sequencing with 454. And that same year, Selects emerged with Lynx, which became Illumina sequencing. So the pressure was on, right? My Polyny sequencing was working well, but not really, right? And so, we should have been risk averse at that time. But for some reason we weren't. And that was a really remarkable experience because I think we were brainstorming so much about the potential applications of second generation sequencing that we just felt like we just needed to skip ahead, right? Even if it was already going to come out, we had to figure out how to do this. And I probably could have kept playing with Polynese and graduated with a really rigorous but completely obsolete technology, but instead we went for it. And so this was kind of the biggest learning experience for me was working with Rob, and he was like, we have to find the killer application for second generation sequencing. And this is the critical thinking that I learned from him. So we had to make informed guesses about what the technology that wasn't released yet was going to be capable of doing. We had to figure out how to avoid competition. It was an incredibly crowded field. We really wanted to have a high clinical impact. And then Rob coined the term, for me at least, how many miracles does it actually take to develop some of these technologies? And I still use this process daily, right? How do we figure out what to do? And this was also a point where I got probably the only, or at least the most important career advice. So when we thought about what we were going to do next, the question was, what should we be doing, and is it really worth the risk, right? We should be risk averse, or should we go for it? And Rob, who had an engineering background, said, this is way more interesting than measuring tolerances on ball bearings. So, and his other advice was, if this doesn't work out, I can go be a hedge fund trader. But I think what he was trying to articulate was probably what this Teddy Roosevelt quote was, which is, far and away the best prize in life has to offer is a chance to work hard at work worth doing. And so I feel like that was my big training at WashU, is actually trying to figure out what is the work worth doing, right? And so that leads me to my second lesson, which is free-range learning. And so when I started, early on, Barack said, if you're getting A's in your classes, then you're not focusing on your research enough. So ironically, he was really surprised when I got to be in his class. So, but it made the message was clear, right? Which was that we weren't going to be spoon-fed the information that we needed to know in graduate school, and we had to go out and learn it ourselves. We had to go find it with support, incredible amount of support. But it took some audacity, right? And so I wanted to highlight a couple things I remembered about this time. Where we had support for audacious activities and free-range learning. And so the first was, we decided we wanted a student-only seminar, right? We wanted to have our own. And the reason was we wanted to save space to basically embarrass ourselves in presentations, and then also to challenge our half-baked ideas, really kind of work it out amongst ourselves. And so we asked for support for this. We asked for food and everything. A totally reasonable answer would have been, you can pay for your dinner yourself. Like, what are you asking us for? And instead, we got pizza, a room, a projector, and we got the support of the department. It was amazing. Another example, you heard about the Young Scientists program, right? We would go out and do these outreach projects in the St. Louis public high schools, and a totally reasonable answer would be like, you're kind of far behind, like you should really focus on your research. And instead we were like, we were told from the PIs and the faculty in the department, here's a laptop, go make a little module to teach them about sequence analysis. And probably the most important one to me was actually when I told Rob that I thought the best application of our technology was for cancer epigenetics. And I asked him if he would pay for me to go to a conference for a field we didn't even work on, right? And a reasonable thing he could have said was, we don't work on that, choose a different application. And instead he said, yeah, go to the conference, and let me introduce you to Paul Goodfellow, go to the Siteman Cancer Center, connect with people and learn. And that had a huge change on the trajectory of my career, so thank you, Rob. And that's where I got connected with Paul Goodfellow and David Much and spent a lot of time in the Siteman to learn this field. All right. So, and that brings me to kind of the third lesson that I learned at WashU, which is you need to surround yourself with people who work hard and celebrate success. And so this free-range learning is really a team effort, right? And so Ting and John Krusty McCutcheon were grad students ahead of me, and it was before I had digital camera, so no embarrassing photos. Sorry, Justin Gerke. But they were, all three of them were great role models ahead of me of how to persevere. So they were really hardworking, and they were frequently grumpy, right? And then they would get up and give these energetic, optimistic presentations about their research. And it was just such a great example of how to work hard and how to push through, and then how to be optimistic about it, even when things aren't going well, and to celebrate success. And so it was, it's so important to have those people in that support group, and the department really fostered that. So it was a really great experience. And I had these two quotes on my desk throughout graduate school, which is opportunity is missed by most people because it's dressed in overalls and looks like hard work. And results, a lot of results. I know several thousand things that don't work. And those kind of attitudes were what were pervasive amongst our community here. And it really changed my career. All right, so, and the final thing is miracles can happen if they're precisely selected and meticulously attempted, but we got it. And so I got my little mini wizard's hat in 2009, and we did not invent second-generation sequencing, but we did use it for targeted sequencing of mutations in cancer, for single molecule DNA methylation sequencing, to look at intratumor heterogeneity, molecular barcoding. It was such an exciting time to apply this new technology to all of these new applications. And so that led me into my postdoc fellowship at the Hudson Alpha Institute, which is also an audacious enterprise at the time. It had just started. It was open for less than a year. And I got to work with Rick Myers on the ENCODE project and collaborate with UAB School of Medicine to do breast cancer research. And we sequenced a lot, and it gave us some of the first glimpses into DNA methylation and gene expression differences in cancer and associated with prognosis. This was also a really exciting time because I could start a company. So I helped co-found Kailas Genetics at the Hudson Alpha Institute, and our goal was really to put genetics into practice. And the core technology, so I make this joke, right? We pivoted more times than we changed our logo here. But the core of it was always to do the targeted sequencing that I developed in Rob's lab, and really to try to get it into clinical practice. And we focused on pharmacogenetics and hereditary cancer risk. And this is where I really stared into the abyss of translational biomarker research, translational medicine. But we did build some bridges. And so remarkably, the Veterans Affairs healthcare system actually adopted pharmacogenetics and set standards. And now veterans and their families receive pharmacogenetics in their medical record now. And we also did a program so that people, even without health insurance, could get cancer genetic screening in the Southeast and underrepresented populations. So it was a really fun opportunity to try to push genetic sequencing out into the community. All right, so then I started my lab at the Huntsman Cancer Institute. And when I thought about what I wanted to do, I really asked the question, after like decades of intensive research and drug development, why do women still die of breast cancer? And I'm showing you this particular graph because I think it really makes the point that there's been major advances that have changed the shape of these curves, particularly for white women. And black women have a higher rate of death of disease from breast cancer than white women. And so this tells us that we still have a problem to solve, a big one. Right? And this is a fantastic article talking about the racial disparity in breast cancer mortality in the US. But it's still a major problem, an important one to work on. And so when I started my lab, I really focused on 3 different problems, and I'm just going to give you little tiny snippets of each of these, a little more on the middle one if I can. And so the first problem is that mammography actually misses 20% of breast cancers that present at the time of screening. And the reason for that is actually because, well, there's a lot of reasons. One of the most common reasons is because that tumor is sitting in very dense breast tissue. And so they can't see it on mammography. And it turns out that black women are more likely to have dense breast tissue than white women. So there's a problem, right? So we need a different modality to try to detect cancer early. And so you all have probably heard of circulating tumor by now. Tumors shed DNA into the bloodstream. Other organs do too. And the DNA can be protected if it's wrapped around a nucleosome so it doesn't get degraded. So you end up with these tiny pieces of DNA, 176 base pairs long. And in patients with early stage disease, GRAIL has done enormous studies to show us that the fraction of that cell-free DNA that's tumor DNA is about 1 in 10,000 molecules, or 1 in 1,000 molecules if you're lucky. And so they've been developing assays for large-scale screening. I'm sure you've heard about them. But I wanted to point this out, which is that their ability to detect stage 1 breast cancer is extraordinarily low. It's 2–3%. So this problem is not solved. And even though it's really crowded, just like the work during my PhD, it's worth doing. And so I asked the question, what would Rotmetra do? and I thought we need to do some math. We need to do some really clever molecular biology, and we need to do some really deep targeted sequencing. And I wasn't going to take the time to go into that, but basically I want to show you that we've identified 300 loci that have cancer-specific DNA methylation. We really weighted this panel to be for breast cancer, and we included other loci just to make sure we could distinguish breast cancer from other cancer types to spot it. And then now we're getting to the point where we can actually detect those really rare circulating tumor DNA molecules in early stage breast cancer. And so back to people, right? So it's important to surround yourself with people who want to work hard and want to reach goals. And so I'm really lucky to work with the team to be working on all these clinical studies. And I also want to call out Atunji here at WashU, right, in order to try to collect blood plasma from patients to see if we can validate this across the spectrum of treatment in breast cancer. All right. So the second topic that I wanted to focus on when I started my lab was triple-negative breast cancer. And as you heard, triple-negative breast cancer is an aggressive disease. It's also more prevalent in black women than white women. So this is something we need to address. There's no one-size-fits-all treatment, and there's no established biomarker to try to figure out which of these treatments should be used. So there's a problem. All right. So actually, during my postdoc, I did RNA-seq on a bunch of triple-negative breast cancer primary tumors. This was kind of our first glimpse into this, which was very exciting to move off of microarrays and into sequencing. And what we found, just very briefly, is actually that the MHC class II antigen presentation pathway, which is the sister pathway of the one you just heard about from the Hansen lab and Dr. Gillian. So this pathway was highly expressed in a subset of patients that had a great prognosis. And so that was really exciting. No one thought this pathway was actually expressed in epithelial cells, so it took us many years to actually discover and validate that it was the breast cancer cells themselves that were expressing this. So unlike class I, which is on all our epithelial cells, we thought this was only in specialized immune cells, but here it was in breast cancer. And as more data accumulated, we actually saw that this replicated, that patients with high expression of this pathway did really well. And remarkably, they did well even if they weren't given systemic therapy, no chemo, right? They still did well. And there was old mouse studies actually from the time we heard about from the Hansen lab where they started studying these pathways for vaccine development really early on, which showed that the expression of this pathway in tumor cells was actually sufficient to induce an anti-tumor immune response. And so I turned to it and said, all right, can we develop a clinical scale assay to assess risk of recurrence in triple-negative breast cancer patients? And we had to switch from RNA-seq to a different platform that could handle these little shriveled up tumors that are the clinical specimens you get out of the real life clinic. And so I used the Prosigna assay. I used the NanoString platform because the Prosigna assay was actually a clinical test for a different subtype of breast cancer. So let's get all on one platform and see if we can get in the clinic. And so we did more math to calculate one single score. That would be the risk of recurrence score. It's immune activation score. And so patients with really high expression, we had 100% specificity in our institutional cohort. And in this cohort, we actually weighted it for patients who refused chemo. And we could see even amongst patients who refused chemo, they did really well, right? So there's something really special about the expression, this gene expression in these tumors. All right, so it seems like we've got this. You know, there was headlines. This is enabled precision patient care. I started hearing from patients across the country, I need this. But it turns out that's not all it takes. So this is a research essay, not a clinical test. And so I turned to Chuck Peru and Phil Bernard and Torsten Nielsen, who had developed the Prosigna assay, and said, what does it take to actually get this in the clinic? And it turns out for them, it took 16 years and thousands of specimens, right? And I said, okay, we've got a road ahead of us. And they weren't even dealing with a lot of the issues that we face now related to this beautiful but gut-wrenching article. It's probably the only scientific paper that's ever made me cry about the academic uphill battle to personalize treatment for patients with triple-negative breast cancer. So what do? Well, we work together. And so I worked with the I-SPY trial. It's a national trial system to try to identify biomarkers and choose treatments. I also have an institutional cohort trial that we're running. And then when you're in trouble, you talk to Canadians. They have a randomized international phase 3 trial, and they have nationalized health care, so they can't afford to undertreat or overtreat their patients. And so they're highly motivated to help and have great resources. And so I'm just going to give you a tiny snapshot of this study, which is not complete yet, but I thought it would make the point, which is that we've identified these patients with really high expression of this immune activation score. That's the red line at the top. And you can see that they do really well. You can't even see the red line on this second graph because it's buried behind the green line, but they're both up there. They have great prognosis, right, regardless of whether they receive paclitaxel, which is the standard of care chemotherapy. And I want to just call out France, who has been brave enough to start a clinical trial to try to de-escalate chemotherapy for these really low-risk triple-negative breast cancer patients. I have tried for years to do this in the United States and couldn't get it, so vive la France. You'll see that the green line here is where we can really see something going on. That's the patients who have high scores but also have positive lymph nodes, so their disease has progressed before they made it for treatment, right? And so for them, they actually benefit from paclitaxel. And from work I didn't show you with the I-SPY, we've shown that they would also probably just benefit from PD-1 immunotherapy if we could ever give it without paclitaxel. But probably more important from this study is that now we can identify those people in the teal and purple lines at the bottom. And those are the patients with low immune activation scores. And now that we can spot them, we can figure out what they need because they need something else entirely, right? And so that brings me to my last topic, which is really brief, which is metastatic breast cancer. It's lethal. We have a lot of current treatments, but they only add months to life, not cures. And so what is going on here, right? There's been millions of dollars and decades of research. And I'm starting to think that we might be studying the wrong cells. So I've sequenced primary tumors, lots of them. I've sequenced a lot of metastases, right? And I have long lists of thousands of DNA methylation, chromatin accessibility, and gene expression changes that happen, right? But I also want to point out that usually the time between these two things can be a decade or more for these patients. And these thousands of changes, right? It's almost too late. I hate to say it, but I'm scared that it's too late by the time we're looking at the metastases. And so what are we missing, right? What's happening in between these stages? And I think we finally have the technology to actually look at the disseminated tumor cells before they grow into these monstrosities of metastases with all of these changes. And so I'm really excited to work with a big team to look at disseminated tumor cells in bone marrow and also disseminated tumor cells in grossly uninvolved tissue from a rapid autopsy program that Alana Whelm started at my institution. And we're working together on this. In order to really study those seeds, right? When they come out of the primary tumor and they're hiding out in the bone marrow for a decade, right? And they're hiding out in the liver for a decade. What are they doing? That's when they're most vulnerable. And what are those initial changes that we can really focus on? So, and again, it takes people and a village to make this work. And so I'm really grateful for the DoD Breast Cancer Program for this large collaborative award that we have to work on this, and also the amazing team of people that I work with in my institution and across the world. And I want to acknowledge the patients who participate in cancer research studies. They don't benefit from this. They're just doing it out of goodwill, and thank you. And then I also want to acknowledge the research advocates, the patient advocates who keep us going when times get hard. So thank you.
We can have one question.
Okay. Any questions? Rob.
Great talk. That was very good. I'm curious about you have where you divide the two tumors into the high immune state and the low immune state.
Yeah.
I'm curious about what the T-cell invasion is in the two things. You know, is one, is the high immune state, as you would predict, more invaded by T-cells, and low state more invaded by, say, myeloid cells, or what would be the prediction?
Yeah, I mean, so definitely the high immune state is invaded by T cells, CD8s, as you heard about earlier, but also a lot of CD4 cells. And in the mouse studies, which I didn't show you, we actually show that MHC itself will first recruit CD4s, and then the CD8s come in. So yeah, they're all there. And part of the signature is actually both the MHC gene expression and T cell gene expression to see who's in the neighborhood. But yeah, in the other group, it's a combination of immune cold and then myeloid. So yeah, thanks. Yeah. So yeah, this was triple negative breast cancer, and then the study I just showed you is actually also HER2 positive breast cancer, where we're seeing the signature too. Thanks. Thank you.
Next speaker is Dr. Kai Tan, and he's a professor of pediatrics at the University of Pennsylvania and is director of the Center for Single-Cell Biology at the Children's Hospital of Philadelphia, and he was a graduate student at Dr. Gary Storm Lab. Welcome.
Thank you so much. Thank you. Thank you, Susan, and the committee for the invitation. So yeah, I feel incredibly honored for the opportunity to share our work and also celebrate the milestone of the department with this very amazing group of scientists, mentors, and friends. So today I'm going to talk about bits of gene regulation in hematopoietic stem cell. So yeah, so a little bit background about myself. I did my PhD in Gary's lab 1998 to 2004. I have working on computational methods to expand transcription regulatory network in E. coli. I had an amazing thesis committee, actually 6 members. So they, because Dr. Grossman left towards the last year of my thesis. So, but they all helped shape my scientific career. So I also want to thank Gary for introducing me to the amazing interface between information theory and transcription factor protein interaction. So when Gary first told me he can actually quantify the transcription factor binding specificity using Shannon information in information theory, I was like, wow, that's really cool. I didn't know KD and K off. So I said, wow, can I join your lab, Gary? Luckily, the answer was yes. So that's how I get started. Katie, listening to your talk, I think your cohort had a lot more fun than my cohort. But I do have some fun memories of my years at WashU and the department. So I can easily go on spend an hour reminiscing, but I will just add 2 more points to your points, Katie. The first one is really, I'm very lucky to be able to do my PhD really at the epicenter of genomics and computational biology education and research at the time, and still so. So I feel very lucky to be able to actually take this course, Bio 5495. It's Introduction to Computational Biology. The title is deceiving because it's really not introduction. It's a really badass course, and it's really, I still have PTSD. Maybe not too much, but really remembering burning those late night hours to try to implement hidden Markov model from Sean's assignment. You know, Sean's hidden Markov model, Warren, Colin Auerschuh, extreme value distribution, Gary, obviously, DNA motif weight matrix, David States phylogenetics, and Mike Zucker, RNA secondary structure. So it's a really great course. I feel very lucky to be able to be exposed to this course. And obviously, it's equally exciting to be able to witness some of the major milestones in genome science research being reached close to you. For instance, the sequencing of the yeast genome, worm genome, and finally, the human genome. So of course, these my training is not limited to these top-notch scientific training. The other very important training the department gave me is really to be able to critically evaluate a variety of beer through the Friday afternoon hour happy hour. I don't remember drinking tea, but I remember sample a lot of beer. So this is a picture of such a training session led by Dr. Waterston and other faculty members. So that's my fond memory. Okay. So just one slide about my thesis research. Really, I work on two related problems in regulatory genomics. So the first one is really given you have a transcription factor, you know the DNA binding motif, but there's an incomplete set of target gene. How do you go about try to fill in the missing target genes? So we did this based on observing the conservation of both the DNA binding sites and the target gene sequence, something called the phylogenetic footprinting. The second related problem is really, so assuming you don't know the DNA binding motif for a transcription factor, how do you connect to it? So again, we use a similar trick. This is before you have ChIP-seq, all that good stuff. So back then there's any 3 sequence genome, E. coli, Haemophilus, and yeast genome. So again, our approach is really try to identify all the DNA binding motifs in a given genome and all the transcription factor encoding gene in the genome, and then try to match them through the considering of sequence genome conservation, the property of DNA protein interaction. So that's how. So these two projects really get me started to think about how can we approach these transcription regulation from more genome-wide and system level. So that's what I went on to study after I established my own lab in 2008. Really try to model these transcription regulatory network using both experimental and computational approach. So we've been using hematopoietic stem cell or HSC as our model system. So we're interested in trying to figure out how does the transcription regulatory network is wired during the normal development of the HSC in the mouse embryo. And also we're interested in how this core network is deregulated in human leukemia. You heard James talk about leukemias. So just a little bit about HSC. So as we all know, HSC is the ultimate source of all cell types in the blood, right? So HSC actually in the mouse embryo, also in human, developed from a very specialized endothelium cell called hemogen endothelium. And then they go through a process of multiple precursor cells called pre-HSCs, and finally the functional HSC first emerged in fetal liver around embryonic day 12.5 in the mouse. So there's this very critical transition process called endothelium to hematopoietic transition, or EHT, where you hear me talk quite a bit today. So in about 5 years ago, we published this paper where we did the first systematic analysis to identify important cis regulatory element, enhancer promoters, and transcription factors that underline this HSC ontology in the mouse embryo. So we first generate this large data set of RNA-seq and histone-marked ChIP-seq data using purified cells from 5 developmental stages of HSC, endothelium cell, hemogenic endothelium, pre-HSCs, and fetal liver HSCs, and adult functional HSCs. And then we try to apply our tool to really try to model the transcription regulatory network. So over the years, my lab, after left Gary's lab, my lab also developed a suite of computational tools to really allow us to model the transcription regulatory network by combining transcriptome and epigenomic data. So this is just a brief overview of the different type of tools we developed. First, we can identify cell type specific enhancers using epigenomic signature, and we can link the transcription factor or link the enhancer to the target gene by the consideration of sequence conservation and the biophysical property of chromatin folding. And then we can identify the combinatorial binding of multiple transcription factor to an enhancer. And then finally, we try to identify master regulator by considering what is the regulatory potential of that transcription factor in the entire network. So we applied this toolkit to our data I just mentioned. Here are the top predicted transcription factors we predicted for the 4 transitions from endo to hemogenic endothelium and from fetal liver to bone marrow HSCs. So maybe not surprising, we can identify some of the well-known transcription factors. Some of you might know RUNX1 is a key master regulator, and GFI1 and SFI. PU1 basically. But also surprisingly, what we found, as you can see highlighted in pink, is some of the top predictions are actually widely expressed transcription factors, including SP3 and MAS. So this is kind of surprising. Generally, you think about transcription regulation, you think about the key master regulator, but these are very cell type restricted, but we found these factors are actually widely expressed, but they are top prediction. So we went around to test these 2 hypotheses using zebrafish and mouse models. So here's just a piece of data. So we can show actually in the zebrafish, knocking out either SP3 or MAS, that significantly reduced the abundance of HSCs by using flow analysis. And secondly, in mouse, we show that the SSP3 null embryo has significant fewer number of hemogenic endothelium. You know, that's the precursor to HSC compared to wild type embryo. So these provide some evidence that these factors could potentially be important. So as I mentioned, we also study leukemia. Especially we're interested in 2 questions. One, what is the prevalence of these mutant HSCs in different type of acute leukemia? And secondly, so what could we identify a core transcription regulatory network across this different type of leukemia? So the pie in the sky hope eventually you can target these core factors. So recently we did a really large-scale single-cell analysis of leukemia samples from human patients of several major types of acute leukemia, as you can see here, B lineage, T lineage, myeloid. So this is basically our atlas has about 1.4 million cells. What I want to show here is really a course level annotation of different malignant cells and healthy cells. So the first impression you get is, so this is RNA-seq, this ATAC data. So the tumors are really heterogeneous, and we know this is not over annotation because the healthy cells from different patients group together, so as we expect them to be. But the leukemic blasts really separate out, suggesting a large level of heterogeneity. So as we know, cancer is a disease of arrest differentiation. So in order to really determine the developmental arrest states of those leukemic blasts, we need a reference. So we went ahead, generated the single cell and RNA and ATAC data using thymus and the bone marrow sample from healthy donors. So this is our reference UMAP. As you can see, we get very detailed developmental trajectory for both for the 3 lineages, right? This is the myeloid lineage. The color means level of differentiation. Going down is B lineage, and going left is the T lineage. So then we project our patient data onto this reference is shown in gray. The red ones are the leukemia blasts. So what I want to show is really, if you remember, this is this is the B lineage. As expected, most of the bloods are B lineage, but you can see there's scattered some dot. If you remember, this is myeloid trajectory, right? So clinically, these patients are diagnosed as B-cell leukemia, but we do see myeloid, we confirm these are mutant cells by DNA sequencing. So we do see these cells somehow has myeloid-like behavior. So suggesting these cells quite plastic, as James mentioned, they observe B-cell leukemia in T-cell zebrafish experiment, right? So these all suggest these cells quite plastic. We want to figure out what controls this plasticity. So back to this HSC like cells, we do turn out we do observe different level of HSC-like cell, malignant cells across the different type of leukemia. And then, so we do see support of these cells at epigenetic level. For instance, all these HSC-like cell, these two CD34 and KIT are two stem cell factors. You can see their promoter and enhancer are open compared to differentiated cell types. So then we try to link our observation to a clinical outcome. So the way we did this is as following. So in leukemia, minimal residual disease means cells left after therapy. So it's the single most important prognostic factor in leukemia. So you wonder whether the level of MRD is correlated with the abundance of these HSPC-like cells. So this is what we've shown here. Going down is the level of MRD decrease, and each cell here measures the abundance of a projected leukemic blast at each developmental stage as shown here. I hope you can appreciate these patients with higher MRD level tend to have higher abundance of HSPC. And then we can quantify that it's much more clear. Definitely, if you have a more HSPC-like cell, you have higher MRD. And then finally, this is my last slide. Okay, 4 minutes, good. So we basically put our tool together and we generate this core, we call it core HSC-like transcription regulatory network. It has 6 transcription factors, 3 HOX gene, HOX A3, 5, and 9, 2 AP1 factor, FASB and JUNB, and 1 CEBP alpha, and 45 genes. And we show that the activity of this core tRNAs is very strongly positively correlated with the level of MRD as indicated by the color. And also, more importantly, we show the activity of these tRNA can predict the overall survival of the patient independent of MRD. So this really suggests these tRNAs is really underlying some process for these leukemia subtypes. We're in the process for using both in vitro and in vivo models to test these factors now. So the hope is, as I mentioned, we can eventually target as a common targeting mechanism for many acute leukemia. So with that, I want to thank my lab member, president, and the past. It's really a privilege to be able to work with this talent group of staff and trainees, and also our collaborators, and also patients and family and funding. Thank you. Of course, 3 minutes, not too bad. Yeah.
Nice talk. I have a question. Since you several of the transcription factors, they are targets of a histone modification, such as the ASO reader ENL can regulate these expression. I wonder, in your system, do you also want to expand to these chromatin readers, chromatin factors, because a lot of them have some potential inhibitors available, so that can boost the future fundings.
Are you talking about the normal HSC project?
No, I'm saying that for example, the ASO histone lysine reader ENL, that protein can bind to these several HOX genes.
I see. Yeah. I mean, well, now mostly focused on really the factor has DNA binding specificity. So yeah, as you know, a lot of these chromatin factors, their specificity is not themselves. They are, yeah, definitely those are very important. But now we're just that's another whole field. All right.
Great talk. So I'm wondering, like SP3 is uniformly expressed, right? So what's the mechanism and whether it's part of the core program?
Yeah, so it's not part of the leukemia program, but that's a great question. No, we can speculate, but no, it's also my speculation is the specificity could be provided by other lineage-specific factor. Actually, there are some evidence, for instance, SP factor can physically interact with RUNX and GATA. So that's one mechanism. Or the other hypothesis is that somehow they are somehow preparing the promoter chromatin environment, and then the RUNX1 factor like RUNX1 at the enhancers, then bring that to the promoter. But that's how you achieve specificity. But that's my speculation.
Thanks.
Yeah, Katie.
Yeah, great talk. Really exciting that you found this HSC signature, right, in these leukemias and its prognostic association. Can you tell which transcription factors you need to activate to drive them into differentiation state so that they'd be maybe less aggressive? Can you start to build that model? Can you push them down the differentiation pathway, basically?
Yeah, that's a good question. How do we identify actually factors driving differentiation? Yeah, right now our data, those are diagnostic sample. Don't know. Maybe we should collaborate with James with zebrafish model. That would be great. Yeah.
I want to thank all of our speakers this morning for absolutely wonderful talks that display amazing science and the atmosphere of the genetics department. Secondly, we're going to head to Couch Building for lunch, and you should make sure you go on the first floor because you can pick up your lunch, and then you can head up to the 4th, 5th, and 6th floors to both eat and see posters from the current students and postdocs. And we'll see you back here at 2:10 promptly. Oh.
What? Oh.
And you actually finished on time.
Yeah, I was. Now we can go to lunch.
So everyone, before you leave, there's a QR code that's up on the screen, and it will help you to find the posters that are available to look at with their numbers and their map position. So please take just a moment before you depart to scan the QR code that'll be up momentarily.
Sorry. Are you going now?
I can put my computer up here.
So then I will
follow you. I'll do that from there, but I want my notes on my computer.
You can do, yeah, that'd be fine.
Instead of paper.
Sure. Sure.
Because I'm not doing a standard talk. I've given thousands of talks or whatever.
I know.
Using result slides.
Yeah, this is really different.
reminiscent.
Yeah.
Yeah, sorry. I don't know if you could tell from my slides, but it's not, not data. It's just,
No, I think that'll be wonderful. Yeah, we'll scoot over so that everything is here.
Okay. All right. Thank you.
And you have one of your introducers. Make sure you get over. They're going to try to pick you up.
Okay, everybody, let's settle down. Okay. All right. Thank you, everyone. It is my immense pleasure to introduce Dr. Bob Waterston, who is a professor at the University of Washington. He was a postdoc with Sidney Brenner and joined WashU in 1970-something, don't remember. And he's our next speaker. Thank you.
1976. And I spent my first three months in Cambridge, England. Well, really, thanks very much to the organizers for letting me share this occasion with you. It is a real honor to be back. So let's get started. Microphone. I turned it. It's on. It's on. You can't hear me? Is that better? Okay. All right. I got to do this. Yeah. All right. So I'm going to start with President Clinton. Oh, no. It didn't. Yeah, oh. Nearly 2 centuries ago. Oh, you can't hear this.
Thomas Jefferson and a trusted aide spread out a magnificent
Can you hear that?
map. Yes. A map Jefferson had long prayed he would get to see in his lifetime. The aide was Meriwether Lewis, and the map was the product of his courageous expedition across the American frontier all the way to the Pacific. It was a map that defined the contours and forever expanded the frontiers of our continent and our imagination. Today, the world is joining us here in the East Room to behold a map of even greater significance. We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind. It was not even 50 years ago that a young Englishman named Crick and a brash, even younger American named Watson first discovered the elegant structure of our genetic code. Dr. Watson, the way you announced your discovery in the journal Nature was one of the great understatements of all time. This structure has novel features which are of considerable biological interest. Thank you, sir. How far we have come since that day. In the intervening years, we have pooled the combined wisdom of biology, chemistry, physics, engineering, mathematics, and computer science. Tapped the great strengths and insights of the public and private sectors. More than 1,000 researchers across 6 nations have revealed nearly all 3 billion letters of our miraculous genetic code. I congratulate all of you on this stunning and humbling achievement.
Okay. It's not often that science gets announced in the White House, and especially with such lavish praise. And it's even more remarkable today, I think. Hard to understand even. The Department of Genetics is obviously a key player in that accomplishment. And what I want to do today is give you some background on how all that came to be. And on this slide, shows a picture of Maynard, and he was a candidate for a faculty position in 1979, and I was on the search committee, and I was having lunch with Maynard in what is now Shell Cafe, I think. Anyway, Maynard had given this job talk on his work with Ben Hall at the University of Washington, other Washington, in which he had used DNA polymorphisms as a genetic trait, really one of the first uses of that. And he mapped the yeast tRNA genes. But that wasn't what he wanted to talk about today, or that day. Instead, he wanted to talk about his plans to map all the DNA of the yeast genome. And his idea was to take thousands of random clones from a yeast genome library, digest the DNA with restriction enzymes, and size the fragments on an agarose gel. here. Okay, there, yeah. I'll get used to it. So the idea is shown here. You have a region of the genome up above. Clones that come from the same region will share fragments, and you can deduce that they overlap. And Maynard's thought was that by looking at thousands of clones and using computer algorithms to detect the overlaps, he could reconstruct the whole yeast genome. And that would produce an encyclopedia of the genome from which he could retrieve any desired fragment. As Maynard described what he wanted to do that day, I thought at first he was crazy. I had once heard the adage, in biology, do every experiment an odd number of times, preferably less than 3. Maynard wanted to do thousands of essentially the same experiment, one after another, and the scale was really unheard of. But his plan was so carefully laid out that soon I was buying in, and I realized that it probably could work. And the result would be a genome in a bottle. That would be remarkable. And that, to me, was the birth of genome science. So the department at that time was really a new creation. Carrie was saying over lunch that it's not often you get new departments. But in 1963, James S. McDonald, Mr. Mac as he was known, proclaimed in his commencement address that the future is DNA. In 1967, Bill Danforth, then Vice Chancellor, persuaded Mr. Mac to act on his insight and establish the department through a large gift. It was to be housed on the 7th and 8th floors of McDonald of a building that Mr. Mac was also going to fund. Lore has it that the building was initially going to be only 6 stories until Bill Danforth pointed out to Mr. Mac that at 6 stories, the building wouldn't be visible from Highway 40. So Mr. Mac increased his gift, and 3 more floors were added, including the space for genetics. The School of Medicine actually then launched searches for a new department chair with really little success until 1975, the date we're celebrating here. And Don Schreffler at that point, a renowned human mouse immunogeneticist from the University of Michigan, agreed to come on one condition, that he not be the chair. It's true. But with no chair on the horizon, after a couple years, Don eventually agreed to become the chair and to take on the task of building the department. Maynard was the first recruit. And when he arrived, the space on the 8th floor still wasn't even finished. It had been left as shell space. So he camped out in Wayne Barnes's lab in biochemistry for a couple months. Paul Levine soon followed. Let's see. I meant to do this. Okay. And Don recruited these other people, which Eric will say more about. I moved to the department I moved from anatomy and neurobiology, where I had started in 1976, into the lab next door to Maynard's in 1980. And from the start, you've heard things today about all the wonderful camaraderie and so forth in the department. It was from the start, it was a remarkably stimulating and fun place to do science. We started a weekly journal club with just a couple labs to begin with. In time, that grew to take in labs from across the med school. We overflowed the conference room on the 8th floor. We started a social hour, which you've heard about. It originally was in my lab, with just Maynard's lab coming over. Can you imagine that, having a happy hour in a lab? Anyway, we moved to the kitchen alongside the autoclave and the washing machines, and the discussions were so lively, covering everything from science to just plain gossip. And for a few years, there was a Christmas party and a skit where the trainees adapted a movie the current movie of some sort, to spoof the faculty, and followed by chair races. I won't say any more about those. We won, though. And whenever the ventilation failed, Oda Mouse wafted up from the 7th floor mouse facility that Don had down there. Anyway, as you know, Maynard did go on to complete the physical map of the yeast genome. And with that experience, he was called to testify in front of Bruce Albert's National Academy's committee that was considering the advisability of doing a human genome project. Maynard later became a full member of the committee and was a major contributor to that final report, which was remarkably prescient. And as you hear from Eric, Maynard was also a key contributor in the early efforts to map the human genome. And he also provided key testimony in 1998 at a hearing. This is at the start of the fabled race with Solera. And I think his testimony was very key in ensuring that the public effort continued to be funded. Okay, so now I want to shift gears and recount the events that led to the founding of the department's Genome Sequencing Center. In the spring of 1989, John Selson, pictured here, along with Alan Colson, Bob Horvitz, and myself, were in Jim Watson's office at Cold Spring Harbor. Bob and John later shared the Nobel Prize for the C. elegans lineage and cell death, along with Sidney Brenner. Watson had recently become the director of the National Center for Human Genome Research at the NIH, and really the de facto head of the Human Genome Project. We were there trying to convince him that the C. elegans genome should be on the list of projects that would be eligible for support. Watson was skeptical, but gradually became receptive. And at that point, John blurted out, coming from a very different funding model, if you just give us $100 million, over the next 10 years, we'll do it. Watson barely paused and said, John, that's not how we do things in this country. So, how did we get to Watson's office, and what happened next? Well, it goes back to 1983. John in Cambridge, England, was quite independently of Maynard, trying to construct a physical map of the worm genome. It's much larger, and the overall strategy was similar, but different in detail to accommodate the worm's larger genome. The project really took off when John was joined by Alan Colson, who had worked with Fred Sanger on the sequencing on inventing DNA sequencing. Over the next couple of years, they analyzed thousands of clones, and from the overlaps, they could begin to piece together the worm genome. But by the time I arrived there on sabbatical in 1985, and a sabbatical is a good thing for everybody to think about, a problem had become clear. And that is that the clones of worm DNA in E. coli were not really random. They were missing pieces, and as a result, there were persistent gaps in the map. And I was working in John's lab because there was no other space for me at the LMB in Cambridge. And I became increasingly involved in trying to devise a solution. The solution came on a trip back to St. Louis. David Burke and George Carl, graduate students in Maynard's lab, were developing what eventually were called yeast artificial chromosomes, or YACs, as a means of cloning other organisms' DNA into yeast. And because they were fashioned like yeast chromosomes, they could actually, theoretically anyway, contain much larger fragments of DNA. My thought was that yeast, as a eukaryote, would have very different biases from that of E. coli, and perhaps they would have the missing bits of the worm genome. John agreed, and that was really the start of our very close collaboration that went on for the decades. When I returned from sabbatical, and David had completed the steps in making yaks, I started making worm libraries. Initially, the insert sizes were small, but test experiments done with John showed that they could indeed contain missing DNA. So I made more libraries with bigger inserts and so forth. And together over the next few years, our lab added lots of them to the map. And by 1999, when we met with Watson, the map had come together remarkably well. So that's shown here. This is the back hall of Bush Hall in Cold Spring Harbor. Alan taped together sheets like that, and the yaks, so you can see the yak bridges, but anyway, he taped them together and we strung them across. That's chromosomes 1 through 6 of the worm, and you can see chromosome 1 isn't so good, but chromosome 2 goes across the whole wall. We had chromosome continuity. We made sure Watson saw it because it was exactly what one needed to begin whole genome sequencing. And why were John and I, with two people with no real expertise in DNA sequencing, even thinking about sequencing the worm genome? Well, in February, Bob Horvitz, a former student of Watson's, and then at now at MIT, he attended a party with Watson. And Jim let it be known that while coli, yeast, and Drosophila were clearly on the list of organisms that would be test beds for sequencing to see if it would work on humans, worms were just too small of a field, wouldn't warrant the investment. Bob realized immediately the worm would be at a terrific disadvantage if it didn't have a genome sequence and fly did. So the next day, he was on the phone with both me and John, telling us because we had done the map, it was our responsibility to sequence it. So after talking with others, it seemed like no one else really wanted to take it on, so it was indeed up to us. We arranged to have that meeting with Watson, and he did indeed see the map on the back of Bush. Someone heard him say, "You can't see it without wanting to sequence it, can you?" So we knew we had him. So by the end of our meeting with Jim, he did give John and me approval to submit a joint grant. He didn't give us $100 million, but he gave us approval. While on the one hand, we were delighted that we had won him over, we realized we were taking on a commitment that would change our lives forever. Indeed, after the worm meeting at the train station in Macias, John turned to me and said, "The jail doors have just clanged shut." This is a scene he recounts in his book, The Common Thread. That fall, we did write the grant. Ladina Hilliard joined us in September, and Rick Wilson agreed to come in December. We had incredibly ambitious goals, promising a total of 3 megabases of fully complete and accurate sequence in the first 3 years. Sounds like nothing today, but at that time, it was far bigger than the biggest project ever sequenced, which was a couple hundred KB. Even though it was ambitious, we felt like if we couldn't do that much in 3 years, we'd spend the rest of our lives sequencing the rest of the genome, and that we didn't want to do. Anyway, our collaboration had success, as you heard. We completed most of the worm genome by 1998, and we contributed to the finishing of the yeast genome and its release with the help of Mark Johnson and Maynard's map. We did other genome projects, and of course, in figuring out how to do the worm sequence, we figured out how to sequence the human sequence. And our efforts, and that includes various people in this room, led eventually to the so-called race with Craig Venter and that White House announcement that I started the talk with. So I don't have time to talk about the many stories, and this is really not the right place to go into them. It is told well in John's book, and I recommend it to you. But it did lead to the emergence of the Genome Center as one of the premier genome centers in the world. That we ended up contributing about 25% of the human sequence, second only to the Sanger Center. And critically, in addition to the sequence, the Worm Genome Center constructed the clonal physical map of the human genome that not only fed our center, but other centers as well. It also formed the basis of it guided the assembly of the draft sequence that Clinton announced, and even Celera used it in their assembly. And critically, it formed the basis of the groups actually finishing the human genome, filling in those gaps and making it high quality. Apparently, it took like 63 liters of agarose over a year and a half. Anyway, before I close the talk, I want to talk about how the public project became committed to free and open data release, especially for those of you in the room who take it for granted, that it's just out there for everyone to use. When the pilot phase of the human sequencing started in 1996, human gene hunters were notorious for hoarding data and only releasing things only as much as they were forced to release on publication. Many of the mapping centers were formed by gene hunters, seemingly hoping to use the resources from their genome studies to find their favorite genes. John and I came from a very different ethos. In the 80s, the worm community was small, with almost everyone connected back to Sydney. People across the world shared results in the Worm Breeders Gazette. This is Casino rhabditis. It was a semi-annual collection of one-page updates from the latest in labs across the world. Couldn't be cited and was not to be used unfairly. So when John began the map, he announced his efforts in the Gazette, and he shared his progress in the Gazette later through BitNet and dial-up modems and everything else. But it was shared with the idea that the map would be most useful if people could have access to the latest data, and then in turn, they would send their results back to John so he could tie the genetic map together with the physical map and make a genome map. And with that as precedent, we adopted similar practices when we sequenced the worm, putting things on the internet when there was an internet, and we did it daily. So in 1996, there was a meeting of the groups that were coming together to sequence the human genome. And John and I came to the meeting hoping to get the group to adopt policies similar to the worm. The topic came up in the final session, with John jotting down points on a white easel as they came up, and I was leading the discussion. And after some very vigorous discussion, to our delight, the group agreed to these principles, which later became known as the Bermuda Principles, because that was where we were meeting. In addition to these three principles here, we agreed also not to patent any genes. These principles helped with coordination. They also made clear to the wider community that we were not exploiting the enormous resources granted to us for our private gain. All the data was going to be available to everyone equally. And by eschewing patents, we eliminated financial considerations. But probably even more importantly, we realized that the sequence is part of our common heritage, and it would bring the most benefit if it was available to everyone all the time, for any use whatsoever, to speed discovery. The Bermuda Principles were the cornerstone of the public project. They were at the core of this dispute with Celera, and we were under considerable pressure to collaborate with Celera because it would potentially get the job done faster. In December 1999, Harold Varmus, director of the NIH, Francis Collins, director of the NHGRI at that point, and Martin Bobrow from the board of directors of the Wellcome Trust, and I met with Craig and three Celera representatives to explore what had seemed like very promising preliminary discussions. But these face-to-face negotiations went nowhere. Tony White, the CEO of PerkinElmer, insisted that any combined product would only be available through the Celera website for years to come. and they would be the only source for any analysis done on it, including any gene annotations and so forth, including anything that we had found. They clearly wanted a complete monopoly on the genome sequence and any and all discoveries that came from it. In the end, we did not compromise, and the working draft sequence and the finished sequence were out there for anyone to use. We even got Clinton and Blair to, oh, that's a reference to an article on the Bermuda Principles if you want more detail. This is the Clinton-Blair statement, and I just draw you to that they commended other scientists around the world to adopt this policy of open data release. And I gather the genome model has been widely adopted across other sciences. And just to conclude, I would say we would be in a very different world if it hadn't been for Maynard's testimony, our commitment to open data, and a few other tipping points. So appreciate it. Anyway, in the last couple decades, genomics has revolutionized biology, and WashU genetics was central to that revolution. So thanks very much for the opportunity to share these recollections, and have a great next 50 years. Thanks.
We're going to hold all the questions and let Bob and Eric answer questions together at the very end.
I'll sit here.
All right, please welcome our next speaker, Dr. Eric Green, who until very, very recently was the director of the National Human Genome Research Institute at the NIH. He actually did his MD, PhD at WashU, and also did his postdoctoral work with Dr. Maynard Alston, which we heard about before he went to the NIH.
Well, thank you very much. It's absolutely a pleasure to be here. The timing couldn't be any better. I have a lot of spare time on my hands. I'm between jobs, but I'll get there at the end. You will quickly notice a slight difference between Bob's talk and my talk. There's two things I absolutely cannot give a talk without using, and that's a lot of PowerPoint slides and my hands. And so you will see a contrast of style between me and Bob. What I decided to try to do is to really pick up from where Bob left off, but particularly focus on the time in the department around 1990. It was a very magical time. It was also one that had a lot of personal consequences for me that are really quite special, but also has a lot of history, I think, for the department. In looking around the audience, I noticed there's a lot of more senior folks, but a lot of very young folks. And so for the senior folks, this is going to be a walk down memory lane. And for the more junior folks, it's going to be a history lesson. So there's a little bit for all audiences. It's going to be sort of like a Pixar movie, is what I can tell you. So some of you may not realize the deep roots I have to Washington University. I was born here, literally born across the street or just down the street at Jewish Hospital. I was raised in West St. Louis County. I went away to college at University of Wisconsin. But then I came back for the MD PhD program in 1981. Speaking of having shock syndrome, this was where I had first year of medical school. So all day long, I've been a little unsettled. Lucinda in the back row, you're in my chair. I got some of the best sleep I've ever gotten in my life during anatomy of first year, sleeping in that chair. And I can't believe people all day have left this up. I was told none of this was going to be on the test, and so I was learned you could just ignore it if it's not going to be on the test. So for graduate school, I had two wonderful mentors, Jacques Benziger on the left, Irv Boehm on the right. But I didn't work on DNA. I could barely spell DNA. I studied sugar molecules on glycoprotein hormones. It was a fabulous experience. And I had a wonderful 6 years. I graduated, as you can see, in 1987. Again, this morning I got a little unsettled because my favorite place to go when I was an MD PhD student with my medical school classmates, my graduate school classmates, was Tom's Bar and Grill right there in the corner. It's now the Good Farm Cannabis Dispensary. What the hell? So, but okay, I have bigger problems now than that. I'll deal with it. So I graduated in 1987. 1987 was a significant year. For those who don't know, again, for the history lessons, the word genomics may have been conceptually coined prior to that, but the word did not exist in the medical or scientific literature until it was put into this first editorial in a new journal called Genomics, where the field was described. And that happened to happen exactly. So I never heard the word genomics once in this auditorium, never heard it in the 6 years I was an MD PhD student. I had to make a decision just prior to graduating what I was going to do in terms of residency. And Jeff Milbrandt was actually instrumental, because when I was thinking about what to do, he was one of the people I went and spoke to, and John Lau and a couple others who were doing this thing called laboratory medicine, which was a light clinical responsibility that got you back to the lab quickly. That sounded like the secret ticket to me. Little lightness on the clinical, get back to the lab as soon as possible. And so I signed up. I was a house staff starting in 1987. And within that year, I had to go make the decision. I had to go find a postdoc position. Now, so that brought me to 1980. But I also had when I decided to do a residency here, I had a two-body problem. My wife was a medical student, so I had to stay here for residency. And it meant I had to do a postdoc at the same institution that I was a graduate student, which at least back then was really a stigma. They always said, change places, change this. Well, I couldn't change. I had to stay here. So I said, I need to change fields. And while I loved what I did as a graduate student, I knew I needed to do something different. Genomics had just gotten coined. Jeff Milbrandt was teaching me about cool things in molecular diagnostics, things. We did Southern blots, really cool stuff. I mean, it was very, very early days of molecular diagnostics. But it seemed that this genomics stuff was pretty good. Maybe I should look into it. And then I said, well, who can I go speak with about this? And one of several people I spoke to, but clearly the person I had to work with was this guy, Maynard Olson, who you heard about. Each person I'm going to introduce, I'm going to give you my own persona of what they're about. He's the intellectual giant. You already heard that from Bob. Now, what did I know about Maynard? Well, I will tell you that MD PhD students who tend to be a little mean and arrogant, and I was one of them, when they heard about Maynard when he got recruited, they all said, he's that weird guy building those maps on the 8th floor or wherever he was. So this was not somebody that a lot of MD PhD students gravitated towards back then. On the other hand, I will tell you that Maynard, as I watched all of my colleagues getting their PhDs and putting their thesis committees together, he was one of the most popular people to put on your thesis committee. Again, there's probably MD PhD students in the audience. Back then, everybody said the same thing about when you go to populate your thesis committee, you need 3 different types of people, at least one of each. You need somebody who really knows the stuff about what you're working on. So get one of those. And then you have to get somebody who's really, really nice, that no matter how bad you do will be supportive. And then you need to have somebody really, really smart who knows something more than anybody else. And Maynard was like on every thesis committee because he was the smartest person around that everybody wanted. So I knew this guy was smart. And so I went and talked to him. And I talked to him for 2.5 hours. And I said, I'm all in. And the next morning when I called him, I said, Maynard, I want to do my postdoc with you. His first response was, oh, really? I didn't know how serious you really were. After 2.5 hours of talking to him the day before. But it was magical. And I walked into a magical place at a magical time. Now, the other thing about me, besides my hands in the PowerPoint, is I save everything. I am a complete memorabilia person. And I was able to dig out of a box this. Any of you more senior people remember this from 1991? Washington University Magazine chronicling this incredible constellation of people that have come together and were doing genomics. And so what was described in this article, and some of the pictures I'll show from this article, is really what I will call the secret sauce. Because some of you may ask me, what was the secret sauce of genetics in circa 1990? And they're really just, these are just my thoughts. I did that, yeah, there we go. There was a dream team of people. I'll introduce you to them. There were some key scientific elements available to us. And I think as Bob pointed out, there was some unfettered audacity. Just go for it. Yeah, there's always a little bit of luck. You always have to admit there's luck. So who were the dream team of people? Let me introduce you to them and their personas. Well, this guy you just heard from, and if you heard him talking about data release, you know why the persona is the moral compass. He's the guiding light, brilliant, helpful, wonderful, a great leader. And Bob and I became instant friends. Even though he wasn't technically my mentor, he was always my mentor and always my friend. You also heard about this guy, the elusive genius, I'll call him, Phil Green. He was brilliant, he was unique. We also shared the name Green, and people would frequently get us confused. I have no idea why you would get confused between Phil and I. First of all, he's crazy smart, unlike me. He also has a very rabbi-like beard, unlike me. And also, unlike me, I am meticulously organized. This would be what my office would look like. This is what Phil Green's office would look like. In fact, the reason Phil would change jobs every 4 or 5 years is he just didn't want to clean his office. He'd rather start from new. And if I had $1 for every person that got lost up on the 8th floor of McDonald Sciences who would wander in my office and say, is this Phil Green's office? And I would say, does this look like Phil Green's office? And then I would send them down to Phil Green, and then they'd come back and say, I know exactly what you're talking about now. So, and then there was this guy, right? I couldn't figure out what his persona. He's just BFF. I mean, he's just best friends with everybody forever, and he's got a lot of spirit, and he's got a great laugh, and he's a lot of fun, and he's a kindred spirit of mine, and I always enjoy time I spend with Mark, with the exception of when he wants lunch, in which case he always wants to go to White Castle. Now, here's the problem with this photo: this is a modern day White Castle. Notice a recycling bin and a newspaper thing. Are you kidding? What Mark would always want to take you to is the seediest looking White Castles, where there would be guns on one side and needles on the other. And in the middle of lunch, if you hear gunfire during lunch, he'd buy you dessert because he thought that was special. But true to Mark, I heard he had lunch at White Castle the other day. So some things never change. In order for at least the Human Genome Center to form, it required an organizational mind and somebody who would just make all the things happen. And we needed one of those, and that was David Schlesinger, who I guess had a joint appointment in the department. I think he was in the microbiology department, if I remember right. And he was the diplomat, and he could get the deans and the chancellors and other people together to get done what needed to be done to build the Human Genome Center, in contrast to the Sequencing Center that was created as part of the Human Genome Project. And David was a remarkably helpful and advocate of those of us who were involved in the genome projects, especially the trainees. And we had a chair, Dan Hartle. And Dan, you can see these photos, was sort of very much of a helpful, stay out of the way kind of a chair. He would be helpful when you needed him, but he really didn't get in the way, and he was very supportive of all these activities going on. And we also needed some human geneticists, and that included Helen Donnis Keller on the left and the late Danielle Gerhard on the right. And they taught us a lot about human genetics because none of the rest of us really pretty much were human geneticists, but we started to build human maps. It was very helpful to have them around. Another person that arrived, as you heard, on the scene about the time I arrived, or maybe a little bit after, who was also a good friend and colleague to the present time, is Rick Wilson, who I will describe as the maverick, because I think a little like me, he doesn't always follow the rules. And the one example I would give, which I won't name the person in the audience who came up and told me about this just before, my talk, who remembered this story. But what some of you might not realize is that in the first year of the Human Genome Project, in both the Sequencing Center and the Mapping Center, the Human Genome Mapping Center, the rate limiting factor to get the work done was this stuff called Taq polymerase. Because it was a monopoly. It was so expensive, it was consuming our budgets. But it was happening everywhere around the world. And so there were these expression vectors with the Taq polymerase gene in it that you could express, and then you could purify ridiculous amounts of Taq polymerase with a simple one-column purification step in a cold room. It was completely illegal, but we had to map and sequence the genome. They had to map sequence the worm. So Rick and I, the maverick, we just did it. And I remember we spent one weekend in a cold room together up on the 8th floor, and we truly did purify I think $2 or $3 million worth of Taq polymerase. And we made a tactical error. We canceled our purchase orders with the company because we didn't want to waste the money. And the next thing you know, each of us get a letter like I've never gotten before from the General Counsel Office at Washington University saying, if you keep using this Taq polymerase, we won't defend you when you get sued. And that scared the shit out of us, actually. And so we stopped. And it actually led, to be honest with you, it led then to a big a big, horrible to-do involving the NIH. It was between directorship of Jim Watson and Francis Collins, and it was Michael Gottesman was the acting director of the Genome Institute, and he was able to get a cut a deal with the manufacturer of Taq polymerase at that time, such that if you had a Genome Center grant, you got half-price Taq polymerase, and that solved the problem. So, but it's the scariest letter I've ever gotten to date, because as a postdoc, you just don't want to be sued. Okay. I should also point out that the kids in the candy store, at least the scientific candy store, were all the other members of the Olson lab. This is a true diaspora. don't have time to go through all these people. They're all around the world now. They're all leaders. They're all wonderful. They were great colleagues. And I was among the first. I wasn't the very first postdoc in that era that was doing genomics, but I was among the first, and many followed, and many even followed when Maynard went out to Seattle. And it was a wonderfully collegial and incredible group. I do want to call out one, and some of you should be a little embarrassed when I start talking about this for the following reason. The one is this guy, Pui Kwok, who he was a dermatology fellow, another physician scientist. He was the next postdoc in after me, and he and I were asked to start a new lab down the hall. So it's just the two of us in this lab. We sat back to back in the lab. He looks about 12 there, but he still looks about 12. But Pui was the dermatologist in chief for the entire department. And what ended up happening is that it became a bit of a clinic. Because what you may not realize is that when you have a dermatologist around that you can just ask a question about, everybody has something to show him to get advice. So what happened was we would work in a laboratory that looks like this, but people would come in and they think it was a dermatology clinic that looks like this. So how did Pui have it organized? He had his desk, and he had on one side was a drawer that had all of his pipette men and lab stuff, and on the drawer on the right were all these ointments and pills and medicines that he stole from the dermatology clinic. I'm looking around the room. I've seen some of your rashes because they didn't care that I was pipetting and working. They would come in, they'd rip up their shirt, they'd say, "What is that?" And he would say, "Oh, try this ointment. Is it itchy? Try that ointment." It was a little unsettling. Now, I will tell you that after a while, it would be really bad because I would really want to get work done. People would come in, "Where's Pui?" "Well, he's not here. Can I help you?" They go, "Well, you're not a dermatologist." I said, "No, but I've watched Pui a lot." And then they'd show me something, and I'd go, "Does it itch? Does it burn? What happens when you scratch it? Here, try this and come back in a week if that's not helping." So, but people are not shy when they get free dermatology service, I can tell you. And I feel like gouging my eyes sometimes what I ended up seeing. All right, so those are the people. Let me tell you about the science. And Bob introduced you to this because there was a drumbeat of discussion, starting with some early articles that led up to the naming of the field genomics. And then there was this famous report that you heard about and other reports, and then eventually the hearing in 1989 that led to the funding of the Genome Project that started in 1990. However, what I will tell you, these were all nascent ideas, but they were enough to get Watson to convince the Congress to give the money to get it started. And so I really think that in history, we will go down of thinking as 1989 as a truly historic time. Because really everything changed. It just completely changed in 1989 as far as I'm concerned. You know, first of all, Taylor Swift was born. Okay, so let's appreciate that. But you know, at the same time, 89 was the ramp up, launch up, getting ready to launch the Human Genome Project in 1990. Okay. So off we went for the Human Genome Project. Now here again, I think all of you here either lived it or learned about it, but it was a wild ride. It was incredibly audacious, it was incredibly stressful, and it was incredibly successful. And so by every metric, it was really quite wonderful. However, I don't think young people in this audience adequately appreciate what an incredibly primitive time it was to do something this audacious. So let me just take you back before you were born, in some cases, to 1990 and tell you things that did not exist when we said we are going to completely read out humanity's blueprint. You'd think we'd be ready for it technologically. Yet we didn't have web pages. They came in 91. We didn't have texting, 92, or live streaming, or even web browsers. And in fact, when the Genome Project started, it was estimated only 3 million people around the world had access to the internet. Sony PlayStation came out 4 years later. GPS didn't exist till 5 years in. Amazon didn't even exist 5 years in. Otherwise, we would have ordered our attack polymerase from Amazon. Even Windows didn't come around till 95. USB cables in 95, DVDs, Wi-Fi. I mean, no internet. We didn't even have Wi-Fi until 97. Google didn't come out until 98, social media in 2000. And then none of the following things came available until after the Human Genome Project ended. So imagine, no iPhones, no YouTube, no Facebook, no et cetera, et cetera. It was an incredibly primitive, in fact, some might even argue a prehistoric time. We did have film cameras, and sometimes we were caught on film cameras. So I remember one day being caught with somebody who was able to photograph me as I was coming to work in McDonald Sciences building and getting into work riding my dinosaur. So if we didn't have all of those things, what did we have? What was the secret sauce upstairs in the genetics department? I mean, what I will tell you is that we really were the key epicenter, and everybody was watching us. And I know that because they all visited. And everybody around the world practically would come and visit to figure out what we were doing, what science we had, what tools, what technologies, how we were being applied, and to talk to the brilliant people that Bob introduced you to and that I've introduced you to. So what was the science that especially allowed us to eventually map and eventually sequence the human genome, but particularly the mapping centers? Well, Bob introduced you to these two guys. They were graduate school classmates of mine. We all were in the same graduate class. I was MD, PhD, they were PhD. Dave Burke, George Karl. George Karl in particular invented a number of technologies for separating large pieces of DNA, pulse field gel electrophoresis methods, and Dave Burke partnered up with his good friend and colleague George to invent YAC cloning. So these were the inventors, these were the tool builders. And I was so frickin' lucky because they went off to their postdocs, and 3 weeks later I joined the Olson lab. And what I was able to join was having available a brand new YAC library, actually a series of YAC libraries that really had just been made, but didn't really have methods for how to use them. But this was the key technology that was going to then be available for the early mapping of the human genome. What I applied to it under Maynard's guidance was another key technology that happened to come out a few years earlier, and that's, of course, PCR. When I walked into the Olson lab, I was shown where there were 3 water baths to have 3 temperatures because the Department of Genetics in 1988 did not have a PCR machine. I can tell you, it wasn't until Maynard got me a PCR machine, and it wasn't even on this history of PCR machines, it was even before that. And it didn't have Peltier heating and cooling. It had hose that you hooked up to the water, in the sink, and that was what was cooling. It was very prehistoric, but it worked. And what I quickly learned is there's only one thing more popular than being a dermatologist, and that's having one of the only PCR machines on this campus. Because people would then line up, can I use your PCR machine? And I had to end up having a sign-up sheet. But I ended up doing some of the first PCR in the department. And what Maynard and I had cooked up was this idea of the way to isolate clones of interest with pieces of DNA of interest was to develop a scheme that didn't involve hybridization, but involved PCR. And we published this in PNAS in 1990, which basically was developing PCR assays and having a scheme to identify which clone in the library had that targeted sequence that could be detected by that PCR machine. It then became apparent to Maynard and I and others that not only could you isolate YAC clones, big pieces of DNA with PCR, you could also use those landmarks that the PCR, the elements of DNA that are being detected as landmarks. And the idea was that if you just had a PCR assay that could identify a unique piece of the human genome, that becomes both a tool for getting the clones and a tool for mapping. And Maynard loved this idea, as did others, and they wanted to really propose this as a way forward for building the first map of the human genome en route to getting it sequenced. And Maynard got together with some of these luminaries, as you can see, to write this perspective piece, but they had to have a name for what we hadn't named it, the sequence tech site. And it's just one of these vivid memories I have standing out in the hallway right outside the autoclave room for like 2.5 hours. And I swear every senior member of the department walked by, but it was Maynard and Phil Green and I swear Mark walked by, I swear Bob walked by, and we just kept throwing out different 3-letter words or things to do. And after about 2.5 hours, we settled that sequence tech sites were the right name for this, and that's what they put in this perspective, and off we went. And that, I then took this strategy out for a real test ride. We went for the cystic fibrosis region, which was the first really large region of the human genome to be mapped using this STS content mapping strategy. The strategy is shown here, and Phil Green and I and Phil had written software and gotten involved in some of the theoretical aspects. And there's a paper that these are the two major publications that sort of showed the way for how to build these large contig maps of human chromosomes. The Genome Center that David Schlesinger was the PI of, and Maynard and others, and Phil and I and Helen Donis Keller, and et cetera, were involved in, had two major goals. David Schlesinger was overseeing the mapping of the human X chromosome, and I signed up with Maynard to map chromosome 7. As soon as they got the grant, Maynard said, see you later, I don't want to do this, you do this. And so he gave me full opportunity to sort of run the show and run with this on behalf of the Human Genome Project. I started building a lab even when I was a postdoc. Bob Fulton was one of the early technicians who I brought into my group, and off we went to build maps. It worked. Instead of displaying them at Cold Spring Harbor, we displayed them in the hallway. These are the kind of contigs that we were able to build. And if I just fast forward, one of the very best YAC-based maps of a human chromosome was chromosome 7, which we finally published in 1997. And we've got basically the entire, or essentially the entire chromosome in 22 contigs, and a given contig you can see is quite deep and quite broad and really quite helpful, and served as a foundation for moving forward to the next phase to eventually get back maps and eventually sequence it. Let me emphasize, the brilliance of STSs that Maynard saw is very much in the spirit of what Bob was talking about with data sharing. Is what we loved, Maynard and all of us loved about STSs is you didn't need to share anything but the sequence of your 2 primers. And so it was always this idea that it was purely transferable as a marker, as a tool, just based on sequence of 2 oligonucleotides and maybe some information about how to do the PCR. You didn't have to mail reagents, you didn't have to plead for reagents. It was just required. If you're going to describe an STS, you got to tell us the sequence of those primers. So this was a success story. But let me take you back to make sure you realize this was not easy. And what the times were very hard times, and we faced some obstacles. Some realities is first realize that when the Genome Project began, we didn't have a bunch of genomicists. The word had just been coined. It was really a toddler field. And just like you had mathematicians and worm biologists and physician scientists trained in pathology all coming together, different combinations of things, that's how the field was basically founded. Oops, the second thing, there was no detailed plan. There was some early idea how to get started. Trust me, the entire Human Genome Project felt like this. It was like building an airplane while flying it. And don't you think that looks like Bob Waterston? Every time I see that cartoon, I thought, what a great cartoon that is. And you know, we just got to keep going, even though we would hit all sorts of barriers, obstacles, hassles, venters, the whole thing. And you know, you just handed out vomit bags every once in a while to keep yourself going. But there was no start to finish plan. The other thing that there wasn't was consensus that this was a good idea. And actually, Bob asked me to make sure to include a little bit of this in my talk. The scientific community had very mixed opinions about the Human Genome Project, which was a little scary, even for the mavericks like Rick Wilson and myself and Elaine Martis and Pui Kwok and others, that we were doing something where some people were telling us we were sacrificing our careers. Well, why were there mixed opinions? I mean, young people might think, well, how could the Human Genome Project be a bad idea? It's created what we have now. But there was really some serious pushback on this. And I will tell you that my former institute, NHGRI, we had a history program, and we recently created an exhibit that talks about a writing campaign that I want to share you just a little bit about that took place just before the Genome Project started, where they really tried to stop the Genome Project, scientists truly trying to stop it. I mean, it was all described in a New York Times article that came out 3-4 months before the Genome Project began. And it says, the Human Genome Project is bad science, it's unthought out science, it's hype science, said Dr. Martin Reichsteiner, a biochemist at University of Utah. I know there's somebody here from University of Utah, I'm so sorry. Some critics have been aggressive letter writing campaigns urging colleagues to harbor similar sentiments to write Congress. This was all based on this very lengthy letter that this Martin Reichsteiner mailed in to some key people a few months earlier, where he said, if you agree, and he distributed this widely, if you agree with the arguments presented, then write Senior Advisor Alan Bromley, NIH Acting Director William Robb, Senator Al Gore, Senator Ted Kennedy, and express your reservations, concerns, or opposition to the Human Genome Project. In short, the Human Genome Project is a waste of national resources and is detrimental to the training of young scientists. I urge you to take the time to voice your opposition. 5,000 letters would have a significant impact. They ended up getting 55 letters. Let me show you what a couple of these letters read like, and you will see what we were up against. So this gentleman that I'll redact the name, from University of Minnesota, talked about how the Human Genome Project has a costly, wasteful, and inappropriate allocation of research funds. The Human Genome Project, little useful training and no intellectual stimulation to young scientists like me and Rick Wilson and others. Finally, 95% of DNA does not code for proteins and is thought to by many, including advocates of the Human Genome Project, to be junk. This junk argument was just pervasive, even from somebody as wonderful of an institution as my alma mater, University of Wisconsin, who wrote, "I am certain that most of the sequence information that is generated will never prove to be of any scientific value, especially since probably greater than 90% of the sequences are non-coding or junk DNA." And this is what they thought the genome was all about. And I would contend that's completely untrue because we can make beautiful, beautiful double helices from junk. But they went on to say the Human Genome Project is a brute force and indeed stupid way to determine those sequences that will be of biological interest. That's what we were up against as young researchers. And that's what we had to face. Similarly, in another accounting, it's not a letter, but in this article that was written by Michael Waterman, he talks about that while drinking brandy after dinner, Wally Gilbert wondered how to find the labor to do the boring, repetitive sequencing. Gilbert proposed that we use prisoners to do the work. Give one group the Crick strand and the other group the Watson strand, and they'd create competition and quality control, he said. I knew that Gilbert had no idea what he was talking about. And I did come up against scientists and said, why would a physician scientist be involved in the Human Genome Project? It's really work for prisoners. And when I would get really, really dejected, I would go talk to the moral compass. I would go talk to Bob. I'd say, Bob, they're being so mean. They're telling us we should, like, it's for prisoners. This is not for us. Bob, how do you stay so optimistic? How do you stay so happy? I don't get it. They think we're prisoners. And Bob would say, no, Eric, you just have to understand it. Haters are going to hate, baby. I'm just going to shake. I shake it off. It was brilliant. The problem is that Bob didn't think to trademark his idea, because had he done it, the department would be the Swift Department of Genetics as opposed to the McDonald, because her pockets are deeper than McDonald's. I can tell you that in any case. Oh, and by the way, the other problem with the Genome Project, besides the haters and the lack of a plan, was that we didn't have an internet very functional at all. The Genome Project started, this is how sequence was distributed by GenBank. Then we upgraded. It was upgraded. Then they started mailing around CD-ROMs from GenBank. You'd get it like every few months. And that's how you had your sequence. And it did eventually get better, but not at the beginning. And now I'll have to tell you a true story. It is absolutely true that the Department of Genetics fax machine was the one that very early on, I had a collaborator named Bob Moises out at one of the national labs who was sorting chromosome 7, and then he was cloning bits of chromosome 7, and then he was sequencing small bits of chromosome 7, and he didn't have a way to transfer it to me electronically, so he would write out the sequence and fax it to me. And we didn't even have a plain paper fax machine. It was one of those, thermal things, and they would curl in the notebook. It was horrible. But literally, the first 100 or 200 STSs made on chromosome 7, the data for it came by fax, if you can believe how prehistoric that was. True story. But nonetheless, we did it. The Genome Project was wildly successful. Great celebrations, ended in 2003. And since 2003, we've now had 22 years of reading out the human genome sequence, interpreting it, et cetera, and it all started up, in McDonald Sciences building in around 1990. So in the last just couple of minutes, let me just quickly tell you that, my journey changed a bit. I got recruited away from Washington University by Francis Collins in 1994 to join when he became the director. And so I moved from St. Louis to the NIH and have been at the National Human Genome Research Institute until very recently. I arrived as a mid-term tenure track investigator. I became the intramural director by 2002, and then in 2009 I was appointed the director. And I was very proud to be among one of the first, or the third director of 3. There was Jim Watson, and then Francis Collins, and then me. I was so excited about becoming the director of the Institute 15 and a half years ago because I really saw the promise that was first described when the Genome Project ended of melding together genomics and medicine, because that's what everybody was talking about when the Genome Project ended. And yet, while Francis had started to get moving in that direction, even when I took over the Institute, I think genomic medicine was a very blurry concept. We didn't really know what it was going to look like. We didn't really know how we were going to get there. But between when Francis led the Institute just prior to when I took over, and then as I had the directorship of it for 15 or 16 years, we saw a lot of magical things happen. We saw the cost of sequencing a human genome to reduce over 1 million-fold. We saw millions of human genome sequences get generated, lots of information about genomic variation, profound advances in understanding how the human genome functions, and significant advances in understanding the genomic base of human disease, especially rare genetic diseases. And genomic medicine is starting to be seen on this campus and really all around the world, whether it be in cancer genomics, whether it be in the diagnosis of rare diseases, undiagnosed conditions, or rapid genome sequencing for acutely ill newborns. I'm thinking about incredible opportunities for prenatal, non-invasive prenatal genetic screening, pharmacogenomics, and others. And what this has led to more than anything is bringing genomic medicine into focus in a way that I'm not sure any of us could have predicted. I certainly can tell you that when I was meeting with Jeff Milbrandt, he convinced me to go into lab medicine. I never thought this would happen in my lifetime. And the fact that it happened on my watch is extremely gratifying. And associated with this has been a remarkable growth of the field from just a small group that Bob and I have introduced you to when the Genome Project began to a much larger, more interdisciplinary and increasingly clinically involved group, as you've heard even some presentations at this symposium today, and that's incredible as well. But things have changed, because then came January 20th. This happened. And then this happened. And you may say, well, what happened to you? Well, I drew the short straw. Institute directors at NIH, we have 27 institute directors, you have to be renewed every 5 years. And my 5-year, I'd been there for 15 years as director, my 5-year renewal date was coming up on March 10th. I was the first of the 27 to have to be renewed under these people. And they chose just not to renew me. So I did not get reappointed. And as all of you, or many of you may have read, I call how I failed to dodge the bullet, if those of you know how much politics you know. If you know what Doge is, yeah, I couldn't dodge the bullet, and I was out. So that's what happened to me last month. I have to learn to laugh about it, because that's the only thing I can do. But I am proud of being some of the notable firsts. I was delighted to be the first institute director appointed by this guy who's smiling. And then, presidents don't smile anymore. That's the first thing you should know. And then I ended up being the first institute director now of 6 to be fired by Trump too. And yes, indeed, there have been 6 of us who have now been terminated, and I think there's probably more coming as part of a slow decapitation. People have been wonderful. Many of you have been wonderful. Lots of wonderful emails and calls and so forth. My favorite was a personal note I got from a colleague of mine with a card that said this: "Thank you for not being a conspiracy Adel science denying idiot." It does tell the story. Now, some of you might be interested, what do I think is happening at NIH from what I've seen? And I talk to people there every day. I orphaned a very large staff of 500. We were at 650, now we're down to about 525. Basically, the current state of NIH looks something like this. I was on campus just last week. It looks something like this. And I worry, 10 years from now, it might look something like this, with those of you who know the Planet of the Apes. I wish I could tell you I'm optimistic, but I'm very worried. I do wonder, just like the New York Times, what is actually the point of treating the NIH this way? I lived through, I was no longer the director, but April 1st was a very dark day where some of my best people were laid off. And 4 more institute directors, and now is 5, plus me is 6. And they're doing this to increase government efficiency. Get me going at the reception. I'll tell you about how inefficient this is. Radical transparency is one of the main mantras. Yeah, get me going at the reception. I'll tell you, they fired every communication staff at every institute. Nobody's updating their websites. That is not radical transparency. Oh, we're making America healthy again by terminating all of these grants and no longer doing COVID research, no longer doing lots of other research. It goes on and on. I don't buy it. I'm very excited to hear what is exactly the cause of autism by September, as we've been promised. That's great. I'll believe it when I see it. So that's what I could say. But I don't want to end on a negative note, and I decided my last slide had to be a positive note. And if I thought about it, it sort of ended where Bob began, is to say thank you to Maynard. I mean, he gave me this incredible opportunity, and I feel like it changed my life. And it was all because he was just a magnet that just brought me and others in. Here's a picture of Pui Kwok and I and him in Taiwan. We were all together there for a week in July of 2023. And then the last thing I want to show you is I've had a picture of a very special thing that was left behind by Maynard, and I've looked for a reason to show it, and I haven't shown it because I've never had the appropriate reason. But I'm going to show it right now. Because what happened, and one more little anecdote, when I became a faculty member in 1992 was exactly when Maynard was leaving. And in a typically gracious way, Maynard said, Eric should just take my lab, physical lab up on the 8th floor. And so he actually moved out of lab and let me start to move in, and Bob Fulton helped me set it up, and so on and so forth. And then came the day in 1992, Maynard was gonna leave. And we knew what day he was leaving. Now, any of you who know Maynard, he's not one to come around and make sure he says goodbye to everybody, because he's just a little too shy for that. But instead, he just sort of disappeared that day. And then I never said goodbye to him on that day. And then when I came into the lab the next day, this is what he had left. He said, sorry, I could not find everyone for a personal goodbye, because he didn't look. He said, stay in touch and turn out the lights when you leave. And that is prototypic Maynard, and something about the spirit of the department as well. So I think I may have got a few minutes over, but I hope at least, if nothing else, I entertained and educated you all. Thank you very much. Okay, so now any hard questions you ask Bob, any of the really easy ones I'll take.
So we can't leave this without some questions, so we're gonna have 5 minutes of questions.
Mark had his hand up even for Bob, so
Bob, your goals in the early 90s to sequence the worm genome were incredibly audacious. And I'm wondering, when did you start thinking of sequencing the human genome, which seemed impossible at the time? When did you start thinking that was going to happen?
Well, so that happened on a visit to the Sanger Center. When John Sulston took on the leadership of the Sanger Center, that was funded by the Wellcome Trust. And they had the expectation that he would do human genome sequencing. And that, we were sitting in his backyard, sipping sherry. We'd had our lab meeting, our joint lab meeting. We took all the people from our center and transported them for 3 days over to Cambridge. John had been depressed the whole meeting. And he, I don't know, he was just, I said, what's the problem? And he just said, you know, the Wellcome wants me to sequence human, and we don't know how to do it. And so on the plane ride the next day, I started thinking, I got to do something. And I got out my calculator watch. You don't know what a calculator watch is, but
super nerdy. I had one too.
And I started doing numbers, and I realized that the advances that we had made, Phil Green's software in particular, but advances in all the different steps of sequencing. It's been described as a process engineer's nightmare. There are a million steps, and none of them, you solve any one of them, it doesn't make much difference, but you got to solve all million. Anyway, we had solved enough of those to make it possible. And I had the numbers, and the next day I sent them to John, and so then we decided we knew how to sequence the genome.
And that was 5?
94.
94, oh my
September
God.
1994.
Okay. And I just want to point out to the audience one more thing. In addition to reading John Sulston's book, I can highly recommend to you a memoir that Bob Waterston wrote that was just recently published in the Annual Reviews of Genetics and Genomics. I urge you to read all of that. You'll get more.
Thank you. Hard question. So I work on genomics every day. So I know it's very important. I totally buy this huge achievement for humankind when we have the human genome sequence. But when we look back, did we overpromise to the public about the impact of the human genome? I mean, did we underestimate the difficulty to reinterpret the genome 20 years later? That's my question.
So, I don't know, you want to answer
Oh,
that?
no, I'm so used to that question, but I don't get paid to answer it anymore. I'll answer it, but you go for it.
Well, you can counter.
I'll counter. No.
But I think I think we underestimated how hard it was going to be, at least certainly some people underestimated. There were others who were skeptics. How hard it was going to be to make an impact on clinical medicine. And as Eric said, it's starting to happen now, and it still isn't where we might have hoped it would be, even in 10 years after the genome was sequenced. But I think the other area where we probably underestimated its impact is on biology more globally. And certainly from my perspective, you don't do anything in biology without a genome now. And I did not see that coming.
My answer to this is typically that if you read what people were saying in the euphoria of completing the Genome Project, there was probably a little bit of hyperbolic language of excitement. However, and if you take it totally exactly the way it was said, maybe that's a little bit of overpromising. I think if you would have asked anybody in 2003, when we had just spent $1 billion or so, or whatever number you want to use, and you were going to say that it wouldn't even be 2 decades before we would have reduced that to the cost of a very reasonable clinical test, people would have said, you're crazy, it'll take you much longer than that. If you would have said within 20 years, we'd be able to take an acutely ill newborn and within 24 hours get a diagnosis and save some fraction of life, they would have said, there's no way. So there's enough successes, but for the same people that just thought all the DNA and most of the DNA in the genome was junk, well, it turns out to be really complicated stuff. And so you're right. We've barely scratched the surface. And if anything, I would hope the young people in this audience would get just as excited about genomics now as we were then, because it's a hell of a lot more complicated than we thought. We need probably even more talented people than us to figure out all the complexities and then to fully operationalize it, to maximize its contributions to medicine. Because I only talk about vivid examples of the early examples that are exemplars, but we've got a lot more to do.
No, in terms of that, the future is still there.
Well, more than ever.
You're lucky.
Barack, we're going to let you finish up the questions.
Eric, as you were talking about the list of things you didn't have, I think there's one more really important thing on that list that you didn't have, which is the sequencing technology, right?
Right.
It depends. I was a graduate student in 1990, and I think in 1990 we were still using slab gels that worked about 1 in 3 times to get about 150 base pairs. So
but here's the key. No, you're absolutely right. Depends how you look at it. But I vividly remember, Bob's the one that said, that really changed the discussion, was when the Genome Project began. It's actually written in some of the strategic documents. We will not start sequencing for serious, certainly not the human genome, until we found something other than Sanger sequencing. So the fact is, I think it was shocking in retrospect that we actually used Sanger sequencing to actually get the first sequence of the human genome. It happened because of getting away from slab gels. It happened because of Phil Green software. It happened because of automation, capillaries, blah, 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 blah. But going in, I don't know, Bob, did you think when we started the Genome Project, we'd still be using Sanger at the end of the Genome Project? We'd have new technology.
Well, I was worried about the first year. And Sanger was what we had.
Yeah, so you just did
And we had 1 million steps, and we had to make each one of them better, and that's what we came in to do. Every day, it was something different. You fixed one problem, and something else was the problem, and we had to fix that. And it wasn't just our center. It was, big dyes, attack polymerase, substitutions, robots, and it came in from everywhere, but we had to make it all work over in 44. And so it worked the first year, it worked better the second, and by the 4th year, we knew it was good enough to do the human.
Let's thank these 2 people one more time.
Okay. Thank you, Bob. Thank you, Eric, for a pair of outstanding talks. So our next speaker is Heather Broihier. Heather Broihier was the first postdoc in my lab. Heather was great, and I knew enough at that point that if you have someone really, really good in your lab, the absolutely best thing that you can do is get the hell out of their way and let them succeed. So I'm incredibly proud of Heather. She's a professor at Case Western Reserve University Med School in the Department of Neuroscience. Heather?
All right, thank you. Thanks to Jim and Susan and the organizing committee for inviting me to speak today. It's really an enormous honor to be here. I feel like I drew the short straw of the seminar list. I have to follow that, but I'm going to do my best. So when I was putting this talk together, I actually went back and looked at some of my old lab notebooks from my postdoc and made a little timeline here of when I was in Jim's lab. I started in early '99 and left to start my own lab at the end of 2004. And I did my PhD in a genetics lab, spent my entire PhD doing 4 genetic screens in Drosophila, felt like I had learned kind of what I needed to learn about doing genetic screens in Drosophila, so the thing to do now was to clone a gene. And I'd seen plenty of postdocs come into my lab, my student lab, and spend years doing a screen, and then they had to clone the gene to get the job. And so my bright idea was I'm just gonna skip the new screening part and come in to clone a gene. I met Jim at a Drosophila genetics meeting, maybe like 1997, coming out of a super fantastic postdoc in Christo's lab where he had generated an enormous number of really cool mutants that affected neuronal cell fate in the Drosophila embryo. And I thought, okay, that looks super cool. I'm gonna go in there, clone a gene really fast, you know, be off to the races. Okay, didn't exactly go that way. So I started early 1999 and until, so 99 through the beginning of 2000, I accomplished absolutely nothing toward cloning this gene, taking kind of old school genetic techniques to try to find a gene he called XX that was responsible for a change in cell fate in the Drosophila embryo. Nothing was happening. I should have just taken a vacation. End of March 2000, the Drosophila, the first iteration of the complete Drosophila genome was released. And we knew enough about where this gene was in the genome that I could just take a candidate gene approach. And I first started by just doing in situs across the region to look for genes expressed in cells that could maybe be relevant. So I actually went and looked at my old lab notebooks, which was a hoot, and found, so that's about 3 months after the Drosophila genome was released, I actually set up the sequencing gel to sequence the alleles of this gene that Jim had identified. And that results of that sequencing run were the first figure of the first paper that I published with Jim, which came out about a year later. And so, for those of us who drink the Kool-Aid on model systems genetics, this was such an incredibly kind of gratifying experiment to do. Jim had generated 4 mutants of this gene, and I haven't thought about this gene in 20 years, but when I went back and was looking at it, I still remember exactly what these alleles are called, 20 years later, because I spent so long trying to figure out what they were. The 4 mutants that he had identified were all stop codons in the coding region of one specific gene in fly. And it turned out to be the Drosophila homologue of a major motor neuron determinant in mammals called HB9, which seemed prescient since my name is Heather Broihier. So that was all great. We submitted the paper about a year after I ran that gel, and was accepted shortly thereafter. And in the meantime, I realized that there was actually a cohort of kind of motor neuron determinants that were conserved across phyla, so mammals to flies. And so I just went after them kind of simultaneously. So another one, the next paper we published was on NKX6, which is another of these guys. Did the cloning late 2000 and published that paper in 2004. Like all good postdocs, I had a side project. Mine was in human genetics, and it was published in early 2003. So there was a super, super exciting time to be a member of a outstanding genetics department. I felt incredibly fortunate to be in Jim's lab. When I was thinking about memories I had of the department, one of them involves Mark Johnston that he probably doesn't remember. I was giving a practice talk for faculty jobs, and I had already come through one pretty harsh round of critiques of this, so I was feeling like this was like a really good talk, and I was expecting the faculty to tell me this was like awesome, and you were not impressed, and you listened to the talk, and then you followed me back to my bay, and you took my laptop, and you started changing all of the titles of my slides because they were too long and the fonts were too small. And if you remember this, but it had enormous impact on me because now I constantly tell my students, I can't read your font. Okay, so I left in late 2004. Yeah, you know, I tried to make them bigger, but it looks so big. All right, it's bigger than I wanted. Mentorship from Jim did not stop when I left the lab. So I was talking to Swathi, and we thought we should come up with a term for Jim's treatment of your text, that we could call it ski-thized. So I went to Case Western, I wrote a grant a couple months after I started, and I sent it to Jim, and he FedExed it back to me in a box with, I think, more handwritten pages than I had sent him. He just, by hand, and if you've read his written text, it's actually pretty tricky to read, really small font, page after page after page, rewriting this grant for me. And I mean, I learned a tremendous, you know, you don't get taught how to write. I learned a tremendous amount by actually incorporating the comments that he gave me on that grant, which I got. So, you know, I'm just felt incredibly blessed to have been part of this department at that time. So I'm in a neuroscience department now, and I love the Drosophila embryo more than anyone, but you know, you really can't study kind of experience-dependent plasticity in the Drosophila embryo. It's just not very well suited for it. And so most of my lab now has moved out of the embryo, and we work on early adults. And what I'm going to try to convince you of today is actually that baby flies are a whole lot like baby humans. I mean, you can't argue with the first one, right? That's totally obvious. They're both super adorable. They both sleep a lot. Okay, so if you're thinking about fly lifespan, roughly a day of a fly life is equivalent to a year of human life. So first day of fly, first year of human, 2 years of human, 2 days of fly. So it's really remarkable that baby flies, so flies over the first 24, 48 hours, sleep a lot more than older adult flies do, and they particularly sleep a lot more during the day. So that's data from Amita Segal's lab on the left, where you can see it's first day of life versus later life, and you can see that day-old flies sleep about twice as much during the day as older flies. And for sure, any of you who have babies or have had babies know that they sleep all the time in the day and sometimes not so much at night. The other really fascinating thing, if you're a neuroscientist, about young brains is they're incredibly plastic. You know that from humans. If you watched your kids learn language, learn motor skills, adapt to their sensory environments, that all happens very rapidly when they're very young. In particular, their sensory systems come on very early. Flies, it's the same. So the data on the left is classic data from our field. They're actually looking at the olfactory system that I'll describe in a little bit of detail for you guys. Looking at the structural plasticity, so in this case, just the size of a synaptic region, so a region of connections in the olfactory system in the fly. This is a particular region of the Drosophila brain that responds exclusively to carbon dioxide. And if you're exposed to high levels of carbon dioxide during the first day of life, the size of this little synaptic region shrinks a lot, but only during the first couple days of life. If you do it later on, there's no change. So there's a really remarkable ability of the early CNS to adapt to sensory inputs. So, okay, so that's cool. We were motivated, let me describe a little bit about the circuit for you. So the left, so as the sequencing of the genomes has created a revolution in genetics, the mapping of the connectome in flies has similarly revolutionized the kinds of neuroscience that we can do. So the left is actually a hemibrain from the Drosophila connectome, and the right is our diagram. And so we're going to focus just on one very simple sensory circuit in the olfactory system, so smell circuitry. And we're looking at the response of the flies to ethyl butyrate, which is a super sweet odor that to me smells like cough syrup. So there's a particular flavor of neuron in the fly brain that responds to this odor. The neuron is called OR42A, okay? It projects its axons into a defined region of the antennal lobe, so the olfactory system. That little region is called a glomerulus. It's just a segregated set of synapses. That little region is called VM7 for this particular class of odorant receptor neuron. Those presynaptic terminals connect exclusively with one specified type of a postsynaptic neuron. Those postsynaptic projection neurons then project to higher order brain regions. We know we have genetic access to all of these cell types, to the inner neurons that interconnect the glomeruli, as well as to the glia that infiltrate, and we'll talk about that in a minute. So we know about this circuit, beautiful genetic access. We had done previous experiments in my lab suggesting that it was pretty sensitive to odor during early life, but we wanted to ask whether there was actually a true critical period for odor responsiveness in young flies. And so we're motivated to do that by this guy, who's Conrad Lorenz, who is an Austrian ethologist working in the early part of the 20th century, who's kind of you can think of as the founder of animal behavior. And so he was interested in goslings, baby geese, and so, came up with the concept of imprinting. And so you all have some intuitive idea about what imprinting is, but the really kind of magical thing about imprinting is that it's very tightly controlled. So what he found was that if you expose goslings between 13 and 16 hours after hatching to a moving object, they'll imprint on it and consider it their mother. That's why he's got them following him across this field. But before and after, no. Okay, so critical periods, the cardinal features of critical periods are defined opening and shutting of the window and lifelong consequences. Okay, so whatever that magical plasticity is that's happening during that window, it stays with the animals for life. So we wanted to test whether the circuitry that we were looking at in the olfactory system could be considered a critical period by those measures. These are the guys who've done the work in my lab. So the first three are current MSTPs. Paola is a super talented technician. Abigail is a senior in my lab who's going to apply to MSTPs in the next cycle, and I'm going to talk about her honors project at the end. Okay, so this is just a test to see whether we can define a critical period. Basically, all my data looks like this, so if you just pay attention for this one, then the rest of it will be pretty easy to follow. So we're showing the exposures at the top. We expose either to mineral oil or 15% ethyl butyrate during the window that's defined. So DPE, that's eclosion is birth for a fly, so that's basically how old they are. Okay, so in this first experiment, we were trying to test when the critical period opened, so we're exposing for the last 2 days of pupal life. Okay, to either mineral oil or ethyl butyrate. We're then we have a automatic analysis where we can evaluate the volume of this neuropill in the fly brain to ask if it's changing in size. Okay, this is a really crude measure, but you can see there's no change if we expose to ethyl butyrate for these last 2 days of life. Last 2 days of pupal life, excuse me. If we expose to ethyl butyrate during the first 2 days of adulthood, we get about a 50% reduction by this measure in glomerular volume. Okay. If we do that same experiment just 2 days later, from 2 to 4 days, there's no change. Okay, so this critical period has a clear opening and shutting, which met our first condition. Then we wanted to ask whether there were lifelong changes. So I'm going to show you by a couple different measures there are. So here we did the same exposures, 0 to 2, but instead of dissecting immediately after, we waited 5 days and then dissected to basically ask whether the changes persisted. Here we're not looking at volume, we're looking at presynaptic content. And so you can see there's a pretty dramatic reduction in the number of presynapses. Hopefully you can see with my small font. Reduced number of presynapses 5 days after we stop the exposure. Okay, so that's a structural measure. We then wanted to do some calcium imaging to actually look at whether the odor-evoked calcium transients were down in response to odor and whether they stayed down. So in collaboration with Andrew Dax's lab, we did calcium imaging of the primary sensory neurons. So these are the OR42A neurons, either exposed to mineral oil or 15% EB. You can see that early ethyl butyrate exposure leads to a reduction in the calcium flux in the OR42A neurons, and the decrease in calcium flux is maintained over lifetime. That actually data point on the bottom is 25 days post-eclosion. So for many, many days after we do the odor, the decrease in calcium responsiveness is maintained. So that met our second condition. Everything I've been showing you so far is just looking at the presynaptic side. We can also look at the postsynaptic side because we've got the genetic tools to do that. This slide just shows you that the remodeling that we see in this circuit is confined to the presynaptic side. We don't see any change at all on the postsynaptic side. Then we wanted to ask, okay, we've really decreased the presynaptic content of the presynapse. The postsynaptic looks okay, but you would expect if the presynapse is impaired that postsynaptic responsiveness would be likewise impaired. And so we collaborated with Masashi Tabuchi to record spontaneous evoked potentials from the postsynaptic cell. You can see it's dramatically reduced immediately after odor exposure, and then it stays reduced for 5 days, indicating that, again, supporting the calcium imaging data that we have, arguing that this change in circuit function during these first 2 days has lifelong consequences. Okay, we're losing the synapses. What is happening to them? I had worked on glia for many years before that and was suspicious that this was an example of experience-dependent pruning by glia of the synaptic terminals, and so we tested that. 2 different glial subtypes that infiltrate these glomeruli. One is astrocytes. Fly astrocytes are a whole lot like mammalian astrocytes. They associate intimately with synapses. Other glial cell type is ensheathing glia, which they're not professional phagocytes the way that microglia are. They have different functions, but they have high phagocytic capacity. Okay, if we're testing for a role for glia in pruning these synapses, the prime culprit who we should look at if we're trying to find a genetic component of this pathway would be Draper. This is fly SED1. It's the major engulfment receptor in flies. Nearly every kind of phagocytic paradigm that you look at in flies has a phenotype in Draper mutants. So we knocked out Draper. It's in all glia in this experiment, and you can see it completely blocks the loss of the presynaptic terminals that we'd seen with 0 to 2 day odor exposure, arguing that glial Draper is required for experience-dependent remodeling of these terminals. In data I'm not going to show, we could actually map the cellular locus of Draper function to ensheathing glia, not astrocytes. Okay. This is an experiment that a reviewer asked us for, and I hadn't wanted to do because I thought it would be a waste of time, asked us to record from Draper mutants. And I thought, okay, there's no point recording in Draper mutants. The presynaptic terminals have probably degenerated. We're going to record. Activity will be reduced, but doesn't tell us Draper isn't doing anything. But totally remarkably, when we record from the projection neurons in Draper mutants, activity of the projection neuron, so the postsynaptic neuron, looks identical to that of the mineral oil exposed animals, arguing that glia are actually playing an active role in degrading this synapse, which we're super interested in following up on. Okay, so this was just mostly what I wanted to tell you today about our work defining this critical period plasticity. Just as a very last little thing, we're geneticists. I'm in a neuroscience department, but I'm still a geneticist, and so geneticists screen. And so it's very easy for us to do cell type specific manipulation in this system and try to identify both neuronal cues and glial cues that regulate this experience-dependent plasticity. So we're screening RNAi knockdown in both neurons and glia at 2 time points, both immediately when the critical period shuts and then 2 days later, because we're curious if we can find any mutants that keep it open too long, that change the timing. It's just this one recent run that I'm showing here. We're focused on innate immune signaling molecule because I have a longstanding interest in them, as well as human disease-related genes. So to my dismay, my lab loves showing me when my pet hypotheses are wrong. So I've been interested in toll receptors for quite a while. In this run, neither of these guys look very interesting. This is glial knockdown, I should say. So glial knockdown of either of these tolls doesn't seem to be doing much, but glial knockdown of both of these human disease genes do. So UBE3A is the Angelman syndrome gene, and NOT is the fly EBF3, and I'll talk about that in a second. And both of those in glia loss of those guys in glia completely blocks this experience-dependent plasticity. My great undergrad decided to follow up on NOT for her honors project. So we've now taken this multiple RNAi lines. You can see that knockdown of NOT in these cells totally blocks critical period pruning. And what is super cool is that it's not ensheathing glia NOT, it's astrocyte NOT. And so that for the first time points to a role for astrocytes in the regulation of this critical period pruning. So what is EBF3? It's the cause of a syndromic neurodevelopmental disorder that's characterized by intellectual disability. And as far as we can tell, no one is even thinking about glia as regulating this being required for this particular etiology. So we're super excited to follow up on this. I want to give a plug for Abigail, who would be an asset to any MSDP program in the country, and she's applying this year. She was the national runner-up at both the 1 meter and 3 meter springboard diving, and she won the award for the top GPA at the NCAA site for all swimmers and divers. So it's the first ever, and actually in my school's history. So she's been a delight to have in the lab, and I think in these really uncertain times when it can be really hard to be a PI right now, having amazing trainees like this who we are able to shepherd to the next steps in their careers has been super rewarding. So just in conclusion, we're really excited about this paradigm. Most people who study pruning in the flies study kind of the dissolving of the CNS that happens at metamorphosis. And so, it's kind of hard to convince mammalian reviewers that that's a relevant form of synaptic pruning. And so we think this is experience-dependent pruning may, be an important model to uncover new genes required for human neurological diseases. So thank you. I have one question. I'm told. But I beat the buzzer, so that's good. I can hear you.
Okay, yeah. So super interesting. So I'm wondering if
I didn't beat the buzzer.
I know. And then PS3, like I said, expressing human, do you have single cell data? Is this precise?
It's in astrocytes in humans, yeah.
And also, I guess it's like a system of the system or is it peripheral system that you think is working?
In our system, we haven't looked at mammals at all. In our system, this is CNS glia.
So we would really love to get a picture of all of you to document this event today. And so when the break comes, we're going to head to the big stairwell in the FTLC. If you want something to drink, grab it. We'll take a quick picture and try to be back here for the rest of the talks after we hear Dawn.
All right, our next speaker is Dr. Don Conrad, who is a professor and chief of genetics at the Oregon National Primate Research Center. And he actually began his independent career at WashU in the Department of Genetics.
Okay. Is this on? Is it working? Okay, great. Thank you to the organizers for inviting me back. It's really just a huge honor and blessing to be here. As was just mentioned, I started out my faculty career here from 2011 to 2018, but I was no stranger to WashU. Much like Eric, I was born very close by at St. Mary's Hospital on Clayton Road, and I grew up at the intersection of Del Mar North and South, and then spent hours in a computer lab on the Danforth campus, and my dad was studying for a degree in computer science. So I could think of nothing better to do as a child than to come here and do science. So it's really special to be here for this celebration. So the work I'm going to tell you about today is directly empowered by having a human genome sequence. I could never have done this without it. I do a lot of things, but my main sort of long-standing interest has been trying to understand the genetics of human male infertility. And it's actually surprisingly common, like given the social invisibility of the condition, you can just see some raw data. This is data from a collaborative of mine from the University of Utah, in which they did semen analysis on 700 random volunteers at the University of Utah. You have this typical L-shaped distribution of sperm concentration, which is the million of sperm per milliliter of ejaculate. You have this kind of like L-shaped distribution. The median is somewhere around 75 million per mil. The WHO definition of oligospermia or low sperm count is having less than 15. And that has about a 15% prevalence in this population. But what I find really shocking is you go to the most extreme end, men with zero sperm in their ejaculate or azospermia, there's a 1% prevalence. And that's a number that's pretty consistent across the world from clinical groups that do these kinds of semen analyses. It really depends on the context, like the environment, what kind of breeding system you have. There are places in Pakistan, for instance, where consanguinity is pretty common, and you might have a number like 3%. Some populations quote like 0.5% or 0.25%, but it's pretty common. I'm going to see if I have a clicker. Is this cork? Yes, great. Okay, so when I was, just like a lot of people and some of the journeys you've heard today, and my postdoc thinking about what I wanted to do to launch my career and how I could translate my expertise in genomics, which I had just picked up at the Sanger Institute, I saw a great opportunity in male infertility. It was really kind of, apologies to David Page, who was the leading man in this space. It was kind of a backwater, other than David. This slide is kind of showing you the same kind of data that I was looking at, albeit 10 years before, in which we knew at that time from spontaneous mutants or targeted mutants that weren't meant to study male infertility, we saw that there was something like maybe 50 to 75 genes that when you knock it out in mouse, you got azospermia. So we knew that there was a strong sort of case for monogenic forms of male infertility in mammals. And then if you looked at what was going on clinically, what was being used to sort of understand what was going on in human beings, it was very modest. And what you've got here is professional guidelines from 3 different societies responsible for managing male infertility. And you can see that it's a pretty modest menu of genetic testing options that are available. Karyotyping, Y chromosome microdeletions, or CFTR sequencing for bilateral absence of the vas deferens or obstructive azospermia. That was the case in 2011, 2010, and it's still the case today. So there was this big gap, this knowledge gap of what must be going on that we knew from mice and what was yet to be discovered in humans. So it was in that context that I started this thing called the Genetics of Male Infertility Initiative, or Gemini, which had a very straightforward goal. We wanted to focus specifically on non-obstructive vasospermia, so those guys in the very extreme end of the sperm concentration distribution, because we figured more severe phenotype, better chance of finding something genetic. We were going to focus specifically on Mendelian causes. And because of the complexity of the sperm and the process of spermatogenesis, we figured we'd need a pretty big cohort. We were expecting a lot of locus heterogeneity. So we were shooting for over 1,000 unrelated cases because collecting families in this kind of clinical context in America was very difficult. So that was set up with Kai Aston, who's still my co-PI today. He was a postdoc at the time when I was getting started. He now runs the IVF lab there. I want to give a shout out and recognize some of the people that helped get Jim and I started here at WashU. So Sanjay Jain was really instrumental in helping me get my first patients locally here in St. Louis. He had contacts and connections into male infertility cohorts. Chris Gurnett, I don't know if Chris is here now, but she helped me get connected with a really good control cohort when getting sequencing data in those days was very, very difficult and expensive. I also want to recognize GTAC. I don't know if any of you are here, but my very first exomes that were made from Jim and I were through GTAC. Tony Sinwell made the libraries, Paul ran the analyses. I also work with Mike and Chris. So thank you guys all these years later. How have we done since then? We're now in our 9th year of funding. We're the only large-scale genetic sequencing of male infertility project funded by the NIH. We've got the numbers on here. We've recruited nearly 4,000 men from 10 countries around the world, sequenced a couple thousand cases. We've either led or contributed to this discovery of dozens of monogenic forms of male infertility over the last 8 years. So this is a kind of a scorecard of how the different disease models break down. We were just flying in blind. We had no idea what we would be finding. This is from a report of the first 1,000 cases that we published a few years ago. It's largely recessive, autosomal recessive, a little bit of X-linked, and kind of surprisingly, maybe counterintuitively, not so much Y-linked, because at the time, when we were getting started, the Y, its role in spermatogenesis was so, central into its biology. But in the big picture of things, it seems to be a relatively minor contributor. And I'll caveat that with looking at single nucleotide changes in indels with exome sequencing. You'll see that we get an overall diagnostic yield, and I mean that in a research context of identifying plausible monogenic causes in about 20% of cases. That comes with a false positive rate. When we do the same kind of analyses, run the exact same data generated from the same platforms to the same analysis pipeline, we get a 6% hit rate in people that aren't thought to have azospermia. So we may have something like a 30% false positive rate with this kind of approach. Some of the key observers might be looking for the dominant disease model. We did look for this, but we don't see a statistical difference in heterozygous damaging mutations and genes related to sperm production and cases versus controls. Intuitively, maybe that really is biologically what's happening here. It's kind of hard, since fertility is the phenotype, it's hard to kind of think about how dominant forms would get transmitted. It also could be it's just a hard thing to solve, and we just don't have the right analysis approach for picking these things out right now. Like we had guessed ahead of time, locus heterogeneity is a really defining feature of this trait. I'm showing you the top most recurrent hits from those first 1,000 cases. We have a modest number of 5 different genes that have been recurrently hit in 3 different cases. All in all, only 12% of genes are seen recurrently. We had another 21 genes that were identified twice, and then the rest of them are singletons, n=1 cases with these first 1,000 individuals. I do really believe that this type of recurrency is a good statistical sign of having the right targets. I'll come back to some of these genes in a minute. PNLDC1 and TDRD9 are pyrRNA pathway genes. Nobody's surprised that we're finding these things at all. They're very well established in flies and mice as being involved in male fertility. They control transposable element and genome movement in the germline, as well as genome integrity. And we'll come back to some of these other ones. But it raises a puzzle now, like how do we make progress on a research front in understanding this huge grab bag of n=1 observations from all the rest of the cases? So we hypothesized that single cell expression profiling of the testes may allow us to group genes together in a sort of pragmatic, objective way based on co-expression. And this leads to the next kind of element of how WashU is formative in getting our research going. We're very keen to adopt single cell sequencing as soon as Steve McCarroll's group published the DropSeq paper in Cell. I guess that would have been around 2015. We had been, up to that point, laboring in flow cytometry, trying to purify different subtypes of testicular germ cells. So we knew, getting these kind of separations in a high throughput manner with DropSeq would be a real game changer for us. And so Jeff Milbrandt, who was the chair at the time, helped organize a single cell working group involving Rob, Joe Doherty, Samantha Morris, and other people that were interested in importing this technology. It was a really exciting time. This is before 10X, so we built our own DropSeq apparatuses and launched a number of related projects that involved single cell sequencing. So as part of that wave of projects, we completed what I believe is the first mouse testis single cell RNA sequencing atlas. This was a project that was led by Min Jung, who is a grad student at WashU, as well as Daniel Wells, who's a grad student at Oxford with my collaborator Simon Myers. And that's the quality of TISNI/UMAP data that we got back in the day with our own in-house DropSeq apparatuses. This is kind of a meta moment when we had Steve McCarroll come to visit to give a talk. This is him up in the Couch building, I think the 6th floor, taking a picture of our DropSeq rig that we had built back in the days. Okay, so the hypothesis that the single cell data would be useful for making sense of the N=1 cases turned out to be correct. This is a very information-rich figure that's condensing that into a few bite-sized bits. We use a latent factor method for decomposing our single-cell RNA-seq data. I'm sure just listening from the talks today, many people are familiar with these approaches. We use something called SDA. It's like NMF. There's a whole suite of different methods that do this kind of thing. This heat map is showing you the result of this clustering. Each column is a different gene cluster that we identified from our single-cell RNA-seq data sets. And we see when we look at genes that we were identifying as damaged in the Gemini cases, looking at how they load across these different clusters, they look much more similar to the mouse male infertility genes compared to the genes that we find damaged in population controls, especially in clusters that are expressed in early stages of spermatogenesis, which we might think to be especially important in sperm production. But to go a little bit further and to give you just a glimpse of how this information is useful and how we're using it, we can see that there's even within the clusters that are hit in cases, we can see heterogeneity. There's some clusters that have more genes than others, specifically like this one that I've labeled here that we call cluster 51. You can kind of go deep and understand where these clusters are being expressed throughout spermatogenesis, what are the genes that are loading on them, what are the functional roles. SDA51 is one that we recognize as being involved in DNA recombination repair. You can see some of the genes, I won't read them out here, that we find recurrently mutated or at least mutated once in Gemini. These are things that result oftentimes in defects in meiotic prophase 1 and lead to things like leptotene, zygotene arrest, pachytene arrest, this kind of thing. The components can be quite distinct. So here's one that seems to be expressed cyclically during the germ cell mitoses prior to entering to meiosis. You can see it in pseudotime, like the expression of the cluster going up and down cyclically. You can see genes like Ki67, some cell cycle genes, CDC45, CCNA2, et cetera. We've got the piRNA pathway. But we also have things that we really don't understand. Like, this is an exciting thing about this particular organ system, is that it's fairly understudied, and some of the steps are very difficult to study in vitro in the lab. So component 98 seems to be cycling during meiotic divisions. The genes that are really highly loading in this cluster are really poorly characterized. All we know is that they seem to be involved in homophilic cell-cell adhesion. Maybe one hypothesis is that they have to do with the incomplete cytokinesis that happens during most of the cell divisions during spermatogenesis. So to summarize this little bit, oh, you didn't start the clock. I don't get a beeper. To summarize this bit, what we're seeing so far in the Gemini study is that we have clearly Mendelian subforms, mostly recessive and sex-linked so far. Just doing some simple statistical modeling, Poisson process kind of things, we're estimating that there's maybe somewhere between 500 and 625 loci that are going to be involved when it's all said and done. We're getting a 20% discovery rate in a research context using exome sequencing with these cases. If you get more strict and use something like ACMG clinical criteria, the diagnostic rate is much lower. But I think that's just a matter of time of working up, evidence. For those of you who are interested in the long-term human genetics mission of linking genotype to phenotype, this is a really important patient population, and I think other reproductive disorders which have been understudied will be equally important. 77% of the genes that are coming out of here have no OMIM entry and have never been linked to any sort of other trait. I already talked about how the expression patterns can help define subforms, and there's a lot of, if you start thinking about it, there's a lot of nice pragmatic applications of using subforms in this way. For instance, designing companion validation assays to help clarify things like missense changes in these genes where you think you might have the right variant, but you're not quite sure. So I'm just going to spend like 4 more minutes, talking about what happens next. I moved my lab to the Oregon National Primate Research Center and Oregon Health and Sciences University in 2018. I have 2 labs now, one at the hospital, at the OHSU hospital that continues this Gemini work, and one at the Primate Center that focuses on rhesus macaque. And so I've thought carefully. It's expensive and difficult to work with macaques and for a lot of things that you might take for granted in other model organisms. But I've been thinking carefully about ways in which we can opportunistically jump in and do things. And I came back, my thought process led me back to this set of recurrent genes. And I've been focusing on one, FBOX 015, which if you PubMed it today, there's 21 papers that mention FBOX 015 ever. In the meantime, since we published this first 1,000 cases, a pedigree has come across my desk that also seems to be linking a family with male infertility to FBOX 015. This is a start-loss mutation, if you can read the annotation up there. So FBOX 015, if you start looking at those 21 papers, it has had its 15 minutes of fame in the past. It was the original IPS cell marker that was used by the Yamanaka lab to screen for factors that can induce IPS cells. In this original paper, they screened for 24 different factors and came up with the Oscar factors based on picking genes that had an ability to create FBOX 15 positive colonies. In kind of one of those interesting quirks of science, like within a year, a different author list out of the Yamanaka lab published a new, better marker of IPS cells, NANOG. They showed that NANOG positive cells looked more similar to ES cells in gene expression space compared to FBOX 15 IPS cells. Also, FBOX 15 positive IPS cells failed to form germline chimeras, but NANOG positive IPS cells could form germline chimeras. So maybe, NANOG is just a little bit earlier in the developmental, process than FBOX 15. But what's kind of like frustrating for me and like sad for me was that FBOX 15 homozygous null mice that Yamanaka generated were fertile, both males and females. So I didn't, I'm not the type of person to let things go easily, and so I kind of kept digging in this, and I started looking at the biology of what happens in those early developmental stages, especially around primordial germ cell specification, which happens around gastrulation, which is kind of a hard time to study, especially in primates. And I started to see that there's some pretty important anatomic differences in the way that embryos are organized around gastrulation, that rodents use something called an egg cylinder, whereas primates use a bilaminar disc. Also, again, very difficult to study, but it looks like PGCs, even though there are some shared transcription factors that are used in primates and rodents, there do seem to be some species-specific things like primates use that rodents don't and vice versa. And there might even be independent origins of PGCs, multiple independent origins of PGCs during primate development, whereas there's only a single origin in rodents. So I'm thinking, okay, maybe there's a out here. Maybe there is a way to reconcile these results of the fertile FBOX 15 knockout mice, but a infertile human, because I really do believe the human genetic data. So we have an amazing, I would say, world-class IVF lab that handles the NHPs at the Primate Center, and we started working to make FBOX 15 null embryos so we could test this hypothesis that maybe it's a primate-specific phenotype. And we started transferring embryos to recipients last year. The gestation time is about 5 months in macaques, but the time to puberty is like 5 years. That's a long time to wait for males, for males. So the voice of Rod Mitra came into my head and said, you need to do a cowboy run. So cowboy run is kind of like a fast and dirty, like gut feeling kind of thing. And I was like, well, if this really is a gene that's involved in germ cell formation, like germ lineage specification, maybe I can see it like early on. Maybe I don't have to wait for 5 years to actually wait for puberty and spermatogenesis to turn on. Maybe if I just let the animal grow large enough that it's easy to experimentally manipulate the testes, I can see an absence. I mean, the home run would be a complete absence of germ cells, prior to birth. So we recovered fetuses at GD 143, which is pretty close to full term in rhesus macaque. And this is PAS staining. It's kind of like H&E, but we use something we use commonly for testes, showing control I guess this is shining up here, showing control versus the FBox 15 knockout. These were images were just generated like weeks ago. I was hoping to have the home run result for you guys. I would say grossly, in my expert opinion, they don't look different. It is difficult to see at this stage of development. These tubules are mixtures of somatic cells and germ cells, and it's difficult to tell which is which. So we're staining with DDX4, which is like a VASA MVH germ cell marker homolog. So I'm still clinging on to hope, but it also seems like that might be the case that the phenotype is more complicated, and we're gonna get the same result as the rodent. This is my last slide before I wrap up. We're also using this expertise and time-mated breeding resources that we have at the Primate Center to obtain prenatal tissue from all 3 trimesters to generate atlases of spatial RNA sequencing data as part of the NHP DGTex project, which we're leading along with the Broad and MIT. And I'll finish with what I call future opportunities. I mean, they're future directions for me, but they're opportunities because I think anyone, if the young people in the audience are thinking about things to do, they're things that everyone can participate in and contribute to. On the translational side, like we really need to work hard now moving our research results into the clinic, and actually that we've started a ClinGen group to actually sort of bless the variants that we're identifying with clinical grade annotation and knowledge so they can start being used clinically. There's also a real interesting opportunity to start understanding genetic comorbidities of male infertility. Men that are diagnosed with male infertility don't live as long as men with normal fertility. On the basic side of things, there's tons of really fascinating, bizarre, special biology, cell biology, and genome biology that happens only in the testes. And we're looking at the role of prenatal development, as I just mentioned, in primate-specific genes. So I'll finish and acknowledge so many people, many of them came through the lab here at WashU, collaborators around the world, and I will finish with a few images and wish everyone a happy 50th anniversary here at WashU. Thanks. It's fine, it's totally fine, yeah.
So please head out toward the stairs in the FTLC so we can take a picture. And you can grab Don along the way and ask him any questions you have.
How long was it?
It was 4 minutes over.
Are you serious? All right, sorry.
It's okay. I also have it on my watch.
Okay.
Hello everyone, let's start. Okay, so next speaker is Dr. Victoria Brown-Kennerly. She's associate professor of biology in Department of Natural Sciences and mathematics at Webster University, and she was a postdoc at Mark Johnston Lab. Welcome.
Okay, is this working? Good. Okay, so thank you very much, and I'm honored to be here. And this day is joyous, right? So we're all here celebrating a really well-earned stellar reputation of this department, of the genetics department here at Washington University. So I'm really thankful to be here, and I'm very humbled. So yes, my name is Victoria Brown-Kennerly, and let me see if I'm doing this right. Good. I am an associate professor at this primarily undergraduate institution, Webster University here in St. Louis. So I work with undergraduates. I teach undergraduates. I'm a teaching professor. We serve a wide variety of students, a wide population, diverse in all aspects, okay? And our student population has several students who come from under-resourced families. We have several first-gen students. We have students who may not come from the privilege of having ever held a pipette bin before they step on campus. So, this is our research subjects. So we do research on all kinds of model organisms in my lab. We do it at the undergraduate level, and we do a lot of research with ants. This is Pogonomyrmex occidentalis, and we know that because on the mandibles, that last little tooth there is giving me 2 thumbs up. And this is a picture out of my lab from some of our microscopes. So, I'm going to talk to you first about the ant, right? So you see this ant. This is Cataglyphus. It's a desert ant. This is also a picture out of the introductory textbook that I used to teach introductory biology about animal behavior, right? And so we look at this ant, and if you ever look at ants, like you're sitting having maybe a beer, it's common, and watching this ant, right? And she's going left, she's going right, she's up, down, she's in circles. I mean, it's a random walk. And I was thinking, oh boy, she's lost, she's a goner, right? But if you keep on watching, right, of this random walk, once the ant finds something, a prize, right? So she'll find a dead cricket, or maybe your peanut butter and jelly sandwich, right? The first thing she wants to do is run back to the nest and tell everybody. And when she does that, she's in a hurry, and lo and behold, she's not using pheromone trails clearly because she doesn't take this circuitous route back to the home, right? Back to the nest. She turns around and runs straight back to the nest. So what seemed like a lost ant, right? She was just on a random walk, had no idea what she was doing, and there was no direction or purpose, right? She was gathering data, she was navigating, she was pathfinding. And there are, at least 4 or 5 different ways that we know that ants take in sensory information from their environment to be able to figure their way back home quickly, back to the nest, okay? So we're working with undergraduate students, and I'm teaching students who are just beginning their pathways in STEM, and they start from very different starting points, okay? So the sad fact is that there is an attrition rate of students in STEM. So from the day they step on campus as a freshman to getting them to graduate, there's about a 50 to 60% attrition rate of students in STEM disciplines in our nation. So they may drop out of college altogether, that's about 20%, and the rest of them change majors. Okay, and so we're losing that pipeline, and we're losing a lot of diversity in that pipeline as well. So one way to address this is to get students involved. And what we do is we have these individualized undergraduate research experiences, or UREs. So where I'm at, these are required for every student and every major that comes through our program. Undergraduate research, okay? And if you can imagine, it's not based on GPA, it's not based on performance anywhere else. Every single student must do a year of research with a faculty on campus or with our collaborators off campus in order to graduate, and it's a requirement of the program. So if you just pause for a moment and think of the diversity of students in your introductory biology classes or your sophomore biology classes or any of your undergraduate classes, right? So this is a big task. And invariably, it terrifies the students. And it terrifies them, and it transforms them. So we end up being also their academic advisors, especially for those who are maybe first gen and they don't go home or have phone calls home around a table or with a support network that helps them work through obstacles, right? It's mentored, it's high intensity. We end up sort of being life coaches. And it's really difficult to scale. But this is what it looks like, is that we have independent research, which is a lot like grad school rotations. Independent research, you can take it for 1 credit. You can go into, pick a PI, work in their lab for 8 weeks, and you can do that up to 8 times. So you could have 2 full semesters and then 2 full years of some research experiences before you ever hit your senior year, which is fortunate because senior year, there are 2 semesters that are absolutely required. And the first one is research methods, where you partner up with a PI and do an extensive lit review of the question that you want to ask, write the proposal, including a budget, and then apply for funding. Okay? And then the next semester, that student goes into the lab or into the field and pursues that project, writes up the manuscript and the conclusions at the end of their semester, and then has to publicly defend their senior thesis. Every single student that comes through the program, right? So much like the ant, these students are finding their pathways, right? So they're gathering information, they're searching for resources, and they're finding their home in STEM, all right? So I do have to say that often it looks like a random walk in the beginning. Okay, so that said, this is an example of the first student-initiated URE that was happening when I stepped on campus at Webster University. And so I joined Webster 2 weeks before I had to start lecturing in 4 different classes. And after my first lecture in biochemistry, Rabia comes into my office and says, I want to do senior thesis research with you. I want to do research methods with you. And I was so excited. And she was like, I want to study ants. We got to study ants. And so I had never touched an ant in the laboratory before unless I was trying to squash it, right? I had no funding. I had no syllabus for a couple of my classes yet, and we had to start in on this research project. So what you see here is basically, Rabia was really interested in how ants do this innate behavior. So they do this innate tunneling behavior, and they'll do it in all kinds of substrates. And this person was ecology-minded and wanted to understand how contaminants in the soil and water are affecting invertebrates and ants and their behaviors. So what you see here actually are the old school CD cases, the big ones, the thick ones, not the slim ones. We ripped them out, burned holes in them, and then bought kilograms of the ant gel from like Uncle Milton's ant farm. And so Rabia made literally like throughout those 2 semesters, I would say hundreds of these small ant farms or ant gels, right? And so they're individual replicates, and would load each one up with 10 ants. We got ants from a biological supply company. Those were the pictures that you saw on the first page. And watching these ants tunnel, and then we could look at what the contaminants would do over time. And so what you can see here is the tunneling, so we can quantify them really easily because these are 2D, not 3D. And we can use NIH image, so very straightforward. And so what you can see is as the herbicide was increased, the tunneling decreased. And so that was really, really cool. And so I didn't believe it at first, and I assumed that they were using milliliters, not microliters, when they were spiking contaminants in it. So this was reproduced again and again and again, and it was very robust, and it didn't matter what type of ants we used, it worked. So that was a really great senior thesis. We could do a bunch of statistics on it because you can get literally hundreds of these individual habitats. And it was a great success. And then another student looked at it and said, well, that herbicide is pretty complex. And so she went into the literature before everything got taken off the interwebs, and she figured out all of the individual chemical components of this herbicide and tested them all individually in this assay, and she found the culprit. So this polyethoxylated amines, this surfactant, right? And so then another student comes through and is interested and says, well, okay, so what are they doing here, right? They're not dead, they're just sitting there. So what's going on in their noggins? What's going on in their brains? And so this student, we worked out a protocol to purify mRNAs from the brains of these ants, and this student looked into the literature and found some candidate genes to look at gene expression. And lo and behold, this is actually one of the herbicides, and the herbicide, when the ants are exposed to it, increases expression of the ant homologue of the HTR1B serotonin receptor. And so this student figured out how to study this gene because previous studies had noticed that when ants start to forage, which means pathfinding, and in that developmental pathway in their lifeline, the serotonin is increased and the serotonin receptors are increased in their brains. And so this was a really, really interesting. So this story is not done, but this is just an example of one of the experiments in my lab. So we literally have dozens. And so then we have another, a course-based undergraduate research, which increases persistence and belonging in STEM. And all students in the class gain experience. So this is more scalable. So during the curriculum then, if we offer many different ways to get these undergraduate experiences, every student gains an authentic experience prior to graduation. And this is us at the NCUR meeting in Memphis. Another cure that we're doing is the C-phages. And so this is, oh boy, Science Education Alliance Phage Hunters Advancing Genomics and Evolutionary Science. That name and the funding and the support is brought to you by Howard Hughes Medical Institute and Graham Hatfield's group at the University of Pittsburgh. And it's a 2-semester cure where we isolate bacteriophages from environmental samples, and then the second semester is we sequence the genomes and annotate the genomes using some bioinformatics. And then the students can attend an annual C symposium. And so this is an example of one of the posters that my students took to this meeting. So we leverage that, and now we run a phage camp for high schoolers, and we serve about 24 students a summer now with me and Dr. Mary Proust. She's my colleague there. And the great part about it is that our undergrads then get teaching assistant practice using their cure-acquired skills to mentor high schoolers, right? And so they really have a sense of ownership. They name their phage, they put it in the national database. We get pictures of it at the Molecular Imaging Corps here at WashU, and it's been a really great experience. And that was funded in part by Bayer Corporation. And so the final cure I'm going to talk to you about is DNA barcoding, and this was just an off-the-shelf, like molecular biology kit thing that we could do in our genetics labs. The students went out, collected a bug, named it, photo documented it, put it up in iNaturalist to get a hypothesis about its ID, and then we would lovingly crush up our bugs and PCR up the CO1 gene, and it's a DNA barcode that can specifically identify insects down to species. We'd send it off for sequencing, and then we'd spend another lab week in the bioinformatics. Okay, and so this is the process that you see there. And so Jeffrey Petraka at Cold Spring Harbor Lab, I called him up and I was talking to him about it and somehow convinced him to come to St. Louis. So I recruited him to St. Louis to come and train folks at the Missouri Botanical Garden and the Earth Waste Center, who then, with me as a collaborator, set up the Citizen DNA Barcode Network and had a series of bug lab community science events. And so this is us, there's our team, there's the community science lab events, here's some of the bugs. And again, our undergraduate teaching assistants could practice their Cure-acquired skills as science interpreters for citizen science projects and contribute to a national project. So as I looked at this then, I started to have a little bit of an existential crisis because this was eerily familiar. And in fact, it kind of mimicked my career trajectory. So, you know, I was rural, I grew up, I collected bugs, I talked to wildlife, I was that quirky kid. And I went to University of Michigan as a French major. And then I just quickly realized I loved biology and changed my major to biology. I worked with Jeffrey Chamberlain, he's now at UW, on Duchenne muscular dystrophy. It was fantastic. And so I was retained. I persisted. I was first generation college. So I went to Emory University because I liked it so much. I went to grad school and I studied immunology, but pretty quickly I realized genetics was a love, and so I worked with Stephen Warren on Fragile X syndrome. And then I went all the way over to the West Coast in the Genome Sciences department and studied toll-like receptors at UW. Do you see how this looks like a random walk right now? Right? But what I can say is I worked on all kinds of different stuff, but I worked with great mentors. And then I came back to WashU to work with Mark Johnston on Candida albicans and genetics. Right? And then Mark went to Denver. And so I had just gotten here. So I searched for resources and worked at Sigma Aldrich with Xiaoxiao Kiwi, who now is the director of the stem cell core here. And then went back to WashU and worked on the Diane project and 16S profiling. And then finally came back to Webster University and I work on ants, phages, and barcodes. And really, that has really dovetailed and it's kind of brought me back home to my roots. And so that's Dr. Miller-Strutman. We're working on eDNA with her and with the zoo. And we're working on the BD fungus that decimates amphibians. And we're working on the spring peeper frog project. Questions?
We'll let you ask Victoria questions at the reception.
Okay.
As you find your way.
All right, we'll move right along. It is my great pleasure to introduce Dr. Alison Albee. She was a postdoc in Susan Dutcher's lab, and she is now a professor of biology at Lindenwood University.
So thank you. This has been an amazing day. I feel like I have got to hear so much amazing science. But before I get started, I definitely, absolutely want to thank and acknowledge Megan Waltz. She is my co-instructor. I often refer to her as my work wife. We know a lot about successful collaborations in research, how two labs can coordinate to work together to produce amazing results. And I feel like I have that with Megan Waltz. We are both very passionate about undergraduate education, and she is a fantastic colleague that I can go to when I'm wondering about how I can best teach a particular topic in my class. And we can bounce off ideas with each other, and we're really able to better our teaching through this collaboration. So as you go out to teach in your own labs, I encourage you to also find your work spouse, to have someone to teach, to bounce ideas of your teaching off as well. The second person that I want to thank is I want to thank Susan Dutcher. She was an amazing postdoc advisor, and what made her so amazing as a postdoc advisor is that she let me pursue my passion, and my passion was teaching. I did the research because I had to, and I liked it, but my actual passion was my research. My actual passion was not the research, sorry. It was my teaching. And so as a postdoc, Susan let me become a glorified TA with Kathy Miller over on the main campus. And I was able, with Kathy, to teach 5 lectures of a developmental biology course. So I got to actually pursue my passion. That opened up other opportunities. I again went back to Susan and said, can I do some more teaching? And so I become an adjunct instructor at Harris-Stowe State University while I was still a postdoc. And I taught a biochemistry course there, as well as I also taught a microbiology course. So for all of you PIs, Susan was gracious and amazing to let me pursue my passions, even if they weren't research passions, while I was still a postdoc. And that experience was absolutely necessary and important for me to be where I am and to be the teacher that I am. I got to have great experiences. Also, as a biology educator, I'm also very used to reading a room. And today has been so amazing, but I don't know about you, but my brain feels a little full. So I'm not going to tell you about the talk that I was. I'm very happy to talk about it afterwards. Instead, what I want to tell you about is how you can be a better teacher. If you are maybe 30 or 35 or older, please don't teach the way you were taught. Please don't stand in front of a classroom and lecture the entire time. You will be bored as a teacher, and trust me, your students are going to be bored of you as well. There is an entire field of research that is called the Scholarship of Teaching and Learning. You need to find that and you need to read it, because there is scientific evidence how students learn best. And students learn best through active learning, participating in their learning, not passively getting information, but using that information. And so I encourage you to find how students learn best. WashU has an amazing teaching center. If you have not discovered it already, you need to go to the main campus and you need to go to their seminars. They will help you become a better teacher. The teaching center also has a teaching certificate that you can get. Again, Susan let me go to all those seminars to get my teaching certificate so that I got experience and I got trained how to be a teacher. If you do not seek out those opportunities, you will not be taught how to be a teacher. It is very different teaching undergraduates than giving seminars such as this. And so it will absolutely help you if you know how to teach before you get put in front of a classroom. The other thing that I wanted to tell you about is that there are lots of resources. If you are a beginning teacher, please do not feel like you are all on your own and that you have to come up with all of these amazing lesson plans. There is lots of places where you can go. I make use of case studies a lot in my classes. It helps the classroom stay active. The students are learning the information and they are immediately applying it. I write a lot of case studies myself. I have my students write case studies that they have to present to each other. But as a beginning professor teaching for the first time, there are a lot of places where you can go. The National Center for Case Study Teaching in the Sciences has lots of case studies. The EvoEd CourseSource is also an amazing place. They have case studies as well as other activities that you can do and incorporate in your classroom. And the last thing I want to tell you about how to be a good teacher is you are being trained right now how to be an experimentalist, how to come up with a hypothesis, design experiments, and then carry out those experiments and analyze your results. Don't stop that just because you get into a classroom. Experiment with different types of teaching techniques. Experiment with different ways to communicate information to your students. Gen Z gets crapped on a lot, but they are some of the most gracious and some of the most empathetic people that I have ever met. They will give you grace in the classroom. If you tell them, hey, guess what? I'm going to try out this new thing. You try it out. If it fails, but the students will give you grace if you come back the next day and say, you know what, that really didn't work, so we're going to do something different. the students are okay with that. They will trust you as an instructor to make mistakes. You just have to be vulnerable with them. They will give you the grace that you need. So that's really all I wanted to tell you about, is how you too can learn to be a good teacher. Thanks. I don't know if anybody's got questions, so yeah, sure.
Hello, everyone. It is my great pleasure to introduce Dr. Christina Gurnett, who is a professor of neurology and director of Division of Pediatric and Developmental Neurology at Washington University. And she was a fellow in the lab of Dr. Ann Bocock. Thank you.
And Juan, this is it's a real privilege to be here with such distinguished former faculty members and trainees. I'm going to tell you an interesting story, I think probably the most interesting in my career, and anytime you can include a manatee in your talk, it means that you're living on the edge and have a little luck. I had a lot of fun working with Ann during my postdoctoral fellowship. And I'm a neurologist. I take care of kids with epilepsy, and I will say that we use genetics every single day. I cannot take care of a patient with epilepsy without having their genomic data on a clinical test. So we are using this absolutely every day in the clinics. But the story, when I came to Ann's lab, I had a background in my PhD was on voltage-gated ion channels, but I was very interested in identifying some of the causes of neurodevelopmental disabilities. And so I work on clubfoot, which is 1 in 500 kids are born with this condition. It can be treated, although in certain parts of the world it's not. And you can see a child on the very left, the feet are quite deformed. But now we do a fair amount of bracing in the first 5 years of life to get the foot back into a normal shape. This actually ended up being a family field trip down to Springfield, Missouri. So that happens to be also the home of Bass Pro Shop and so we recruited a very large family with clubfoot, which is how this story came about. Turns out King Tut also has feet that at least by x-ray look like probably had clubfoot. So you find this everywhere. And a lot still to be discovered. So back in 2004 when I joined Ann's lab, the state of the art for linkage, I really came and brought some of the families into her lab. She was working on a couple other neurologic problems, including paroxysmal kinesiogenic dyskinesia with a friend and colleague of mine, Louis Tachek. But she obviously has a lot of expertise in genetics, but more on skin conditions. I came and brought a lot of my patients with epilepsy and other neurologic problems. But this was very painstaking, as the history of science here shows. To do linkage back then, we had to use microsatellites, and it was a lot of effort to get those microsatellites so that you could eventually do mapping across this family. And so in the middle, you'll see the pedigree. You can see it's not complete penetrant, so it looks fairly dominant, but there are a couple individuals who had no problems at all with their feet. My little patient on the left had a much more severe clubfoot as well as polydactyly of the right foot. He ended up having, as you can imagine, with tibial hemimelia, also his tibia was very small, ended up having amputation. So my kids all came along, they played with him, they didn't understand why he didn't have a leg. So a fairly severe condition for this. We were able to get the photos actually of the grandfather of this child and also had clubfoot that was fairly well treated. But this was the family we did linkage on, and lo and behold, we discovered a variant in a gene called PITX1, which is a transcription factor that's very involved in pituitary development, as well as uniquely hind limb. So if you look at the expression in a mouse embryo, it is only expressed in the hind limb. It's not expressed in the hands at all, which kind of an obvious thing if you're studying clubfoot, if you find some genes that are really restricted to hind limb. So this kind of put us on our first path of understanding some of the pathway involved in clubfoot. Unfortunately, this gene really only accounts for a small percentage, maybe 1 or 2% of kids with clubfoot, may even be smaller than that. You know, we haven't so much talked about the history of all the different genetic tools that were available, but in 2004, 2005, just started doing microarrays. So this was kind of the area of copy number variants. I have plotted on here, I take care of a lot of kids with intellectual and developmental disabilities. Back then, we thought a lot of this was recessive. The whole concept of de novo variants was not on most of our radar screens. We were thinking that all these individual sporadic cases were probably recessive. But the state of the art around 2003 when I joined Ann's lab was, fortunately, the Genome Center started doing 10K microarrays, so immediately I wouldn't have to do all of the microsatellite repeat analysis, and very quickly we could look at copy number analysis. And in my field of neurogenetics, obviously this was a tool that really took off because we were finding these recurrent and sporadic different size deletions for autism, intellectual developmental disabilities, epilepsy, and schizophrenia. And I still think this technology of looking for copy number is less well applied to other birth defects than some of the neurogenetic conditions. But this opened up the possibility of identifying lots and lots of variants, including we found a deletion, a large deletion of the entire PIDX1 in a small family. This is David Alvarado, who was in my lab, who's now at Merck. We were able to find other deletions of another transcription factor called TBX4, which is also a hind limb restricted transcription factor with both duplications and deletions that were involved in clubfoot. And then, you know, I guess I'm giving my age here by putting a cartoon in here. But as we started filling in this pathway, a lot of this was transcription factors involved in early limb development of the hind limb, as well as SHOX genes. We found copy number variants in SHOX genes. So Jillian Buchan was a grad student. Ashley Quiggle was postdoc in the lab, and Brooke Sadler did a lot of copy number analysis. And on the left, it's the dog sitting on the couch and says, well, if I have to be honest with you, they're not really accidents. And so the pathway became a little more clear as to what we were finding. But the really interesting part of this story, and about the time that we published this, David Kingsley gave a talk and happened to show a lot of the work that he was doing and looking at development and of the stickleback fish and looking at the evolutionary loss of the pelvic structures in fish when they go from a marine environment to a freshwater environment where there's less predation, probably less calcium. And so they were able to show that PIDX1 was almost always lost. And the manatee came about because around the same time, people were discovering skeletons. When manatee die in Florida, you can pull these up. And if you looked at the pelvic structures of them, they often had this right-left asymmetry where the right side was much smaller than the left. Now, if you know the story of manatee, they were land-walking mammals who went back into the water. And it was an evolutionary advantage to lose your pelvic structures and be able to swim a little bit better. And so he was able to map out the right-left asymmetry in these skeletons, and again, had some evidence that PIDX1 was probably involved in that evolutionary adaptation, as well as all of the data that he had already compiled for stickleback fish. More recently, in the last 5 to 10 years, people have actually identified that it's not the loss of the entire PIDX1 gene that's involved in the evolutionary advantage, but it's specific hind limb specific enhancers. The pituitary specific enhancers are not involved, but there are hind limb specific enhancers. The first he identified was unique to the fish lineage, so we couldn't study it in our human patients. But more recently, they've just started identifying some other more conserved regions that are, in fact, conserved in humans. And so this is an experiment that he was able to do to take that hind limb specific enhancer, re-express PIDX1, and the pelvic structures come back in some of these stickleback populations that he has. So we've just actually sent, I think we have 30 or 40 genomes at the MGI right now, and so we're going to be looking at these non-coding enhancer regions. So it's still driving what we're doing more than almost 20 years after we found the first linkage to this gene. So I just wanted to end with that story. We're doing lots of other things in my lab. In particular, I love the work that Brock and many people in this room are doing on deep mutational scanning, because that's where the other bottleneck is interpreting what these variants do. But I thought I would share this story and share, the joy of doing science, and it comes full circle, and the many people that you touch. And I absolutely thank so many people in this room. You've been great collaborators. I guess the one last slide about this same story, that we did go one step further, and Kevin McCall, he's a lab manager that's worked with me for, I think, 15 years. We got the mice in the lab, the PITX1 mice, and these had been studied for their role in pituitary. No one had ever noticed that about 10% of the haploinsufficient mice, you can see here, have a little clubfoot. And so Kevin noted as we were breeding these that, wow, some of these have clubfoot. And then Kevin and then Gabe Haller, who now has a lab here in neurosurgery, he did some of this work as well. I think we were the first people to ever put a mouse in an MRI scanner, and to do micro MRA, looking at the blood vessels, and we could clearly show there were developmental field defects where all of the muscle, everything was smaller on that side. And actually it does come full back to the clinic, where now we do electrical stimulation to the muscle that's there, because we know that it's smaller in patients that have this deletion. So lots of fun, lots of great collaborators, and I congratulate the whole department on their success in 50 years and look forward to the next 50. So thank you very much. Okay. I'd be happy to take questions while.
She has questions. I have to get things out.
Okay.
Hi, very cool. Am I here? So, for the PTX1, have you also identified whether it interacts with other transcription factors that are also known to regulate limber development, like Hox genes and things like that?
We have, yes, there's definitely interaction between some of the Hox genes that PITX1 will actually activate some of the Hox genes, and we have found Hox genes that are also involved in clubfoot.
Cool.
Yeah.
Thank you.
Great. We're okay. Okay. Thank you.
All right, I have the very special honor of introducing today's this symposium's final speaker, Dr. Jeff Milbrandt, who actually doesn't need any introduction. Jeff is the James McDonough Professor of Genetics, the Executive Director of the McDonough Genome Institute, and also Jeff is by far the longest serving chair of WashU Genetics. Welcome, Jeff.
Thank you, Ting, and thanks for the invitation to tell you a few things about what we did and what we're doing that hopefully will impact the medical center and especially the realm of genomic medicine. So as Bob and Eric related to you, we were the center of the Human Genome Project. There were others, including the villainous Craig Ventnor, but we were front and center. And like any great party, and it was a wonderful celebration, a party, there are those who go through the whole night and are inspired and stay awake all night and they're ready to go the next day. And there are others who, well, they have a hangover. And we had some of both. And so in the end, we thrived, and we're going to tell you about some of those things that we did. So after Bob left, Mark became
the interim head. And he and Jeff Gordon had an early attempt now to leverage this new genomic insights from the genome to solve biological problems. So this was the Center for Genome Sciences. It was populated primarily by genetics faculty. And there were good things and bad things about that. It contributed to the diaspora of the department. But it was really a great organization. It's been wildly successful, and as you see, celebrated its 20th anniversary a few months ago. So upon becoming the head of a department at WashU is not just responsible for the department, but historically, they have been shepherding the discipline of which they represent. So in this case, genetics. I took that seriously and worked hard to make sure that all of our genetic endeavors would be impactful to all departments, centers, institutes, what have you, the hospitals themselves. And these are sort of the things that we wanted to try and do. We knew, and Eric mentioned it, that this would eventually impact medicine, so-called genomic medicine. We wanted to be a part of that. To do that, we wanted to focus on interdisciplinary studies, and then we wanted to pursue recruitment that would be joint with our clinical partners. And we did that. Another quote I wanted to bring up. So I didn't make this up just now. This was when I started, which was 2009. So this was George Bush's economic crisis, not Donald Trump's. But we had to go through it again, and there was great stress in the system, exactly the same things, NIH budget, medical care reform, and a huge problem in the stock markets. So those were all the uncertainties, but what was certainly true is that there were vast opportunities opened up by the genomics that was done here and elsewhere, and we wanted to take advantage of that. Now, one other problem we had was that we had a small department, usually about 20 people, 22 when you count Debbie and Sherry, who were absolutely critical to the success of the department. But we had those 20 people spread out into 4 or 5 buildings. You can imagine that created some problems with communication, with collaboration. And so we negotiated with the dean to get us a new home. And that home is the Couch building. And that was built, I think, 2013, 2014. It's a spectacular building. Most of you are aware of it. And here's our benefactor, George. George loves, he loves ribbon cutting because he gets it. But then he also loves genome engineering. I don't know why, but he always loved genome engineering. Now he's really into AI. I think it's because he bought a bunch of NVIDIA stock. But at any rate, George is really into this sort of thing. And so he donated money for the building. Now, as we move through this, our genetics department is at a medical school. And I thought this was important and it should shape our work. And so we encourage focus on fundamental science of disease mechanism, and that's reflected in some of the people that we hired. Often joint appointments. I put Ting up here because he wasn't a joint appointment, but it turned out okay. I mean, it turned out well. And then Joe was a joint appointment with psychiatry and Don with pathology. And you've heard from at least Don. And so those joint appointments continued throughout the time I was chair, and we have quite a few. I don't have time to go through each one. But the other thing we did that I thought was really important is that we needed to be better informed about what the clinical problems were. If you were going to try and use the power of genomics and all the expertise and equipment that we had assembled in the department, it would be good to know what some of the clinical problems were. And the way to do that, especially if you've never gone to medical school, and even if you have, is to actually brush shoulders with people who deal with those problems day to day. So we started embedding people, Albert and Hiroko from neurosurgery, Gabe eventually from neurosurgery, and then Sid from ENT, to be in the department as full-fledged members in terms of using all the common equipment we had, going to faculty meetings, et cetera. And I think that paid great dividends because they are highly successful, and I think it influenced people in the department as well. Now, all this is great. We want to do this genomic medicine and everything. We had one huge problem at WashU. And I know you're not really going to believe it, but it's true, that 98% of the people at Washington University, despite the biggest sequencing center in the world, was in our department. 98% of the people of all of WashU could not sequence DNA in 2009. And so we wanted to build a new facility to promote unfettered access to all aspects of genomic analysis to any investigator who needs it. So they have the tools to do that. Anybody that could pay, and even some of those who couldn't pay, were given access to anything that we had there. There was no priority given to anybody. It was all first come, first serve, and that's the way it is to this day, and I think that's served many people well. So here's so Eric, I hate to pick on Eric, but he's talking about naming things, and they're having some meeting in the middle of the 8th floor of McDonald's. When we wanted to name something, we went to a bar. And so this is Scottish Arms, and we had to name this facility. So we knew it had to have these 4 letters, but how were we going to put that all together? And a few drinks helped, and then Michael Brandt came up with Genome Technology Access Center, which is still what it is today. And Rob, well, that's what he looked like before. So that's Rob, and he was instrumental. And then I'm forever grateful to Pfizer. Pfizer continually tries to reform themselves and gets rid of people who are fantastic for no good reason. So we were able to recruit several of those people. Karen Seibert, my late Karen Seibert, my dear friend, and Rich Head, who really played a major role in getting GTAC off the ground. And they'll come up again in a minute. So you see Scottish Arms is back. So if you didn't know that, here you go. If you didn't even know it was closed, shame on you. So we had GTAC now, and Eric had mentioned that the fruits of the Genome Project were hopefully going to be genomic medicine. And we started that, we had that idea, and we started that very early. And you see this, Karen wanted to launch this at a propitious moment, 11/11/11, only comes up once every 100 years, right? So she thought that was great, and whatever Karen wanted, Skip and I just had to go along with. It's a Skip version. And so we joined forces with pathology and genetics to form this Genomics and Pathology Services, whose sole mission was to do clinical genomic testing for patient care. And it was very successful for in a lot of respects. But one thing I learned, and I should have known at the time, is that if you start a new business, we all know that it's really bad to be too late to market, right? The competition's already there, it's very hard to dislodge them. It's also bad to be too early. And we were too early. We were out in front of almost everybody, and we had all of these companies coming in and seeing what we did, actually using it as a blueprint to go out and raise capital and then compete against us and basically take our market. So what happened was that we were not able to get reimbursement from Medicaid nor from insurance companies. It was very hard to do that. And so there was a lost decade. Now it's revived. And so MGI is doing a lot of clinical genomic sequencing, including the rapid whole genome sequence for the patient's newborn intensive care unit. So we turn that around in 24 hours. So the turnaround at the sequencing level is faster than it is to get the sample from the baby and get it down to the MGI. So within a few days then, the sequence is interpreted. There's been a number of cases, and Chris could probably tell you about some of those, that have been changed, the diagnosis has been changed, or the treatment or non-treatment has been done because of this very rapid sequencing. So not everything in genetics is of course, DNA sequencing. There's much more than that, right? And so with the invention of the iPSC technology, it basically rescued Missouri from itself, from the throes of the stem cell debate that we were having, and allowed Washington University to use some of that technology to model human diseases. Other than that, other than iPSCs, we would have been shut out for a while. So we wanted to start this, and you all know now how this goes. It's a skin biopsy, and eventually get pluripotent stem cells, et cetera. So how are we going to do that? Well, Sherry Moore came to me one day, not long after I'd become chair, and said, you know, we kind of own the Tissue Culture Support Center. I said, what? You know, what do we have to do with that? You know, it's a historical fact. It was made in the 80s or 90s when people didn't know how to culture cells very well. And so that's what it was, tissue culture support. And you could, well, that, of course, had been long gone. That market had vanished. And so that equipment was sitting there. So we had this idea like, oh, maybe we can just repurpose this equipment in the basement of the old dental school and create an iPSC center. And that's what we did. And so that was working. It was going okay. And then I got a call from a person at Sigma, Chandra Miller. Chandra is a force of nature. And she called me up and said, you know, I'm doing this zinc finger nuclease stuff at Sigma, and for gene editing. And it's sort of working, but I'm pretty stifled here. And there's this new technology called talons. And, you know, what would you think about me leaving and coming to WashU and helping you set up a center for genome engineering, and we could do all this talon stuff? So I said, okay, that's great. Okay, so we went to dinner and we solved this, we got it all arranged. And the next day, literally, comes this paper with this weird protein called Cas9 CRISPR. And we hadn't really invested much in talons. And we see this, and so we quickly humanized the Cas9 clone. Little did I know that we should have patented it, that you could have made a lot of money just off of that simple. That was a big mistake. But we humanized that clone, and then we never looked back. We banished all of the other things, and we started off with CRISPR. And so Chandra was great, and then she shocked me and said, well, I want to go run my own lab at St. Jude's. I couldn't talk her out of it, but it turned out to be one of the best things that ever happened to me, because shortly thereafter I was able to recruit Sasha Kui from Horizon, another industry transplant, who has taken this center to unfathomable heights. Really, the discipline of being at a company, of focus, of finance, et cetera, made it into a world-class facility. And we actually do more iPSC mutant line generation than any place in the country, commercial or academic. It's really quite amazing. So now the department had grown and we were doing fine. And then there was a change in leadership at the McDonald Genome Institute, and the dean asked me eventually to take over as executive director of the MGI. Now this was a real stretch. First of all, I'm not a genomicist. That was number one. Second of all, I already had a full-time job. And there's a third. The third thing, and I don't want to pick on Eric, but he's such an easy target sometimes, you know. So Eric was the head of the NHGRI, and we were always getting $30, $40, $50 million a year to do these major sequencing projects at the MGI. Well, it was only rational and inevitable that those would begin to sunset. And that was happening right when David asked me if I wanted to take it. So I'm not a genomicist, I have a job, and he wants me to preside over a diminishing enterprise. That doesn't seem very wise, but it's a fair question. Why would you want to do that? But I'm glad I did it, and together with Rob Mitra and the leaders of the MGI centers, we transformed the Institute into a hub for personalized medicine. And I'm going to tell you about a few of those things and the impact they've had on the medical center. So we have 5 centers within McDonald Genome Institute now. It starts out with GTAC, which is the genomic analysis that we've discussed, and the GESC, which is the engineering we've discussed. And then one of the more recent things is MTAC. So this is mass spec. As Mark Johnson told me last night, oh, that's one of those protein things, you know, you use for some protein thing. So it's mass spec. Yes, you can do proteomics, Mark, with that. And then we have a center that's for cellular phenotyping. So this is imaging, AI of imaging for CRISPR screening. We can do all kinds of machine learning to do analysis of images. And it was spatial transcriptomics. Of course, this is becoming ever more important for us. And then finally, the latest addition is the Center for Translational Bioinformatics, and Rich Head is now going to head that. And its role is to take all of the clinical record information from the BJC system, so that's about 7 million lives, and couple that with all the preclinical models of disease that we all know about in the public record, and then couple that with any kind of omics generation that we do at MGI. So all those samples are tracked. And then it's associated with a biostore. So we have a new $5 million robotic freezer that can store 6 to 10 million samples of either patient DNA or patient serum for all types of clinical trials and biomarkers and that sort of thing. And then we do a lot of outreach and education. We're deep into community health, and we've also been doing a lot of biotech workforce development. This was started during the pandemic when we didn't have any workers and everybody was leaving to go make more money in industry, but we have had some success with that. But what we really care about is catalyzing the development of new treatments for disease. So not only are we trying to develop all of these centers so that anybody at WashU that's doing their projects can get closer to developing a treatment, but we're also doing some of that ourselves, as you'll see in a minute. So we have a few guiding principles that I think are really important. Bob talked about principles of free access. We think it's important that the MGI have principles on how they perform. So we try and perform and support world-class research, helping to translate and do clinical work. This is really important. We celebrate the talented and dedicated team of MGI. So Eric mentioned Bob Fulton. So Bob and Lucinda and many others have been at the MGI for decades and decades and decades. And I can say that because whatever I say, I have more decades than Bob. So, but the respect and loyalty they have for each other and the fun they have working and what they do to help all the investigators at WashU is really extraordinary. We tried to be very inclusive and collegial and let everybody have unfettered access to all of the things that we develop at MGI. And then finally, we are constantly, as long as the money holds out, which of course is a big question, we are embracing the rapid technological advances, either that we make ourselves or that other companies make and try and sell us their fancy gizmos. And so we've been very lucky in being able to finance most of the most up-to-date technology, and we hope to be able to continue to do that. So I think the infrastructure and the expertise of the MGI was on full display for the medical center, for the undergraduate campus, for St. Louis and Missouri during the COVID pandemic. Within a couple months, we had implemented and actually developed from scratch a microfluidic saliva assay, and using all the expertise and all the equipment that had been developed over the years, paid for by some of those grants from NHGRI. You see lineups of PCR machines, and we had to shut down most things, right? And most of the labs were shut down, so there wasn't a lot of business coming in but we pivoted and did hundreds of thousands of COVID tests during that time. And that was a real tribute then to the dedication of the people and the expertise that we had accumulated over the decades. We also have time for some fun. And so we always try and spread the MGI word. We go to local high schools, the science center, with the Balloon Glow. We have a booth at the Balloon Glow, and here's another educational thing. So we have some great people who really love to do this, and they're extraordinarily good at it. So here are the technology hubs, and just a quick list of what we do, and the leader, so Rich, Sasha, Young, and Willie, and then Bob overseeing everything. And at the overarching then is the CTBI that I mentioned before and what it does in this center. So what are we gonna, so we let everybody use this and they can use it however they dream of using it. Well, what are we gonna do with it? How are we going to leverage that at the MGI? So the final pivot, at least during my tenure at the MGI, will be MGI as a drug discovery engine. So what we are really good at in academia in general, at the MGI in particular, is target identification. So what targets might be best used to treat disease? We're really good at that sort of thing. What we're not so good at, oh, and I should say that this is all well and good to be really good at targets, but targets don't have a lot of value in today's world, mainly because of patent law. You can't protect a target. It's a naturally occurring beast. And so what happens is academia discovers it with all their talent and money, and then they publish it, and then industry takes off with it because they developed a drug to it, and they develop a treatment, and they get the rewards. We're trying to stop that, and so we're going to become a drug discovery engine. There are already places at WashU that do the conventional drug screening, but more and more it's coming down to AlphaFold and the successors of AlphaFold that use AI modeling to identify pockets in proteins that you can target. And so we are now, I don't think we are capable of getting all the expertise at WashU to do this, so we are partnering with companies, and you probably see the news, there's an AI modeling company every week that's raising $1 billion to do this. So we're partnering with some of those companies to get initial compounds, and then you can go through and do medicinal chemistry and all, which we do well here, to eventually go through this strategy. And I think this commercialization strategy is what's necessary at any world-class medical center now, and we hope to lead the way at the MGI in that regard. So just briefly, what are we going to do? So we're trying to answer and use as a fulcrum a simple question, but it's very difficult to answer, and it's been known for a long time. If you have a Parkin mutation, that's a universal mitochondrial protein, as is SOD1. If you have a Parkin mutation, your motor neurons are fine. You don't get ALS, you get Parkinson's disease, so your dopaminergic neurons fall apart. Conversely, another ubiquitous mitochondrial protein, SOD, motor neurons degenerate and dopaminergics are just fine. So how is that? How can that be? So we think it has to do with homeostasis. When I was in Africa, I was amazed by the women carrying these heavy water jugs and being able to do this. And believe me, it is all women that do this. And they carry water for miles. And so let's take an example here. See, this woman, to balance her water jug, she needs 2 hands. Most of them only need 1 hand, so their homeostasis is a little better. And then there's a show-off here on the right-hand side who doesn't need to hold the water jug at all and can just walk down the trail. So she has great homeostasis. And how does she do that? Well, we think it's because of what we call resiliency. So what is the definition of resiliency? It's the ability to resist or quickly and completely recover from deviations from homeostasis. So for example, you have a windstorm, and this person can hold his umbrella perfectly still. He's resilient to whatever force is against him in terms of the wind. This guy is making it okay, but you can see he's struggling. So he's veering off. His resiliency pathways are not that strong, but at least he's still standing. And then you have this person. Now, his resiliency could be affected by alcohol or whatever, but whatever environmental stimuli he has, he's not very resilient. And we want to try and make those cells that are vulnerable more resilient. We want to make them more like this guy and not like this person. And so the hypothesis is really that selective damage of specific neurons in neurodegenerative disease is really caused by a change in the resiliency. That the ability to maintain homeostasis in one set of neurons to a given mutation or a given environmental stimuli or a combination thereof is different. And if we could identify those resiliency pathways and strengthen them, then that would be a clear path towards a new treatment. And that is the goal. And that's what we're trying to do. So in the end, what we want to do at the MGI is make a difference in human health. And all the people at the MGI have been dedicated to this for a number of, many decades. And we're continuing that, only pushing it more towards the drug discovery part. And so with that, I want to show you one more picture. So you see the title here. You see this impish sort of look? So this was only a few years ago, and oops, but he's got this carefree, impish look, and I'm sure he thinks sometimes. Now, David and Andrew are not talking about the financial crisis at this point. They are talking about how great it is that Ting got this endowed chair. Now the conversations are somewhat more weighty, and so I'm sure Ting, how do I go back to those carefree days?
It's time for our closing remarks from Ting.
Before you step down.
No, I stepped down a year ago. No, you're not gonna.
This is well, maybe we can step down. A little token of appreciation.
Oh, geez.
From the department.
Okay. Am I supposed to open this now?
It's up to you. I will.
You go ahead.
Yeah. So I'll go use the mic. Thank you very much, Jeff. And this is actually a original watercolor painting of the Altman Garden in Missouri Botanical Garden by local artist and Botanical Garden plein air instructor Mary D. Schmidt. I hope you enjoy it. Okay. All right. Yep. Do I have that slide? I'm just looking for that slide. Okay. All right. No worries. Well, I'll start. It's a long day, but what an amazing day, right? What an amazing symposium. No one really wants it to end, right? So I have prepared a 2-hour closing remarks, including Taylor Swift's as well as her cats. But Susan told me I need to wrap it up in 2 minutes. So let me get to the point, okay? It is a difficult time when science is under attack, okay? But we as a whole have always been a very, very strong community. And today, it's a celebration and a testament of our history, our legacy, and our commitment to the future. So let us continue to do what we believe in. Every day, there's going to be good news, there's going to be bad news, and the typical question of which one do you want to hear first, right? We've seen a lot of bad news out there these days, so maybe we'll hear the good news. For today, at least, Dr. Erica Green made it here in person, right? That's good news. I really just want to use my 2 minutes, my opportunity to thank for a lot of people. To begin with, I really want to thank Susan, Juan, and the entire organizing committee, Genetics Business Office, Michelle, Jonathan, for their tireless work over the past year to make today a reality. And today we have mic runners, we have photographers. Thank you all for making history for us today. So this is the organizing committee. That's the only slide I want to show. I want to thank, of course, all the speakers for sharing your stories and your amazing science. So everyone will receive your grading from Susan by this weekend. If you get an A, you also get some gift certificates from White Castle. But we're not giving out A's, right? So that means you're not doing enough research, so everybody's getting B's. even if you feel that you made a few mistakes when you were presenting, or you failed your QE or your thesis project get scooped, so what, right? As Mark always tell me, what are mistakes? Why are you afraid of mistakes? Don't be afraid of them. Embrace mistakes. And then he gives me a big hug. Thank you, Mark. As Susan mentioned, 14 of the 17 speakers today are former trainees. I want to specifically speak to our junior genetics PIs and our newest faculty members. Now, you have not been here long enough to have cultivated lab members to invite as speakers to this symposium. But you are the future of genetics. You define the future of genetics, and you are the force to protect the future of science. So keep it up and fight on. And finally, I want to thank you all for coming here today, for celebrating together a remarkable milestone, 50 years of groundbreaking discovery, collaborations, and progress in the Department of Genetics at WashU Medicine. We thank the pioneers who led the foundations, to the faculty and staff who nurture curiosity every day, to the students and trainees who carry the torch forward. You're the heartbeat of this department. We are ready for the next 50 years of pushing boundaries, embracing the unknown, and continuing to lead with purpose and passion to the next 50 years. So with that, I'd like to invite everyone to join me to the reception. There's going to be food, there's going to be drinks. We see plenty about beers and today and in this department, so it's sampling time. Now, I do think that drinking is probably not good for you, so please join me in giving up thinking for the next couple of hours. Okay, thank you very much.